Good morning.
Great, I can hear you, David.
Okay, good. Good.
All right. Welcome everybody. I'm doing a last minute sound check here, folks. So welcome everybody to the 2021 virtual Northeast Aquatic Plant Management Society conference, annual conference. We all wish we could be with each other in the same room, but obviously the world had different plans this year. So, but we do thank you guys for joining us virtually for this. It's going to be a little bit different. I want to welcome everybody. We have a great couple of half-day sessions planned here today, starting with our workshop today and then our general sessions for the next 2 days. It's obviously been a challenging year for everybody, and so I just ask that you have your patience with us. This is not our strength. We're all biologists and chemists. We're not tech gurus, so there's a couple of people that are behind the scenes running everything. So just if you have any problems with audio or listening or seeing anything, please try and troubleshoot those on your own. Send us an email or put something down in the question box, and we'll try and take care of that for you. I would like to thank all of our sponsors for the conference. They are listed up on the screen right now. We appreciate them. Everybody really stepped up this year, and even though we shifted to a virtual conference, they all decided to step up and support us, so we thank them very much. I would also like to extend a special thank you to Earth Science Labs for sponsoring our algae workshop today, so that we could offer that without an additional fee to all of our attendees. So if you're here for the algae workshop, it's because Earth Science Labs decided to step up and sponsor that. And we have Dr. David Hammond, who's going to say a couple of words on behalf of Earth Science Labs before we get things kicked off. Dr. Hammond is a VP of Applications Development for Earth Science Labs. He has 4 patents, half a dozen peer-reviewed publications, and is an environmental chemist consulting on a variety of topics. And he received his master's and his PhD in environmental chemistry from the University of California, Berkeley. So Dr. Hammond, the floor is yours.
Good morning. And are you seeing the first slide of a presentation?
Yes, you're good.
Okay. So this is a quick industry update, 5 minutes, and I'm going to use it to tell you about 5 little updates, 1 minute each. The first is a project that Earth Science Labs and Earth Tech has done this last year in Utah. It's at Utah Lake. It's a notorious lake for bad algal blooms. It's very shallow. It's 1,000 acres, but only 10 feet deep at the deepest. So it gets very warm and really bad blooms. It's never been treated before for those blooms. People have just suffered through them. And this last summer was the first year that they approved a treatment. And that little bay that you see there, a marina, is the area that they gave us to see what we could do to protect against the algal bloom, cyanobacterial blooms. There on the left is a photo when it got started bad this summer, and then post-treatment there on the right. And I've got a quick drone video that I'll show you. That's the marina after treatment. Before treatment, it was, dark green, like the area outside the marina. So successful treatment. No one's going to be treating a 1,000-acre lake entirely, but for high-value areas, this looks like something that they're hoping they can rely on. Sorry, I lost the full screen. Okay, the next project that I wanted to share about is some research being done in Minnesota through USGS and the Minnesota Aquatic Invasive Species Research Center. They're looking at low doses of ionic copper for controlling mussels. They treated a 180-acre bay called St. Albans Bay, the black square there. And the approach was to use a very low dose, 60 parts per billion as copper, so about 6% of what the EPA label allows you to dose. So just a 6% dose. every other day for 10 days. So 5 applications of this low dose, and the results were quite impressive. You see there the bars on the left, there's a light blue bar is the control bay that received no treatment. The dark blue bar is the treated bay, but the day before treatment. And then in the other 2 columns, there is no dark blue bar because there were no veligers found. So in the control bay, the veliger density increased, whereas in the treated bay, there were just no veligers to be found, so no bar there. Settlement was also completely eliminated. The light blue bar that you see is in the control bay, and it's a big bar because there was a lot of settlement, and there is no bar for the no dark colored bar for the treated bay because there just was no settlement. And they also got control of adults. Adult mussels were on the far bottom right where that little arrow is pointing. Those are the remaining adults post-treatment. I'm going to just race through this. They also looked at non-target organisms, and one of the most exciting things was that the native mussels survived the treatment even though the zebra mussels did not. We've always had to assume that, you know, if we're killing zebra mussels, we're probably killing native mussels too, and that's a shame, but you know, we got to control this invasive species. But this was the first evidence we have that native mussels, in this case they're fat mucket mussels, were surviving treatment that's toxic to zebra mussels. So that was very exciting. Next little update is a similar application of low dose to a portion of a lake to control mussels. This is in the Chicago area of Illinois. It's a 100-acre lake that we just treated around the perimeter, and it was not with the intent to eradicate mussels from the lake, just to knock them back from the shore. And in that sense, it was successful. Those 3 photos on the left show 2019 when there was just monitoring, no treatment. And then on the right, 2020, you can see that you know, visually they were knocked back a lot. There were fewer mussels and smaller mussels, 82% mortality of caged mussels, and about a 56% decrease in biomass on settlement plates. So that's exciting for going forward as an option instead of eradication, that you could just do low doses and, you know, much more economical. And then the next quick update is that Earth Tech and Earth Science Labs has expanded into Australia and South Africa. We work in the municipal water space, in the ag space, agricultural chemicals, and recreational water like swimming pools. And those are some of our distributors there, some pretty heavyweight distributors. So we're looking forward to big growth there, in particularly in Australia. We've already had some success stories with some of the larger utilities in Australia, like South Gippsland's Water, which treatment with Earth Tech allowed them to avoid using their desalination plant. It cleaned up their fresh water enough that they didn't have to go to the desalination plant. And Goulburn Water had been trucking water in from outside because their local water was so bad, and we helped clean up their local water well enough that they could rely on it. And again, a quick video. I'm just going to show you a few seconds. Oh, I think I'm ahead too far. That's pre-treatment, the algal bloom, green, and then day 7 after treatment. And then the last of my updates is about the New Zealand mud snail, some work we've been doing here in the US. Started with a project in Arizona. There's an Arizona fish hatchery that was contaminated with the New Zealand mud snail. This is similar to the zebra mussel in that it's very prolific, and they possess an operculum, which means they can close up and pass right through the gut of a fish. They could be eaten by a fish and go right through the gut of a fish and come out the other side. So they're a tough invasive species. We dosed at a low enough dose to be safe for the fish in this hatchery, which are rainbow trout. some of the most sensitive fish to copper. So we dosed just 30 parts per billion as copper. That's half a part per million as EarthTech QZ. And we fed that in, trickled it in slowly to the hatchery for 50 days. And at the end of the 50 days, actually took more like 29 days, the mud snails were all dead. This has been written up, and I encourage you to look up this paper if you want to learn more about it. But the quick overview is was about $5,500 to treat for 50 days at that 30 parts per billion. So thanks for giving us a few minutes to give you a quick update on what's been going on with EarthTech and Earth Science Labs, and look forward to a great conference.
Thank you, Dr. Hammond. Appreciate that.
I'll post this if people wanted to take a little bit more time. It'll be posted. You can run through it yourselves.
Great. Thank you very, very much. I think I forgot to introduce myself. My name is Chris Doyle. I am the outgoing editor of the Northeast Aquatic Plant Management Society, and I am the program chair. So, you know, we worked really hard this year to kind of come up with a pretty diverse, interesting program. We tried to grab some people from out of our region who wouldn't normally come to our conference, and our workshop is exactly that. I would like to just say that we're going to move into the workshop portion next. At 4 p.m. there is an optional live specimen viewing for the workshop. So I would like to thank the New Jersey Water Supply Authority and Solitude Lake Management for grabbing some samples in New Jersey yesterday and shipping them overnight to Michigan, to the lab where one of our presenters is at. I'd also like to just throw out there that at 5 p.m. we have an optional social hour, which will be a different link to the program than this one. So if you guys are still, if you're not all zoomed out or all you go to webinared out by 5 o'clock, please come over and join that. Solitude Lake Management will be presenting a modified version of the aquatic plant quiz that we typically do. That will be a web-based application. So with that, I would like to once again thank Earth Science Labs. I would also like to thank our two presenters, Dr. Anne St. Amand and Dr. Ken Wagner. I'm sure you are very familiar with one of those two. First of all, for agreeing to donate the sponsored money for the workshop entirely 100% to the scholarship fund. So we are very thankful for that. And they wouldn't even let us purchase their attendance fee for this conference. They wanted that money to go to us as well. So another big thank you for both of them. And they are going to present the workshop this afternoon. They have a pretty aggressive schedule, so I'm going to let them get to it. You guys know who Ken is, but let me give you a little bit of background on who Dr. St. Amand is. She holds a BS in ecology, evolutionary and population biology from Purdue University, and a PhD in aquatic ecology from Notre Dame. In addition to lake assessments and algal work conducted on Florida lakes, she has over 35 years experience identifying and enumerating over 42,000 algal samples from all over North America using a unique proprietary mounting method. She specializes in rapid water quality assessments and harmful algal bloom assessments and mitigation. She's been the president and owner of Phycotech since 1990, located in Michigan. And her company has completed programming on an extensive data management system collecting information on nearly 34,000 different aquatic organisms. And you guys know who Dr. Ken Wagner is. He needs no introduction to this group. But Dr. Wagner holds degrees from Dartmouth College and Cornell University. He earned his PhD in natural resource management in 1985. 40 years experience working on a variety of water resource assessment and management projects throughout the Northeast and really the country. In 2010, he started his own consulting firm, Water Resources Services, and he focuses on water supply protection and lake management consulting. He's former president of NEAPMS, editor-in-chief of Lake and Reservoir Management, and is a member of APMS and a former director of our own society as well. So thank you both Drs. St. Amand and Dr. Wagner for joining us today, and I will turn it over to you guys.
All right, so can you see my directory here? You got my screen?
Yes, I can.
Awesome, okay. So everybody should have gotten an email that had two directories, linked to two directories up on WeTransfer. One was the workbook and one is additional materials. And so the workbook has all of the lectures as well as some additional materials that we would have if we were in person. And the additional materials ones has a whole bunch of different PDFs and different types of references, both articles and there's Prescott's How to Know the Algae and Algae of the Western Great Lakes. So those are just kind of some things that we have accumulated over the years. So, all right. I have a little bit less time here, so I want to make sure I'm staying at least kind of quasi on schedule, which is not one of my strong points. So, freshwater algae. Welcome, everyone. Hopefully, this will be a first. I will not get kicked out of my own webinar. That would be awesome. This is Ken's and my email contacts. If you have questions after the workshop or you have images that you would like some help on, both of us are willing to help with the time that we have. It costs nothing, so you kind of get what you pay for, but 9 times out of 10, we're able to help. Warning you, we're going to be repeating each other a lot, both ourselves and each other, and that's because this material is complicated, and if you've attended one of our workshops before, it goes pretty fast. The material is constantly changing, and so repetition helps a little bit. Just to give you an idea, my daughter is helping me update some of our diatom IDs, and she's a college student, so she burst works on it. And the last time she worked on it was a couple months ago, and already the first 15 names that she went in and looked at had already changed. This is kind of the quasi schedule. We'll run a little short, a little long, depending. If you have questions, put them in the questions, and in between lectures and over the break and at the end, Chris will read those out for us, and Ken and I can help out with anything that we can. And at the end, I have a bunch of not only prepared slides, which are wet mounts of preserved things like Aphanosomnon, Dolichospermum, Microcyra, a whole bunch of different divisions, but also the live sample that was sent to us from New Jersey, and I have not yet looked at it. It just got here, so they're concentrating some downstairs for me. So I am actually first up, and so this is the methods part. Again, put questions in. This is actually questions in the question. Webinar is a little bit different. GoToWebinar is a little different, and generally I would say unmute during the lectures, but we're only going to have you unmute during the actual break, and except for a quick bathroom break and a warm break because I have no heat in my upstairs this morning. Yay me. We'll be open to taking questions, and then we'll have everybody off mic as long as there's under 20 or so of us when we're doing the live microscope. And I don't know if you can upload images into the chat or not. That was not something I had a chance to look at. So when we talk about phylogeny, we think about traditional kingdoms. You're a plant, you're an animal, you're a fungi, bacteria, archaea, something along those lines. But when we talk about algae go across multiple kingdoms. So cyanobacteria, cyanophyto, blue-green algae, those are all the same thing that we refer to, but the proper term is cyanobacteria. And we've got algae over in the plants and algae in the flagellates, and it's complicated, right? This is where we are right here. So they span this area, and so the only thing they have in common is that they all have chlorophyll A. So they all can photosynthesize, they can all do chlorophyll A in the presence of oxygen. Everything else is subject to change. So when we talk about taxonomic schemes, Ken and I use the US construct, which is pretty much this Weir et al. 2015, it's in its 2nd edition. Depending on where you go, you could get 13 divisions with the diatoms separated out. Witten et al. Uses the phylum, uses the European construct, which is really the way you should do it. But when you're talking functionality, it's easier functional wise and functional group wise to use Weir et al. And to discuss them as groups or divisions. And diatoms are called out as a second group, even though they are a class within the Chrysophyta, because functionally we treat them differently. So that's always something to keep in mind. So given that, and this is a second edition book, it's a great book, 2015, this is what we're dealing with. So we have greens. They pulled something really difficult. They moved all the, originally the Charophyta were in the greens. Now they're their own division. We handle them in the greens because it's just easier. We have the cyanobacteria, cryptophytes, chrysophytes, haptophytes, which turns out those can be pretty important even though they're tiny. The diatoms, euglenoids, dinoflagellates, and then the ones we don't see very often, browns, reds. Goniosminum, which I might actually have some here, is generally something you see in acidic environments, and it has actually been in almost every division but the Cyanophyta. And this is kind of, you know, the morphological differences that we're dealing with. And if everything were this different and this pretty, life would be awesome. However, it's almost never this pretty, and often mashed and not nice colors. And so as a taxonomist, we're always struggling to try to figure out, okay, if something has been preserved poorly or is not in the growth phase we need, what could it be? And, you know, how do we interpret that data? If you have questions about where something should be, AlgaeBase is generally the go-to place. It's an online database. It is hosted out of Ireland, and so can be really fast or really slow. But it is probably the most up-to-date taxonomic treatise right now. There's also ITIS, which is maintained by USGS. It can be kind of behind, not all the names are updated. The same with Taxonomicon, which is more the European. This is US driven and this is European driven. And Diatoms of North America, which is a great resource as well. It's diatoms.org. But most of the images are SEM, and it's a work in progress. It's not yet complete for diatoms. So if you have a question, start with AlgaeBase. If it's a diatom, then you can move to Diatoms of North America. So all that to say, we have to figure out often how to get our samples. And the reason that we handle methodology in this workshop is because what you see is often a result of how you sampled. And so depending on how you sample, that's going to affect not only the concentration, but the composition of the sample that you get. And so you really need to have a good idea of what's out there and how you chose to sample. As ecologists, as limnologists, we often tend to sample or think of things as simple basins, right? So we think of single basins, go to a central point, but there's a lot of variability out there, both vertically and horizontally, and things are stratified, things are moving around, there's microstratification, there's windrows. And so if you sample up here, here versus here versus here versus here, you're going to get very different assemblages. And you need to have a good understanding of what's going on in your system to understand, okay, well, I collected Cyanura, but my water looks pretty clear and doesn't smell, but yet the water treatment plant's having taste and odor issues. Well, that's because Cyanura likes to be right down here at the metalimnion, which is often where water intakes are for water treatment plants. So it really is helpful to know where your sample was taken. Lakes aren't homogeneous, we all know that. And not only is the wind moving things around, but these guys move around on their own, either passively by having things like gas vacuoles, or actively by having flagella. And the zooplankton and the algae are constantly doing this and doing this. And, you know, it is actually pretty difficult to do a complete balance of zooplankton in a lake because they often go into the littoral zone and down to close to the sediments. And it's hard to figure out where they're actually going throughout the day. We don't do a very good job of sampling the littoral zone in lakes, which can be very important. And so just understand that if you're sampling mostly a pelagic sampling station, you're going to be missing a lot of biology that happens in that littoral zone. And you want to have a good idea of what your sample goals are. So if you are sampling at a deep station, open water station, if you're interested in assessing overall water quality, if you're interested in assessing human risk, then you're going to sample close to a place of human contact, like a dock, like a swimming beach, a bayment where people have a lot of homes or do a lot of fishing. So it's important because when you look at the distribution of cyanobacteria especially, they may be up here, but because they're buoyant, they're going to go up during the day. And if there's wind, they're going to get pushed over to the shore. So this is something very much to be concerned about. You need to be concerned about something like this, but this is a much bigger issue, right? So when we start talking a little bit later in the, when I do the ecology cyanotoxin lecture, I'll talk about what the different levels are for warnings as far as EPA is concerned. But it really affects how much toxin you encounter and what the implications and impact is of those different algae concentrating in different sections of the lake. So when we start talking about sampling equipment, most of us are going to be using something like a Van Dorn. If you're not just sticking your arm into the water, 1/2 meter, turns out conveniently that's at your elbow. Van Dorn is essentially a PVC tube with plungers. They have both vertical and horizontal ones. In lakes, that's mostly what we're doing. There's also Nansen bottles and integrated samplers, which can be as simple as a Tygon tubing in a hose, or as complicated as a pump. Net sampling. So we use net samples. I do nets all the time. I actually have a Wisconsin net I take out. It's qualitative only. It's quick and dirty. It's pretty cheap. But it's also inefficient, and they concentrate larger algal forms. So if you want to quantitatively sample, then you're going to want to be using one of these tools. And this is, if you want to go out and get a quick and dirty, what's there? I'm sampling for fun. I'm sampling for a workshop. I'm sampling to determine I either need to be concerned or don't be concerned, and I need to do some more quantitative sampling, but never use a net sampler quantitatively. This is actually Ken's boot. This is Lake Mendota, and you can see that there's a pretty massive bloom going on here. And I believe this was at the last NEAPMS in Wisconsin. I think it was Dolichospermum lemmermanii. In Las Vegas Bay from Lake Mead, this is the impact. This is what a grab sample looked like. This is what a tow sample looked like. The tow sample was dominated by Phanerozomenon, and the grab sample was dominated by Tetraselmis and small diatoms. Much higher concentrations, much lower concentrations, blue-green cyanobacteria dominated and green dominated. Very different data, very different pictures of what the lake looks like. This tells you, yeah, there's stuff out there. This tells you, no, it's not dominating. So this is a quick and dirty tool, and this is the quantitative tool you want to be using. Once you've got your material, you want to keep it cool in the field. You want to keep it dark. You want to allow for some aeration if you're going to be out too off along. Eventually, once you bring it inside, you want to give it a little bit of light, but not too much. Remember, these guys have been down deep where the white light has been attenuated. So give them a little bit of light when you bring them back into the lab, but not too much. There are some vital stains like India ink that you can use. India ink, you have to actually buy as a concentrate and reconstitute it. So it can be a little tricky. Never do this. When you're shipping samples, never ever do this. These are actually Rubbermaid containers that are meant for a fridge. They are not watertight. This was shipped. Turns out this is microcystis, and the microcystin was 30,000 micrograms per liter. So that is bad in anybody's book. And of course, we had to clean all that up to even look at what it was. And so you always want to use proper containers when you're shipping. Make sure that you have containers not only watertight, but taped, and then have those bagged when you're going to ship them to somebody so that if there's a leak, it does not end up all over the inside of the cooler and expose not only the people who have to open the cooler, but the people who are handling it while it's shipping. So you have these awesome samples. Now, what are you going to do with them? Well, if you need to count, you're going to have to look at them. You have a couple choices. You can use a temporary mounting method. That's going to be something like an Utermole chamber. Utermole chambers are made for use on inverted microscopes, which have a much higher working distance. Or you're going to use something like a Cedric rafter cell, either a gridded or a non-gridded. That's totally personal preference. Grids drive me crazy, and it's a short trip for me. So, I prefer to use non-gridded Cedric rafter when I use one, and I don't use one very often. Or something like a Palmer-Maloney cell or a nanoplankton counting chamber. And, you're going to have a concentrated sample for this. So if your sample is not already at a counting density, which is around 20 to 30 natural units per field at your counting magnification, then you're going to have to pre-concentrate it either by an Utermole chamber or by some other settling chamber. So If you're not going to use a temporary mounting method, and I don't, I use a permanent mounting method. A lot of you probably aren't going to ultimately be interested in this, but it just sort of gives you an idea of what you can do. It's faster. I have a countable sample in 2 to 4 hours. It's easier to make multiple mounts. You should make 3 mounts per count. Most people don't because the cells are expensive and it takes a lot more time and a lot more material. When I use a permanent mounting method, that's easier. I have a permanent archive, which is important for me because I have a lot of long-term data projects that I'm working on, sampling projects, and so I can go back 30, maybe 35 years and look at these things. So it's very, very helpful for me to be consistent. There's a little bit of a distortion, and it takes some skills to make the resin and to do the slides. So right up front, there is no perfect way. There is going to be a problem with whatever mounting method you decide to use, whether it's wet, whether it's Udemol, whether it's a cedric rafter, whether it's a permanent mounting method with HPMA. So what you need to do is make a decision about what errors you can live with and what your limitations are on what you're doing, and pick a method that's appropriate to your sample goals. This is the kind of difference of what these look like. This is the same sample. This is Pseudanabana limnetica in a wet, just on a cedric rafter and a wet mount, and this is on one of the sides. You can tell it compresses all the particulates into one plane. Makes some things easier, makes counting cells a little bit easier, but I also use fluorescence, and if you're going to do HPMA mounts, fluorescence makes your life a lot better and a lot easier. You can visualize cells a lot better. So your choices, there's multiple choices, but the 2 main ones are compound microscopes or inverted microscopes. This would be a compound microscope. And objectives, when you do a compound microscope, objectives come from below. The light objectives come from above, the light comes from below. It's more of a straight path to your eye from the specimen to your eyepiece. In an inverted microscope, it's a little bit more tortuous in terms of the path, where the objectives come from below and the light comes from above. You have more working distance. That's awesome. However, the resolution in my mind isn't quite as good, but it's all dependent on what you start with. So if you started with an inverted microscope, and I know that Andy Chapman at Greenwater has always used an inverted microscope. He loves it. He gets great images. He's used to it. I like the higher resolution of a compound microscope. It's easier to use when you use a lot of oil because you can imagine when you use oil, you're putting it on the cover slip. So it's much easier if your objective is coming from above and a lot less of a mess. If your objective is coming from below, you have that oil that kind of cascades down your objective. But you can get really good resolution either way. The main manufacturers are Olympus, Nikon, and Zeiss for the most part. If you're going to change into another manufacturer, make sure you have a chance to demo the microscope. There's a whole lot of, Russian, Japanese, Korean microscope producers where they don't last very well. And so a lot depends on whether you're going to use it for the short term or you're going to use it for the long term. Trust me, ergonomically, you're going to want a scope that fits you really well. And that's totally personal preference among those main manufacturers. So we have some options for enhancement. We have bright field, which is essentially light straight down through. We have phase, where you use an annulus that blocks out most of the light. DIC or Nemarsky, where you use crystal prisms as beam splitters, and that is the extent of my physics background. But you get really good resolution. This gives you the best resolution, as a matter of fact, on a light microscope, and it looks almost like a 3D image on an SEM. But if you need to highlight something, and I'll show you in just a minute, it's not quite as good if you need to highlight really fine structures. And then epifluorescence. If you preserve your sample in glitteraldehyde or you do live, you can use epifluorescence. If you use Lugol's, it doesn't work very well, and formalin also does not work very well. Make sure that you can see well enough to identify your subject. Probably the biggest complaint I get from people who come and use the scopes during our workshop at our lab, or the ones that I bring around to NEAPMS, is that wow, this is an awesome optic. And I'm like, well, it's clean. So if it's clean, you're going to have a much better time of it. And that means making sure that all of your optics from your eyepiece all the way down to your condenser, all those optics are clean. You never use anything like a chem wipe on them. You're always going to use lens paper and an approved cleaner. And having somebody come in once a year to clean your scopes. My scopes are filthy because it's now been almost 2 years since I was able to have somebody come into the lab to clean them. Even though I'm doing a lot of cleaning every day, but I use mine 8 to 12 hours a day. And you also want to make sure that everything's in alignment. So if you have stuff out of alignment, that's even just minor changes, that's really going to affect the quality of your image. So just give you an idea, this is what bright field images look like. You don't get a lot of enhancement, but if it's preserved or live, you can see true color here. You can use vital stains or non-vital stains depending on whether something was preserved or not. This is in Algal. This is, I think, rhodophyte. I can't remember what the name of the red one is, but these are Uothrix and Crucigenia preserved with iodine. When you go to phase, this is that same Crucigenia picture. Now you can see there's actually a sheath around this whole thing. You can see cell walls, stereonella. Everything in phase gets a bit of a halo. That is a visual artifact that is specific to phase. Sometimes it's important, sometimes it's not, but it definitely highlights really fine structures. If I were to go to 1000x, this is Ankyra, I'd be able to see that little flange at the end of Ankyra very easily. I'd be able to see the flagella on my Denobrian. This is Urtarina, and you can see where the flagella come through the colonial sheath on phase. If you go to Nemarsky, this is that same Urtarina picture. It's beautiful, but now you lose most of that detail because it's pretty fine. So you can kind of tell where that those flagella are coming through the colonial sheath, but not very well. You do lose that sheath or that halo that you see when you're in phase. So it's just kind of depending on what kind of algae you're looking at and what you think you need to see. Nemarsky costs an extra $5,000 to $8,000 to add to your microscope. So if you have to choose, you're going to choose phase first. And I almost would choose epifluorescence, which is another $5,000 to $8,000 over Nemarsky. I have all three on my scope. Epifluorescence is going to be two different basic color groups. So you're going to shine blue light at it or blue excitation, and that makes the chlorophyll A come back a beautiful, nice red, deep dark red if it's healthy. Green excitation, so it is a little bit backwards. Green excitation is going to make the cyanophyte pigments, phycoerythrin and phycocyanin, fluoresce back either a brilliant orange red or an orange yellow. So this is what blue light excitation gets you. These are both chlorophytes, Microsterias, which is a desmid, and Mucidospherium, which is a green algae. And you can see that in the Nemarsky image. You can see the edge of the cell walls. You can see the old mother cell wall connections between these cells. You can see the cup-shaped chloroplast, and you can see the pyrenoid. But when you go to fluorescence, what you find is that these become a beautiful dark red tenor, and you can kind of tell where that is shaped, but not as quite as well. And all of this detail, of course, is going to get lost in that black background. On our Microsterias, this is just a flip-flop of this same cell. You can see part of the cell is actually not doing very well, as is part of this cell. So this half of the cell, and actually that's split here, but this part of the cell, the chloroplast is quite healthy, and over here, it's starting to have some issues. So fluorescence can be quite helpful. This is Dolichospermum lemmermanii, so it's a cyanophyte, cyanobacteria, one of the ones that pretty predictable microsystem producer. And here is bright field. This looks brown because of the aerotopes that are in the cells. This is blue light excitation. So see how this is grainy and kind of washed out? That's because this is a minute exposure. So this is almost instantaneous. This is a minute exposure, and this is about the same exposure actually as the bright field. This is green light excitation. Its primary pigment is phycoerythrin. It has almost no chlorophyll A. And notice it's got a beautiful black, clear background here, and that's because the exposure time was a lot lower. If we look at microcystis, same thing. So phycoerythrin is its primary pigment. This is bright field. That looks brown because of all those aerotopes. This is Nemarsky, kind of gives you that shiny look. And this is using green light excitation. So phycoerythrin is its primary pigment. Chara, which is a charophyte, but remember we're going to cheat and cover it in the greens when Ken talks about it. So it is a macroalgae stonewort. It has kind of a garlicky smell. People tend not to like, but it's often associated with cleaner water. And it looks pretty green. The hard thing is that it's very dense, so you have to overexpose to get any detail at all. When you go to blue light excitation, this green is all the, it's very brittle. That's why it's common name is stonewort. That's calcium carbonate. And then you can see the red chloroplasts underneath. And then this stuff moving through here is rhizoclonium, which is actually a pretty large filamentous green algae as well. And then there's some epiphytes, Edagonium epiphytes on here as well. So we get a nice red, deep red color when you expose it to blue light excitation. So let's talk a few minutes about how you're going to quantify these things and what units you're going to do. You're going to use, you always want to do at least 2 of these, and that is total cells and standard units. Standard aerial units is mostly used, I believe, now by New York. These 2, you don't want to mix and match. Remember, there's a difference between precision and accuracy. So precision is how close you are, and accuracy is how right you are. All data has both those components, and you would like to hit both of those on the dot if you can. And you're going to have to decide what counting method you're going to use: fields, passes, strips, or the whole sample. Whole sample either has to be incredibly in a small sample, or you have to be insane. I never count the whole sample. And people tend to have a preference for either fields or strips. I tend to use fields. So natural unit is the whole thing. So however this thing appears in nature, that's its natural unit. So if you're a Chlamydomonas, you're a single cell. If you're a Serratium, you're a single cell. If you're something like a Desmodesmus, this is 1 natural unit and 4 individual cells. Do not mix natural units. Don't mix and match units. So don't say, okay, well, I'm gonna do Merismapedia, which tends to be in quadrats. Each quadrat's going to be 1 cell or 1 natural unit. You can't do that. The natural unit, think Daphnia. Your Daphnia, whatever you encounter in the water column, that's a natural unit. So this Desmodesmus colony, this is the natural unit, and it has 1, 2, 3, 4 cells. So you can imagine people, when you're trying to count something like microcystis, it makes them want to use cells, but you know, these colonies can be huge with 15, 2, 3, 4,000 cells per colony. You don't want to do that. You want to measure natural units and then do an estimate on the cells per natural unit on something that large. Algae bloom is loosely defined as visible color to the water. It is a bloom is different in everybody's mind, and so it has no quantitative definition. And a harmful algal bloom or a HAB just adds toxins or fish kills to the definition. If it's dominated by cyanophytes, we were saying CHAB, but now it seems as if people are wanting to use HCB for harmful cyanophyte bloom and leave HAB for the marine world, which is more dinoflagellate and diatom dominated. If you're going to express your data, bar graphs are awesome. And each date is a separate graph. So this is a proper way to express your data. If you're going to do algal abundance versus biovolume, they tell you different things. So this is individual cells. I think he's delivering my stuff from New Jersey. So the thing is that this sample was dominated by picoplankton, a lot of teeny tiny blue-greens. They were having issues with taste and odor. Their taste and odor, this brown category, is diatoms. Their taste and odor was actually coming from Staphanodiscus niagarae and Alocosira, not from the blue-green algae. There's lots of things you can look at: dominance, richness, diversity. There's all sorts of quality indices, indexes of biotic integrity as well. Some states have their own. Not near as much for the phytoplankton as for the for periphyton, but there's a lot of different ways that you can look at your data. It's hard to distill all this stuff down to all the complexity of algal data down to 1 number. It's really not possible. So I'm always looking at all of my analyses by biovolume if I have it, cells, and natural unit concentration, as well as maybe ratios of different things, percent cyanophyta, that kind of thing. So I tried to speed it up a little bit, so I'm done on time, and I believe I'm done right on time. So in the couple minutes, Chris, that it takes Ken to come back on, are there any questions?
So we just had a couple of questions early on about, is this the algae workshop? we've gotten those squared away. We're good. The only other question we have is, where are the supplementary materials and the workbook? I'm fairly certain that we sent all of the attendees a link to like a Dropbox type of file share that has all those things in there.
We did.
In addition, I'm assuming that those will be posted on our website as well. So all that supplementary material will be coming to the attendees. I just want to remind folks that if you put your question in the chat, we can't see it. So all questions need to go in the question box. Anne mentioned that earlier. I forgot to, so she stepped in and did my duties for me. And we're switching over Ken. So Ken is going to be doing the next presentation, which is going to be on taxonomy. There is a break at 2:15, just to let everybody know. In addition, you can put those questions in the question box at any time. And then we will get to them once we kind of reach natural breaks between the speakers and whatnot. So feel free to question away, and then we've got them all lined up in a queue, and then we'll get those to the presenters. So without further ado, Ken is going to talk about algal classification. Ken, I think you might be muted.
See the slide okay?
Okay, we can now see the slide and we can now hear you.
Very good. A couple quick notes on the methods that Ann brought up. She mentioned about proper packaging of samples. Generally speaking, even though plastic Tupperware is bad, glass is worse. I had somebody send me some this year, and they did a very nice job packaging, each bottle in separate bags and everything else. Well, guess what? Glass will still puncture the bag, and it was a mess. The other thing is that Lugol's, while it is not as good if you want to use epifluorescence at all, and there are some things that fall apart in it, other things preserve well, and it stains things certain colors in certain ways that can help you with identification. So it's not bad to use it. We do tend to prefer glutaraldehyde though. Oh, and then using temporary mounts is fine. Don't feel like you have to make the permanent mounts. What Ann does is absolutely top of the line, and you can go back to it year after year and make comparisons, which is great when the names are changing and you've lost track. But temporary mounts are quick, and they work fairly well for what a lot of us do. Okay, so algal classification. The bad news, there's no possible way in 45 minutes I can explain all the taxonomy to you. The good news is you don't really need to know all that to do your job. All right, I'm not getting an advance on this. All right, there we go. So the question is phycology an art or a science, and the answer is yes. There's a lot of different systems. Anne explained some of them to you. I won't belabor the point. The algae may not conform to it. She mentioned Goniostemum, which is in the rhapidophytes now. That's been all over the place because it has characteristics that could lump it in there. A lot of the changes in recent years have to do with genetics. You can't see the genetics. You can only see the physical result. So you can't tell often if you're looking at something that's just variation in the same organism, or are they really separate organisms. It takes a lot of practice, and it is constantly changing. The taxonomy keeps getting reorganized and spoke to that. It can be frustrating. Yeah, it may seem like a big yawn, but you really do need at least a rudimentary understanding. For those of you that deal with rooted plants all the time, if somebody calls you up and says, I have a rooted plant problem, isn't your first question, what kind of plant is it? Maybe how bad is it? But you want to know what it is. It's the same thing for the algae. Okay, so the key classification features, you get the pigments, the cell wall, whatever form of food storage it has, which will usually take a physical or stainable visual form that you can cue off of, whether it has flagella at any point in its life cycle, how it reproduces, other cellular structures like spines, and cell associations. Is it unicellular? Is it colonial? Is it filamentous? Is it globular? How does it shake out? Okay, herein lies the real problem. If I showed you this, I think most of us would willingly assume that the top row was one type of diatom, and the next row was another, and the third row was another. Well, it turns out it goes the other way. These are all one species, and these are one species. Each vertical column is a species, and it has to do with the nature of the punctae, the holes through the wall, and again, determined by the genetics. But physically, that's not what it looks like. And these are scanning electron micrographs. You're not going to see this most of the time. So it takes a lot of activity, a lot of practice and knowledge to get to a species level, and sometimes it's still impossible. But you don't usually need that. If you can get to the genus, or at least get it in the right division and maybe the right family, you're doing well. And for the most part, the ecology will not vary greatly amongst genera within a group. Okay, so the modern systems, they depend on ultrastructure, chemistry, genetics, a host of other factors, not all of which we can see. We have to recognize there's a gradient of features and related uncertainty. That weir sheath cociolate volume that Ann mentioned early on is really good about laying out, look, we think this, but we don't really know, or this is probably a species complex, we're not clear on that, and tells you where the work is still needed. We have mostly the same general groupings as we did 50 years ago, but there's more subdivision, more splitting, some reorganization, and still a lot of disputes about what hierarchical level we should apply. Yeah, and again, it helps a lot if you're drinking. Okay, so basic tools, the genetics, chemistry, culture response, and visual inspection. We only have visual inspection to work with most of the time. Okay, so let's go into the blue-greens. Again, cyanobacteria, I've heard people say these aren't algae, and explained it very nicely. Algae is a general term. It's not super scientific. It covers a whole lot of organisms that get thrown together because they can photosynthesize. Some of them don't even photosynthesize, but they're in the group. But they do break out by taxonomic systems into very different areas when you get into the genetics and the details of their metabolism and such. So, okay, blue-greens have chlorophyll A. It's the only chlorophyll they have, and they often don't have much of it, but they have it. They've got phycoerythrins, phycocyanins, some other xanthophylls, but none of these pigments are bound in distinct organelles. It's not in a chromatophore, or for a green alga, a chloroplast. They're in what they call thylakoids, which is just a loose association in the cell. It's not something you can usually see very well unless you're using some serious microscopy. Their cell wall is just sort of this matrix of, it's almost like a water balloon where, you can lice the outside of it and have it fall apart. They often have some sort of mucilaginous outer covering. The cells are usually pretty small, but the conglomerations can be pretty big. They store starch-like compounds, they call them cyanophycean granules. We used to call them sugars. The point is there are little dots in their accumulations within the cells, but it's a very different compound than a lot of the other algae have, and that's important because it's best metabolized at high temperatures. This is one of the reasons why blue-greens do so well when the water's warmer. Other things like diatoms that use oils prefer colder temperatures. They do not have flagella at any stage. That doesn't mean they're not mobile. Some of them have microtubules in the cell walls that allow them to wiggle and move, but you're not going to see flagella. They're not going to swim like that. Okay, there's no true membrane bound organelles, like I said, usually a fairly even coloration, except that gas vesicles, those aerotopes as they call them, and various granules can refract light and make it look like there's more structure than is really there. Again, the cells are usually pretty tiny, but the globular aggregations and the filaments can be pretty big. There are unicellular forms for sure, but usually these are pretty big. You're going to collect them in nets. All right, the taxonomy's been revised over the last, I say few years, really a solid decade, and even goes back further than that, but a lot of changing. And they call it polyphasic analysis, which is just multiple lines of evidence to get us there. And the distinction between the coccoid and the filamentous forms is not really that clear. It appears that some filamentous forms were derived from other coccoid forms and are in totally different groupings than other pairs of coccoid and filamentous forms. So just saying, oh yeah, with the little round ones, and we have the filamentous ones, and we have these other ones, yeah, it doesn't work anymore for good taxonomy. The thylakoid arrangement makes a great deal of difference to the divisional or subclass taxonomy. We don't even see that for the most part. the ones that have heterocytes, we used to call them heterocysts, but cysts has different meanings. They got changed to heterocytes. Those are specialized cells that allow them to fix gaseous nitrogen. Really important. And a lot of genera have been shown not to be one genus. In fact, there's a continuum there of different groups. Dolichospermum has been broken up quite a bit. So is Aphanizomenon, Senecocystis, Formidium, Lyngbia. They've been turned into multiple genera for genetic reasons. And usually there's a physical means that lets us figure that out. The Dolichospermums, which is the old Anabaena as we know them, and Aphanizomenon are pretty closely related. There's a continuum between them. And then the morphology-based species approach to Microcystis hasn't been, so just because it's globular, it has a lot of mucilage versus one that's more spread out and less mucilage doesn't mean they're separate species. That could be what they call a phenotypic adaptation to the environmental conditions. Okay, so the differentiators, there's a list there. I won't just read it off to you. Again, you can go back to this at any point, but there's all these different features that you can look at that will tell you what's going on. You need a pretty good microscope to see a lot of this stuff. All right, so the specialized structures that are really important to understand, both in terms of taxonomy, ecology, and even control, the aerotopes or gas vacuoles. It's an assemblage of a bunch of little gas pockets in the cell. And here we're looking at it on Dolichospermum, and those are just gas pockets that refract the light and give it color and give it structural appearance that isn't really there. Heterocytes, again, that's what we used to call heterocysts. These are specialized cells where they can actually fix nitrogen. And then aconites, which are resting cells that they create that drop to the bottom and let the thing come back the next year. Okay, the heterocytes are morphologically and physiologically different than the other cells. That's where atmospheric nitrogen is fixed. It's an anaerobic environment. They tend to be sort of yellowish to clearish. There's not hardly any structure in them. Sometimes they're in the middle, sometimes they're at the end. All that matters to taxonomy. The aconites are also specialized cells. Their resting stages, their exact shape, and their position on the filament make a lot of difference. They germinate when they're exposed to favorable conditions. Usually that means the following year, but they could sit around on a herbarium sheet for 100 years, then be put in water and come back. They're pretty tenacious. And the aerotopes, again, they're groups of gas vesicles. They can allow the cells, colonies, or filaments to change their position in the water column. They can become buoyant, more buoyant, less buoyant. They can go up and down. Sometimes they blow it, they go all the way to the surface. I don't think they really want to form those surface scums because they usually fry in the sunlight, but that's how we wind up with the surface blooms. Okay, the modern system, there's all these, one phylum, one class. There's actually a bunch of subclasses. Then you have all these orders. We're only going to show you pictures of the first 5, and I'm going to breeze through those just to show you how, I'll say, messed up the taxonomy has become by the genetic work. So here's the Synechococcales. Well, it's not a bunch of little round things or even an oblong like Synechococcus. It includes long, thin filaments. These two are both fairly common ones, Planctolyngbia and Pseudanabaena. Schizothrix, which has a lot of mucilage and often more filaments. Merismopadia, which Ian mentioned, which comes in little quadrats. And Aphanocapsa, which looks a lot like Microcystis if you're not used to separating them out. That's grouping together genetically. We would have never put these together from a physical or visual analysis. And then the spirulinalis, well, they're all curling forms, at least shown here. But interestingly enough, Arthrospira, which is physically similar to Spirulina, is actually in a different order. And a lot of what people buy is those super blue-green, vitamin capsules or whatever. It's called Spirulina, but it's actually Arthrospira. They've been split up. And of course, for marketing purposes, they haven't changed the names to stick with the taxonomy. Crococcales, again, mostly globular forms, and there's the classic Microcystis. We also have Crococcus, Gomphospharia, which has been subdivided into other things as well. And here's a filamentous form, though. Well, epiphytic filamentous form. So again, it's not just coccoid versus filamentous anymore. And then oscillatoriales, again, mostly filamentous forms, but not all. And again, genetically, some appear to have arisen out of others evolutionarily. And then Nostoccales. Now, this group doesn't have any unicellular ones. These all have heterocytes. That's the primary thing they have in common. They may be unbranched, falsely branched, or completely branched. That went on its own. But they all have heterocytes, and all those have been grouped together. Okay, I'm going to backtrack here and give you a little of the names of stuff and pictures, and I'm going to do it the old-fashioned way, which is what most of us grasp easily. You know, unicellular to a few-celled, globular, coccoid, filamentous, unbranched, branched, branched. So here's some of the common ones you would see that are unicellular. Any of these could form blooms. Most of them don't tend to be super abundant, mostly because being small like this, they can be eaten. But we've heard of phytoplankton blooms, which are picoplankton, very, very tiny ones that get abundant enough to actually discolor the water. Pretty much you're not going to have a lot of zooplankton around then because they would eat them otherwise. Okay, then you have the coccoid colonial cyanobacteria where they're in big groupings. You know, the Merismopedia, those are pretty easy to pick out because there's always these flat plates of a whole bunch of cells. Aphanocapsa, Aphanotheca, which looks a lot like it. The cells are almost always paired, usually more elongate, but macroscopically, this you just see microscopically, the Aphanocapsa. You know, you wouldn't ever see a glob of it. These can actually, the Aphanotheca can actually create balls. And then Gomphospharia, which was split off from Warrenachinia and Snowella, and Radiocystis, they're all physically somewhat different, ecologically pretty similar. And Microcystis, which is the one we worry about the most because it's the most often toxic form of these. Okay, the Warrenachinia is one to be aware of. It's a bunch of little cells on the outside of the mucilaginous thing. There's the cells over here on the right, you know, on the outside, just a few left, and you just see the gelatinous stalks that are left. And oftentimes in a preserved sample or even a live sample that you've kept too long, they'll have exploded and it'll look like this, and you won't know what you're looking at because it's no longer in the colonial format. These are not Microcystis. They are different from Microcystis, although they could produce toxins. They most often do not. Less of a hazard, less of a problem, but they can form blooms. Microcystis, on the other hand, has a bunch of different forms that are potentially different species, but you can kind of recognize it. On the when you focus up and down through Warrenachinia, you see cells, then you don't, then you see cells again on the other side. Here the cells are throughout the mucilage, and this is one of the ones we worry about a lot. Okay, the simple filamentous forms, you know, we've got a straight filament with or without a sheath. It may have a sheath that extends past the end, which usually puts it in the lyngbia. That has no sheath at all. That's often formidium, although the shape of the cells matters. The typical stack of coins with varying shapes at the end is oscillatoria. Planktothrix, which will come up, has been split off of oscillatoria because it has erotopes and it floats. Oscillatoria does not. However, these will sometimes come up as chunks, mats that grew on the bottom that float to the top when they trap their own photosynthetic gases, but it's not the same process that the ones that make erotopes are using. Ones that often grow at the metalimnion, down we've got a stratified lake, the Pseudanabena, Planktothrix, Trichodesmium, which tends to be more brackish water, but these are ones that we see blooms of when stratification breaks down later in the year, or in the case of Planktothrix, if it just does a lousy job regulating its buoyancy and all pops up to the top. And one species of this tends to be pinkish to purple. It's very obvious. The Pseudanabena is very hard to pick out. It's just going to make the water greenish. There's also a few epiphytic forms down here that go in this group, but they're not likely to be something you ever get a phone call about. Other simple ones, lyngbia, which again tends not to be planktonic, but it could float up. Limnorhapis, which looks sort of like that, but it is planktonic. And then Planktolyngbia, and there's a couple of those, Planktolyngbia limnetica, Planktolyngbia contorta, which are real fine. And if you're not looking carefully, you can miss them. But they often form at the metalimnion and then can wind up in the upper waters. Microcyra, which we used to call Plectonema, which we used to call Lyngbia, again, they keep jumping around, is a big, heavy mat former that if you have a long enough season to get down to the bottom of the NEAPMS range in Pennsylvania, New Jersey, by late summer, this can pop up. Pretty rare to see this show up at the surface in northern waters. The growing season just isn't long enough for the mat to get thick enough and trap its own gases. Other ones that have spiral morphology, you know, shown here. And again, I've talked about Spirulina and Arthrospire. They're taxonomically separate, but boy, they look the same to me. And then the nostoc halides, which have heterocytes and usually aconites. They don't have to have them in any sample you see. If they're growing just great with plenty of nitrogen around and conditions are ideal, they may have neither, but ultimately they can form them. Dolichospermum is what got split off of Anabaena. The original Anabaena was a benthic form, so it got to keep the name. They had to come up with something new. Nostoc is a dolichospermum that lives in a gelatinous matrix. Aphanizomenon has longer barrel-shaped cells and usually, well, not always, but often can come as a flake, you know, a grouping of them together called a fascicle. Nodularia, which has usually a double situation here that makes it easy to see. And then Raphidiopsis, which is Raphidiopsis and Cylindrospermopsis were considered separate for many years. And I know Ann and I have had these discussions with other phycologists that, gosh, they really seem like they're the same. It's just that the Raphidiopsis doesn't have the heterocyte at the end. Well, eventually they figured out that, yeah, it's just they weren't producing heterocytes. They are the same. Raphidiopsis has taxonomic primacy for the older name. So what we called Cylindrospermopsis is now Raphidiopsis. More of same, you know, just the dolichospermums. There's a bunch of different forms. Ann showed you the Dolichospermum lemmermanii. That one will grow on the bottom and pop up. The coiled forms often start on the sediment, get big enough to be like an 8-foot plate of spaghetti to a zooplankton with no utensils. They can't eat them, and they pop up. This Dolichospermum lemmermanii will sometimes have vorticella or other stalk ciliates already growing on it from sitting on the bottom for literally a month or more. It could pop up as early as June, as late as October. Straight forms, some of those start, well, all of them start in the sediment because that's where the aconites are, but some of them grow down there, then pop up. Other ones grow up in the water column or even metalimnetically. Anabaena, which still exists, is a benthic form. Sometimes it's got, looks like some aerotopes to it, but it doesn't float like the Dolichospermum. Aphanizomenon's been split off or split up a bit. Again, long barrel cells, long cylindrical aconites. Very distinctive sort of ones, not too hard to identify and separate out. Cuspidothrix is basically a Aphanizomenon with a really long cell at the end in each case. Usually the heterocytes are much more elongate as well. Genetically, apparently they're separate. Ecologically, they mean sort of the same thing. And then there's a couple intermediate ones that have popped up. So now we've got Sphaerospermopsis, which you could confuse with a Dolichospermum very easily, and I don't think it would make a big deal if you did, but they are separate. And Chrysosporum, again, same idea. The shape and positioning of heterocytes and aconites are what separate them out. And of course, they've confirmed this genetically, but I'll bet you there's a whole continuum in between. I mentioned Raphidiopsis already, or what was Cylindrospermopsis. Again, you get this terminal heterocyte, but it doesn't have to be there. It can be missing, in which case it's hard, and they're small. And these aren't especially buoyant, so they often are suspended throughout the water column. They are uncommon in the NEAPMS region, but I have seen them. They make much worse blooms further south. And then again, we have clustered ones which have apical basal differentiation, most famous which is Gliotrichia. It grows at the bottom, becomes this entire fuzzball, then pops up to the top. Brings its nutrients with it. It's like a picnic lunch coming to the top of the hill for a nice view. And then some other ones that are out there that, again, aren't going to be something you hear about very often: Cylindrospermum, Anabaenopsis, which has these two terminal heterocytes, Macrospermum, Nodularia. Again, they're out there. You might hear about them, but not going to be very commonly something you get a call about. And then there's falsely branched forms. Again, they're mostly going to be paraphytic. They're going to grow on the bottom, but they could pop up. And there's truly branched forms. Some of the truly branched forms, like Stygonema, are actually in some of the cleanest water. So again, you can't make the claim that blue-greens are just a bad group and that they're in poor quality environments. The hot pools at Yellowstone are mostly blue-greens. Some very clean water in the Pine Barrens in New Jersey has lots of blue-greens. They're a valid functioning part of the ecosystem. Just some of them get out of hand. There's a few more, just Stygonema, Haplocyphon. This Atochthanes, I can never pronounce it. This is the one they call the eagle killer. It grows on hydrilla. And then if the eagles eat the coots that ate this, they can die from the toxin bioaccumulation. So there's definitely issues with these, but not all. Again, Stigonema, I've never heard a bad word about it. All right, ecology notes. Again, photosynthetic bacteria, lots of blooms associated with eutrophic waters, but they're tolerant of a wide range of conditions. There's many clean water forms. The buoyancy with the gas vesicles is a big deal. And most of them, maybe all, we don't know yet, can produce toxins, but that doesn't mean they do produce toxins. We'll come back to that in another lecture. All right, the greens, that's the other really big group that you get phone calls about all the time. That has chlorophyll A and B, and the B makes it much more grass-like. It's got carotenes and xanthophylls, copper ribbon-shaped chromatophores, which in green algae we call chloroplasts. They tend to be grass green in their natural color. They've got a cell wall that's a couple layers of cellulose and pectin, usually pretty rigid, pretty tough. It's why they're hard to kill sometimes with algicides. Rigid and distinct shapes in most cases. The food storage is starch, usually attached to a pyrenoid, which is a little proteinaceous core. They look like little spherical metal balls, but not all of them have them, just most. Flagella, they can have 2 up to 8. Sometimes it's only in the gametes, but at some stage in their life, they do tend to have them. And again, usually grass green in color. The pyrenoids are usually obvious as a little dot in the middle of the cell. If you put starch in them, which comes with Lugol's, they'll turn blue to brown because the starch reacts. Same reason if you spill it on a piece of paper, it turns blue to black. They have well-defined cell and chromatophore shapes, so you should not be confusing these with blue-greens. Taxonomy is also in flux, a lot of split orders and families. Again, it's all based on genetics. We've long known multiple cases where species or genera were nothing more than environmentally induced versions of the same taxon because it got grown in higher nitrogen or lower phosphorus or whatever. However, we found it goes the other way too. Things that we thought were the same thing are actually just very similar looking versions of 2 different things that have morphologically merged. The classic visual taxonomy gets applied here because that's all you can really use. The Chlamydomonadae are the flagellated forms. That's everything from single-celled Chlamydomonas with 2 flagella, or Carteria and a few other things that have 4, all the way up to these multi-celled forms to the classic of synchronized swimming, Volvox, which is a big colony, could be as big as the head of a pin, swimming around and rolling. Most of these like fairly organic water. They're not usually indicators of clean water. The chloralales, they've split up what I've always called the chlorococcales into chloralales and spheropleales, only because they're harder to pronounce. You've got these ball type ones, and they may have connections between them, like Dictyosferum. They may be encased in mucilage like Oocystis. They may be floating free like chlorella. If your microscopy isn't good enough, you're not going to be able to pick some of this up. The spheropleales are symmetrical cell groupings, but usually not round cells. And the big splits have come within Cenodesmus, which we now have Desmodesmus, which has the spines, true Cenodesmus without them, Tetradesmus, Acutodesmus, all sorts of other ones that I think Frank Trainer, who was a Cenodesmus guy, would be rolling in his grave because he thought he could create different ones of these just by growing them in different levels of nitrogen. I'm not sure I'm buying all this, but genetically they claim they're separate. I will tell you there are some ecological differences and particularly some control differences. If you use sonication, it nails these. It doesn't do very well against these. So there are some differences that make a difference to us. Pediastrum's another one. We always had a bunch of pediastrums. Now we have pediastrum, pseudopediastrum, monactinus. There's another one called Steridium. They've broken this up genetically, and they are visually very distinct. You can tell them apart, but it's what we always called pediastrum. The spheropleales also include Hydrodictyon. That's because it reproduces the same way as they do, but it's very different. It's water net. It forms very dense mats, usually in sewage impacted situations in the US. Although we seem to have transferred it to New Zealand where it took over some of their cleanest lakes. Go figure. Cladophorales are the large multinucleate filamentous forms. There's 3 main ones that we run across. These you get calls about all the time. They love high nitrogen. They need to have adequate phosphorus. Cladophora being one that's very common with branching. The other two being rhizoclonium, which doesn't branch much, although sometimes it has a few branches, and pithophyra, which tends to have alternating cells with less and more pigment in them in slightly different shapes. So these are all very difficult to kill. They form as mats, then float up, and they are miserable to deal with. Eulotrichales, which are unbranched filaments, they float around, but most of them are attached. They have these parietal chromatophores that wrap around the outside of the cell. Common in a lot of reservoirs or on the walls or on substrates. An interesting mix, Drapernaldia, which has the central filament with all these differentiated filaments going off of it, is in very clean water. Stygiclonium, which has a central filament with other ones coming off that don't look quite as different than the original central filament, it tends to be from some of those polluted water. So these are very closely related, but ecologically very different. That's an exception to the rule where I've said if you just get in the right family, you'll probably be okay ecologically. Doesn't always work. Edogoniales, another group of filamentous forms that tend to have a little bit of apical basal differentiation. The cells aren't perfectly, barrel shaped. They are mostly attached forms, but they can break free, and some of them do grow as floating masses. They like colder water, are common in the spring. They have interesting male and female parts to their reproduction, which is unusual in most algae. Bulbochaete has long spines and tends to be attached to reeds, that sort of greenish, slime that you see on different reeds and such in the water is often them. Edogonium tends to have low pigmentation and is often floating free. Okay, the Charaales, which we've lumped under Chlorophyta, but are really now considered separate. Charophyta, most of us throw in macrophytes. Nitella, Chara, Nitellopsis. This is a group that is familiar with these. You don't need to belabor it. These we think of as rooted plants, but they're not really. However, the weird part is that what got thrown in the Charophyta genetically were what we used to always call the desmids or zygnematales, which includes some filamentous forms and some unicellular forms. The primary filamentous forms are the Zygnemataceae, Spirogyra, Mujosia, Zygnema. There's a couple other ones in there, but these are the very slimy mats that form. They usually start at the bottom. They often grow in the water column, look like clouds of green cotton candy. They can trap their own gases and float to the top. They're very slimy. They smell god-awful when they decay. They are usually indicators of sort of intermediate trophic category, but they can be opportunistic and create real problems if you get an imbalance in the nutrients. They like more acidic water, but they'll grow just fine in basic water. The Desmidiales or Desmidiaceae got brought in as well. They're all related to the ones I just showed you. They're unicellular, but they're two cell halves that you could split them bilaterally and get two roughly equal parts. And these are really pretty as live algae. These are what my advisor, Hannah Crosdale, studied mostly. They can form blooms. They can be an issue. But again, they tend to be associated with more acidic water, often cleaner water. But if you get a pulse of nutrients into one of these lakes, these are the ones that are very opportunistic and will take off and grow like crazy. Okay, again, tremendous variability. The ecology varies by order or family. There are sewage forms, there are acidophilic forms, there's bloomers and mat formers. The blooms are usually indicative of high nitrogen, or at least a high nitrogen phosphorus ratio. The low nitrogen phosphorus ratio tends to favor the blue-greens. So there's a tip off there when you're getting blooms of one or the other. Okay, so blue-greens, greens, and now chrysophytes, which in reality is about 6 or 7 different groups that we throw together because they all have chlorophyll A and C. They've got carotenes, they've got xanthophylls, which are often dominant and cause them to have a brownish or olive green color. Their cell walls vary tremendously, which is one of the ways we split them up. Some of them are flagellated, another important split. They tend to be golden through olive green to brown. I've seen them look kind of spring greeny, almost like the background in this picture, but they're not usually grass green like the greens are. Cell wall can be silica, it can be calcium, it can be chitinous, it can be overlapping scales, it can be overlapping chunks like silica masses in diatoms. They may have a mucilaginous coating. They may have an outer casing called a lorica, which is denobrium. It's like the little wine glasses that the cells are inside. There's a lot of variability here, and that's caused them to get split up into multiple groups. Food storage is chrysolamin or leucosin, which are carbohydrates, but they metabolize like oils, and they do well in cold water. The flagella varies by the subgroup. They could be absent, or usually they'll have two of uneven length. They have certain characteristics that'll jump out at you. The diatoms are all very rigid and shapes. If you see them up close, they're very ornamented. Most of the flagellated forms have a big clear vacuole somewhere in the cell. There's a big open area in most cells. Some of them have spines like Chrysosphaerella, and some of them will have the haptonema, and that's the premnesiophytes or haptophytes that can be toxic. Generally speaking, this is a group that's mostly feared for taste and odor in drinking water supplies. Again, large group, a lot of taxonomic adjustments still going on. Ultrastructural examination has been really helpful to the people trying to do it at vacations, and a real mess for us that have to do it, looking at it with regular microscopy. There's a continuum between taxa that's present rather than clear breakpoints in many cases. The genetic and biochemical studies are really needed to sort it out. We're covering the key ones here that you might get a phone call about. Okay, again, diatoms, rigid shapes, very distinctive groupings. Usually these are pretty easily identified, at least to genus. They can be spring or summer, or spring or fall maximum abundance. Probably not going to get called about having to treat these, but sometimes they can be abundant enough that somebody's going, what is going on here? Certainly they could clog water filters, and there's a good example. These radially symmetrical forms, particularly the Cyclotella complex, which is actually a group of species, those can be as little as 3 or 4 microns. They can get deep into a filter bed and clog it up pretty badly. Some of them are filamentous, like Alococira, and an unornamented form is Melisira, which is what Alococira got split off from, or more chain of barrels put together. They can be held together by spines, by mucilage, by what look like little Velcro pads. They tend to clomb together because it maximizes their buoyancy. It's not so buoyancy, it keeps them from settling so quickly, because they are cold water forms and they are made of silica, which is basically sand. They're going to drop out when the water's warm unless there's a means to keep the water circulating. One of the tricks is to keep a circulation system going to prolong the diatoms into the summer instead of the greens and the blue greens coming in. We'll come back to that later in the day. Some of the forms are elongate, and even though they're centric diatoms, they fall on their sides, and you only see them on the side. Urocylene and Acanthoceras, very fine, very hard to see. If you're not using phase contrast optics, you're probably not even going to notice them. Pennate diatoms, some of them have no raphe. Raphe is a central slit that the cytoplasm can stream in. It may be a mechanism to get things out of the cell, things into the cell, or to allow them to move like a tank tread. We're not actually sure. We think we created the zipper. Think again. There's a lot of different forms. They are not hard to identify. Again, there's more forms, with pseudoraphes, tabularia being one that commonly shows up and can be a bit of a problem in water supplies and can even discolor the water in recreational lakes. Multiple orders have a raphe. Sometimes it's out on the edge, sometimes it's in the middle. The whole navicular group, which is a huge group, pretty straightforward. You got to see the ultrastructure to sort them out, but there's not a wide range of ecological conditions going on here as far as we know. They're ubiquitous as a group. They hit their maximum abundance in cold water, usually spring and fall around here. They love acidic water. They do very well in it, but you'll find them in the whole range. The planktonic forms, usually the ones without the raphe, although not always. And the slimes on rocks are usually the forms with the raphe, although again, not always. Okay, the other chrysophytes. There's a bunch of different features that split them up. Cyanura have scales. The Haptophyceae have this haptonema, short little flagella look-like thing sticking out. Eustigmatophyceae that doesn't have the chlorophyll C. They have special forms there. Rhaphidophysciae, which is the goniostum that we mentioned before, that seems to be the junk drawer algae, keeps getting passed around. And Tribophyceae, which are the filamentous forms for the most part. So just a few of these that you might run across. You know, they're tend to be fairly delicate forms. They often fall apart when you preserve them, which makes them harder to figure out. This is a Cyanura. At least they stayed near each other. They could wind up as a bunch of separate cells and you'd never know what you were looking at. This is what it would look like before, at the top there, before it dissociates. The Chrysophyceae chromulinales, single cells or colonies. Denobrians by far the most common one, and that could certainly get abundant in lakes. Looks like a bunch of clever wine glasses or fluted champagne glasses tucked into each other. Pretty darned easy to identify. Although I will say that those cells pop out when they're preserved. Now you got this little flagellate sitting there without its lorica, its shell, going, okay, what the heck is that? So you got to be a little cautious. Chrysosphaerella has spines on it and will often form blooms at the metalimnion. Most water supplies have an intake right around there, and this can give you some taste and odor. Ochromonus is just a unicellular version of the same thing. Again, more of same. Uroglena or Uroglenopsis, two fairly similar ones. Again, they fall apart really easily when you preserve them, but they make these big balls. They're sort of the equivalent of a green Volvox. This is the Chrysophycean form, and they can create taste and odor in water supplies. They like acidic water as well. Cyanuraphyceae, again, the Cyanuras, very common. Again, they seem to prefer the acidic water, but doesn't have to be. They produce a lot of taste and odor. They are a real threat to water supplies when they get abundant. They can be facultatively heterotrophic, as a lot of these chrysophytes are, which means they don't have to depend on photosynthesis. They can grow under the ice with very little light. They can suck up organic compounds. They can eat bacteria. They are able to function like an animal. Okay, and then the tribophytes, the filamentous forms. Tribonema itself sometimes does bloom in lakes. It's filamentous. It doesn't look grass green. It's usually this sort of a color, but it can bloom, and you might get a spring call about that in some places. Xanthophytes like Vaucheria, which are the equivalent of Cladophora in the greens. This is just the chrysophyte version of same. I've seen these bloom in cold spring water where they're near farm fields where they get a lot of extra nitrogen. The haptophytes, Primnesiaceae, again, they tend to be more in brackish water or southern reservoirs. I'm not aware of them occurring at any abundance in the Northeast, but these can be toxic to fish anyway. They cause death through the gills. And so they're very real. The euphemistic name for these in the South is golden blossom. Okay, again, often cold water forms, common in low nutrient waters, but there are bloomers, and you give them the opportunity, they'll take off. Many forms are facultative heterotrophs, which means they don't have to depend on photosynthesis to grow. Okay, and down to the very end here, the pyrophytes or dinoflagellates, much bigger threat in saltwater. Freshwater, they're around, but they don't produce toxins. Sometimes in brackish water they do. The naked dinoflagellates, Gymnodinium, Gyrodinium, not very big, not usually bloom formers, not a big deal most of the time. The armored ones, the Peridiniales, more commonly bloom formers. Sometimes they'll give you taste and odor. Again, not toxicity, but they're things you might get a call about. They can really take off. If you have a lake that you've treated and you've killed off a lot of algae and let a lot of organic matter into the water column, these will often take off. These or some of those flagellated chrysophytes, because they do very well functioning as facultative heterotrophs. Again, they're around, most abundant in organically enriched water, a lot of facultative heterotrophs, generally acidophilic, harmful algal blooms in marine waters, but toxins are uncommon in fresh water. They just make the water colored. And then the Euglenales, the main group is the Euglenales. The Coleoeciales only grow in the anal cavity of zooplankton. So if you think you have it bad, you could be a Coleoecium. But most of these are also high organic water growers. They're common. I find them in almost every sample if you look hard enough. But when they bloom, usually you've got an issue with high organic content. Could just be leaves falling into a pond, but oftentimes it's a manure situation or something like that. Again, they're associated with high organic content water. There are some facultative heterotrophs. They can form red blooms if the water turns to blood. This is one of the possibilities. Okay, there are other algal groups we haven't covered, and I realize I have blown through these really fast, but the odds are slim that you're going to get a call from those other ones. Most of the time, it's going to be blue-greens or green mats, and occasionally, if you're a water supplier, you're worried about the chrysophytes. Beyond that, it's not too common to be an issue that we usually get involved with from a management perspective. And that is the end of that slideshow.
All right, and we are right on schedule, a couple minutes ahead. So we do have a couple of questions that came in. So I'm going to actually take them in reverse order. So for Ken, this one's for you from Ed Molesky. Do cyanophytes prefer or require more nitrogen than other algal groups or taxa?
They require nitrogen just like anybody else, but they can get it from the gaseous, the dissolved nitrogen gas in the water column if they have those heterocytes. There's some suggestion that the fascicles that form by some types or even the microcystis globs might create anaerobic conditions that would also allow them to fix nitrogen. But the bottom line is they still require nitrogen. I wouldn't say they need more, but the way we measure it in the water, nitrates and ammonia, they don't much care about that because they can get their nitrogen from the gas, which in our atmosphere is 78% nitrogen. So we're not going to stop that. So a low nitrogen phosphorus ratio, nitrogen being either total nitrogen or the dissolved inorganic forms, ammonia, nitrite, nitrate, versus phosphorus, either total or dissolved. The low ratios, less than 10, favor the blue-greens, not because they don't need nitrogen, but because they can get it another way.
All right, thank you. This question is for Anne from your previous slides, and this one comes from Benjamin Burpee. I think that's his last name. Can you explain the older standard units for algae quantification? I've seen data reported this way, but I'm unsure how to interpret it.
So the aerial standard units, what they equate to, I believe, is 400
20 by 20
micrometers. 400 square micrometers. It was a standard that was used by New York, and I believe is still used by New York by the state environmental management group. It's really hard to quantify what that actually means because things have depth and they're in 3 dimensions, and so using an aerial unit is confusing. I'm sure it was originally pioneered to make it easier for whoever had to do it and fast when they were trying to assess water quality, but and it is still in standard methods. We've left it in standard methods because it is still being used, but I would say that 99.9% of ecologists out there are using natural units and cells.
Okay, one last question before we get on. This question is specifically for Ken. Are aerotopes always very clearly visible, as in common bloom forming microcystis species? I have a hard time distinguishing the aerotypes in limnographs, limnorefs.
Oh, limnorhapis. Yeah, it's true. Well, if they have a lot of pigment in them, they may be somewhat obscured, but generally you can see them if you get to a high enough power. I mean, the easiest thing is if it floats in the sample, you know it's got them. But yeah, they're usually pretty visible. You can usually tell that they're. Admittedly, they're more visible in some than others, but they're usually there. You can usually find them.
All right. We do have one more question, but it says that the attendee has actually left the room. So, but we're going to hold that one because I know this question. This question involves the automated methods for cell counting. And I know that that's not just a quick 1-minute answer. So maybe that's something that we'll dive into over the break, or maybe even during the live specimen examination later on. But we will definitely get Anne's opinions on that later on in the workshop. And with that, we're going to move into our next lecture. One more lecture before break. This is the field bloom lecture, and we're back over to Anne. So,
and this follows through. If you know what I threw makes your head spin, this will help some because you'll see microscopically what it looks like, and then macroscopically what you see in the water column. So all yours, Anne.
Thanks. Okay. So I want to go back here. All right. So everybody can see my water blooms, yes? I hope.
Yes, I can see
Okay,
it.
cool.
I'm good.
Cool beans. All right. So
Your bloomers are showing. Yeah.
Yeah, actually, if I'm a little antsy, it's because my heat is out in my lab. And of course, I'm in the middle of a workshop, so they can't do anything about it. So my fingers are getting a little nippy. So what we're going to talk about is how these things look actually out on the lake. And I'm only using images that we have confirmed what the primary bloom constituent is. And so if I've actually looked at this stuff, the pictures that you see will be from these blooms. So let's start with the stuff that is not a cause for alarm. Often I'll get calls about white powdery stuff on the water, yellow on the water. It's often, especially if it's in the April-May time frame, at least in the Midwest, pine pollen is the biggest offender, although there's, I've seen Vallisneria flowers also look like kind of like this in the water. And essentially they look like little Mickey Mouse ears. And it's only for a short period of time, 2 to 3 weeks, but it looks like it's a problem. It looks like it's an issue, especially if it's more yellowish rather than whitish. And so those will appear in the water. The biggest one are all of the duckweeds. So larger duckweed, major duckweed, minor duckweed, and watermeal. Wolfia is the smallest one. It is the smallest rooted plant, teeny tiny, really teeny tiny. And the thing is that when you look at this from a distance, it looks like it could be a blue-green bloom. One of the things that kind of tells you, oh, this isn't a cyanophyte. One, it's pretty green. It has some relief to it, but it's also at the edge of an agricultural field. And even though ag runoff can be high in phosphorus, these kind of macrophytes generally are in high nitrogen environments, and so often at the edge of fields we see a lot of this. Duckweed, this has a single root that comes down from the thallus of 3 fronds, 2 to 3 fronds, and often you'll see it on the backs of turtles and things like that. It doesn't bother them at all. Sometimes you'll see some lower O2, but generally it's in shallow systems. These are going to be a little bit bigger, so they're going to look like individual plants in the water. There is also a limna major as well, and you could tell on your hand, fronds are a little bit bigger, and cyanophytes are not going to do this. Even the ones that have some substance to them, like nostoc, never looks like this. It actually looks like a plant, and if you look at it closely, you can see the roots. This is a mixed duckweed consortium that was from a bloom on Lake Michigan, and we did get, this is down the bluff from my lab, and we did get a lot of calls about people being worried that it was a blue-green bloom. So some things that will help you determine it's not. One, see this nice horizon right here? So the 3D effect of it, although blue-green blooms can get into pretty thick scums, they don't transition in an even clear line like this. They're going to be blue right out into the water. It's going to be almost like paint or paste out into the water. This has a clearly defined edge to the flotsam that's come up on the shore, and of course there's everything is individual. With blue-greens, even if you can sometimes see the individual colonies, Gloria trichia, you can see the individual colonies, sometimes microcystis, and I have some pictures of that, looks very fundamentally different from duckweeds. These are kind of, I think this is the major one. There's multiple roots, and it's kind of purplish on the underside of the fronds. And it's actually a mix because there's gibba right there. So duckweeds can be very dense, but you're going to be able to always see the individual plants. It's going to look green like a plant, and you often will get a nice clear horizon where there is nothing up against the shore. Water fern or azolla, which is super cool because it does have a blue green in it, but it's actually internal to the frond in a chamber. And it is still an anabana. It technically is attached because it's internal to the frond. But the outer leaf itself, this is a true fern. It can be green, it can be red like it shows here. This is from, I believe, Kansas, and then I have a picture from Botanic Gardens as well in Chicago. This is a big problem because it will cause issues. It's not that it's not a problem in terms of ecology because it covers the surface, it limits the natural native plants that can be there, it causes oxygen issues, but it doesn't produce toxins. Rooted macrophytes, if you curly pondweed, which is an invasive plant but does not produce toxins, these are going to be rooted on the bottom. They're going to have distinct leaves. They're going to be quite distinct from what you would see as a paste or small individual colonies in the water. This is Enteromorpha, which is, this is actually taken during the Olympics, I believe. You know, we don't often see it grow like this in the States, but still bright green, lots of relief to it. So it's got, you know, a 3D conformation to it that you can actually get hands on. That's not going to be a cyanophyte. Cyanophytes don't do that. Even if they do have a form to them like nostoc balls do, they just don't look like they're green plants, and they don't have this bright green color that green plants have. Cladophora and Edygonium, which are some of the most common ones that we'll see on the edge of rivers or ponds, stagnant ponds. They start on the bottom, they grow up. They can be grass green if there's not epiphytes on them. By the time they get here, they're late in the season, they floated up from the bottom, so their whole fascia have come loose, they float up from the bottom, and they have a ton of epiphytes on them. If you see a lot of cochinees out here, chances are you've got a lot of Cladophora and macrophytes over in the littoral zone. It's very 3D, has a lot of conformation in the water, breaks up the, see the reflection here, clearly breaks up the reflection, a lot of substantial biomass to it. So blue-greens don't do that, cyanophytes don't do that. Now, sometimes they can be mixed. And so here's Cladophora, and here is Cladophora, but this is Lammermannii. This is Dolichospermum lammermannii. Notice that sometimes Ken says names differently than I do, which is probably differently than Chris does. Every taxonomist says the names the best they can. There's no right or wrong unless you're, you know, speaking Latin, and in that case, you have bigger issues, right? So say them however you need to say them to get the word out and get on with it. So communication is the key, not necessarily being able to say the thing the way everybody else says it, and we all do say it differently. So this is actually Stony Lake, and I have some other pictures of this later on, but this is the Cladophora, and see how there's some bubbles out here, but this actually is raised up. These filaments are raised up, they're quite thick, there's a lot of substantial biomass there. This is Fracta, has a laminate cell wall. And this is, again, that blue, green, and green mixed in. So this big stuff that's sitting up above the water is the macroalgae, that's the Cladophora. And this stuff that's in and around it is the cyanophyte, and that is the Dolichospermum. This was an interesting situation. So these samples came from Steve Hyscari, who is with the Minnesota Pollution, he's retired now, but was the Minnesota Pollution Control Board. He said, okay, we've had a dog death, and you know, these are some pictures, and he sent me this picture and this other picture from his hand, and said, well, is this a green? I said, yeah, that's one of the conjugales. It's probably, you know, either Magosha or Spirogyra. Send it along. And these are the pictures I took. The thing is that when I looked around a little more, what was mixed in with this was Dolichospermum cercinale, and there was a fair bit of it. And the thing is that to look at this, see the nice 3D conformation generally, I'm getting more paranoid as I get older, I would say this is no risk. It's just a lot of nitrogen in the water. It's not a problem. But there was a fair bit of Dolichospermum mixed in with this. It was producing toxin, and the dog deaths were confirmed to be from the toxin. So, you know, even if you have some nice thick stuff like this, sometimes there's cyanophytes that are mixed in with that. This is Hydrodictyon, one of the few algae that has a common name, waternet, and I love Hydrodictyon. It doesn't necessarily mean great things for the water. Who would go in that on purpose? I'm not sure. But it forms really thick, horribly thick, dense growths in high nitrogen systems, especially in the Northeast. And it can, it's nice in 3D here. What we see is that it's going to be, if you have a bloom like this, it's associated with super high nitrogen. And I've never collected it locally until 3 years ago. About 5 to 7 years ago, maybe even 8, they built a brand new PGA golf course. Jack Nichols, I don't follow golf, but a biggie designed it. And all of a sudden on the Pawpaw River, which is adjacent to this golf course, we are now able to predictively collect Hydrodictyon. So it's definitely associated with high nitrogen. It has a lot of different hexagonal and pentagonal designs, and the cells are often very differently sized. To give you an idea how big this is, that's Spirogyra. Heliodictygon, the interesting thing about this is I've only seen it 3 times, twice collected by me and once by Danielle, and she actually sent me this material. Notice again, it breaks up the reflection in the water. It's not super green like grass, but it's kind of a yellowish green. It is a green algae, but the fact that there's all this sheath, these cells are kind of vermiform and they're peripheral around a really turgid sheath, almost nostoc-like. And it's very cool. And when it blooms, it really blooms, but it's not a water quality issue other than to be unsightly and aesthetically, it's kind of yucky looking. Durinschia and Serratium, both dinoflagellates. Notice how here, so this actually was a deep sample. Ken took a sample. Serratium often will go down deep. It doesn't like a lot of highlight. It is facultatively heterotrophic. Durinschia is a very lightly thecate dinoflagellate, and it doesn't like super highlight either. This is a picture from Oklahoma. Notice you can still see the reflection in the water. It's right up above, right up to the surface, but just below it. So it's trying, it's modal, has 2 flagella, one here and one here. And it can moderate where it is in the water column, same with Serratium. And so it'll sit just down below that surface of the water because it's trying not to have issues with, photodegradation. So it'll be right below that surface. But anything that kind of looks like this, not likely. There's only a couple things this could likely be based on its oil slick-like look on the surface. One is this, and the other is Euglena. This, believe it or not, again, look at how thick that scum is. These are all diatoms. I mean, 100% diatoms. Mostly Nitzia with a couple of others, Navicula with a couple of others thrown in. This is actually slough that's coming off of a stream that's coming in right here. This is a recreational pond where people have like boats that you paddle boats at a park. Amusement park. So it looks horrific. It smells horrific. Kind of cool to look at under the scope because all these guys are modal. But it's other than to cause an oxygen issue, it's not a toxin issue. Purple sulfur bacteria, another one that looks very eerie and definitely warning signs of nature. What it's really telling you is, and you can see the purple here as well, is that there is anoxic conditions, pretty prevalent anoxic conditions, and Thiopedia looks a lot like Merismapedia, but it does not fluoresce. It is pink, purple in the water, which Merismapedia tends not to be, and it tells you the water, there are issues with the water quality, but those issues don't involve toxins. Euglena, again, see this, we got a here, not so much, but here it's, you can see the reflection. And this is a combination actually of Euglena and some Trichelimonas staying right below the water surface. It doesn't like to be photodegraded, like it's too bright up there, so it's hanging down below. And this particular one, most Euglenas do not produce toxin. I have one later in the lecture that's Sanguinea that does, but most Euglenas and Trichelimonas don't. And see, again, this is kind of like that Heliodictyon we just saw, has the colonies, the material stays together and forms clear ribbons in the water. Most blue-greens don't do that. Sometimes you see animal generated stuff, and these are Daphnia fipia, so resting cells from Daphnia at the end of the season. So this would have gone to a whole animal. This resting cell develops in the brood chamber, and these guys go down to the bottom. They're very dark. They can be at some points buoyant before they go down to the bottom. And look like it's algae that's buoyant in the water, but once you look at it, each of the individual things is macroscopic. It almost looks like some of the seed pods from macrophytes that get stuck on your clothes when you're hiking. Pretty tiny. We saw them in Lake Hood when we were sampling with a population of Daphnia magnia in Alaska as well. When they're there, they look, their can be overwhelming in the system. So let's look at some toxin-producing ones. Let's start with open water. So here's Euglena sanguinea. I believe this has now been moved. I don't remember the genus. I don't think it's been moved into Lepicinclus. I think it's been moved into another one, but I have to check. They reorganized all the Euglenas and moved most of the Euglenas into Lepicinclus. Kind of has a dusky red to it. Again, you could see this kind of oily look to the water, this slick look to the water. These guys have gotten pushed up to the surface, but these are still right below the surface, like a lot of euglenoids tend to do. The toxin is an ichthyotoxin that we know of, but then again, breathing it in doesn't do you any good. Aphanizomen and gray seal. all of these pictures are bad, 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 bad. If you see the water look like this, don't do any of these things. If it's this dense and it's got this much biomass in it, you need to be as far away from the system as you can get, because this algae, what he's doing, he's breathing that in now, and he's breathing in a fair bit of it. So inhalation is not very well specified. I'll talk about that a little later in the next lecture. When you look at this under the scope, it has the typical aphanizomenon look to it, except these are not inflated like they are in Flassaque, and the aconite has little end caps to it. That means that in order to speciate this thing, you have to have a mature aconite. So Andy Chapman is really conservative. He rolls things all the way back to the order. I usually have a pretty good sense of what genus it's in, but I'll put uncertain and put a note that I don't have mature aconites. This is what a Cylindrospermopsis, what was Cylindrospermopsis and now is Raphidiopsis raciborski. This is the straight morph. This is Ball Lake in Indiana. This is Thunderbird Reservoir in Oklahoma. Makes the water look muddy. The interesting thing is, or an olive green, it does not scum. It's all the way through the water column, and it is a late season bloomer. So both of these pictures were taken, this was Labor Day weekend, and this was taken in November. We did sampling for NEAPMS when we were in Oklahoma last time. So hopefully we'll be in Oklahoma in person again this coming fall. And this is what the straight morph with aconites or heterocytes at both ends look like. Often you don't have one at one end and you have one at the other end. If you're truly unlucky, you have none at either end and it's just pointed, which can make it look a lot like Cuspidithrix tropicalis. This is what I was talking about with some of that clumped up blue-green. Again, yeah, they're clumps, but you can't see individual colonies. The clumps are indistinct. They have different color. That has to do with how long it's been at the surface and how healthy the cells are. Microcystis aeruginosa can do this, or it can be evenly distributed throughout the water column, and I have some pictures of Virigulus. So here's the issue. They believe that many of the microcystises are actually all ecomorphs. But again, like Ken said with the Synodesmus desmos group, it tells you something. If the sheath is really thick, like Novecii, then that tells you something. And usually it's often in high salt environments. There's also Viridis, there is Flassaque, there's Wessenbergii. So depending on which microcystis species it is, having different sheath characteristics kind of tells you something. This was actually at Disney World, so just beware if you swim at Disney World. This is Microcystis viridis. So notice this one was all nice and clumped and peripheral, scumming up around the edge of the lake, caught in the grass there. This one evenly distributed throughout the water column. It's packeted and the sheath is thick. This thing here is actually Pseudanabena musicola, also appears in Microcystis aeruginosa as well. And it is not obligate, but they often co-occur, and the Pseudanabena, they're real short chains, is parasitic within the sheath of the Microcystis. Delicospermum lemmermanii used to be Anabena. This is a dead carp. Yes, I took that picture, and this is the same lake. This is Big Pawpaw Lake in Berrien County, Michigan, which is just northeast of where I am. And so the year after this bloom was this Microcystis viridis bloom. And then we got Anabena or Delicospermum lemmermanii. They often look like bow ties in the water when they're mature, and they have often little protozoan vorticellas off of them as well. This is the same species, Delicospermum lemmermanii, in Stony Lake, northern Michigan. This is what we call a jar test, which is literally, like Ken said, putting the algae in a jar and seeing, does it float or not. There's very few things that will do this, and Aphippia are one of them, but you know what those look like now, right? And so if it looks like this, it's a cyanophyte, it's a blue-green. Notice the difference in the colonies. Cells are a little bit bigger, a little bit rounder, still in the bow tie conformation, but it looks very different than that. So environmentally, same species, same aconites, same ultimate ID, but the ecology is a little different, so the cells look a little different. This was July, and again, it's forming surface scum and starting to form windrows. Not 3D in the water, right? It's not coming up out of the water like you would expect something like the Clodophora and Eugonium, something like that, to do duckweed. This is that same picture, Ken, close up from Lake Mendota and Dolichosperma mendotae. So unlike Lemmermanii, there's lots of clusters of aconites that are spread throughout the colony. And there is one major difference, which you unfortunately can't see in any of these pictures, but is in the supplemental materials that you have. In Lemmermanii, that goes aconite, heterocyte, aconite, at least aconite, heterocyte. They must be adjacent to each other. In Mendotae, they are distant from each other. So if you see aconite heterocyte, it cannot be Mendotae, it cannot be Circinale, it cannot be crassum. They're always distant. So that aconite structure with this colonial structure, like you see in the bow tie, that's always going to be Lemmermanii. If you see clusters of aconites, they're distant, along with some other cell ID characteristics, then it's more likely to be Mendotae. Again, this is Mendotae. This is from Monona Lake. Now, see now, this is breaking down some, and you get some of that purple pigment. That's the phycoerythrin. There's phycoerythrin and phycocyanin. Remember, one's got a little bit more red to it. One's got a little bit more blue-green to it. And again, kind of flat on the surface, and these are sticks and things caught in the water. But it's scumming in the water for sure, but it's not 3D like you would expect those greens and ferns to be. And here's that cyan color, and then here's the red color. So again, this was in the same lake as this, but this had clearly, this part of the bloom had dissipated quite a while before, so all that's left is the phycocyanin pigment on the rocks. It looks literally like somebody painted it. This is Phanazomenum flossaqua. We threw a rock in the water. So you can see this kind of cloudy edge here, and then it's still really murky. That's very different from what we saw in the duckweed, very different from what we saw in the euglenas, very different from what we saw in the Heliodictyon. So, you know, it's going to be kind of cloudy up to this edge, but it rarely, blue-green cytophytes rarely form a perfectly clear and perfectly open edge. Even here where we threw the rock in, that disturbed the surface scum, you can see all those individual grass flakes. This is the grass flake morphology. In the live session, I actually have a video of one of these flakes from the fascicles from the workshop in November. These are modal within the fascicle. They're not attached to each other hard and fast. So they actually move a little bit within the colonial fascicle there, within the grass flake morphology. They're itty bitty grass flakes, but they still look like teeny tiny grass flakes in the water. This is from Klamath Lake, much bigger grass flakes in the water. You know, these things are on the order of a few millimeters up to a couple centimeters long for the really, really big ones. And again, those are you know, hundreds, if not a few thousand filaments that are right up against each other, and they're kind of doing this, often attached to each other or conglomerating with each other near the heterocyte. This is Rhonchinia nigliana. It used to be the old Ciliospherium. Again, this is just starting to form the scum in the evening. You can still see the reflection here, but here they've all been pushed up to the top, and there's ones coming up from the bottom. This is more brown. This is more kind of olive green with looks a little bit more aged than this one does. And they have these doublets in the cells, and the cells are peripheral in the colony. So what really separated the Ciliospherium into Rhonchinia and Ciliospherium was the fact that Rhonchinias all have erytopes. And it is an inconsistent toxin producer. Again, this is that same bloom. And the colonies look like jelly beans in the water, look like gumdrops. This is Goliatrichia. I love Goliatrichia. You can see the individual colonies look like little dots. They often don't scum. It's unusual to see them this concentrated in the water, but they can be very dense. And you can see these individual colonies. I actually have some to show you later in the live scope part of the workshop. And they're radial, so they're connected down here at the center of the colony. That's where the heterocytes and the rare aconites are. These actually have a curved basal sheath around the aconite, which often does not survive in the sediments very well. It's often a boom and bust bloomer. So you'll see it, and then you won't see it for several years, and then you'll see it for 2 or 3 years, and then you won't see it for several years. And it's hard to predict when it's going to come up. Often Daphnia will chew off the outer parts of this, kind of like deer with woody brows. That's not something they're going to do unless they're desperate, and they'll just leave kind of a shaved colony in the center. This is something you're not going to see. Well, you could see actually in the marginal areas where there's a little brackish water. So this is nodularia. There are not a lot of species of nodularia. It forms both actinites and heterocytes, but they look a little funky. And again, Wayne should not be holding that way. They produce nodularian, which is very similar. It's a cyclic peptide, very similar to microcystin, can be highly toxic. Definitely causes fish kills, definitely causes issues for humans. Notice how brown it is. This is really thickly scummed. This is Antelope Lake, which is near the Great Salt Lake. And you got to be up above 10 to 12 ppt salt, parts per thousand of salt for that to do that. In the terminal that goes from the Lake Tahoe down the Truckee River, and the 2 or 3 terminal lakes are Pyramid and Walker, huge blooms of nodularian as well. The salinity has really gone up in those because of the drought, and it has highly toxic. In fact, the nodularia from Walker Lake, Greg Boyer said was the most toxic population he had ever seen. Planktothrix, and I have some pink planktothrix for us to look at. I'm saying CF prolifica, and the reason I'm saying that, we actually have some out as we speak, but we don't have an answer yet about the genetics. Supposedly, rubescens is European and prolifica is US, although often people will ID the pink one also as pink agarity. It'll be very interesting to see. This particular one was from Kansas, and I have the one that I'm going to show you came from Minnesota. Again, it looks pink. It has aerotopes, but it mostly just kind of looks refractive. It's one of those, when somebody asks, can you always see the aerotopes? You can see the visual disturbance and the refractiveness from the aerotopes in Limnorhynchus and Planktothrix, but it's hard to actually see big voids like you would in a Microcystis or a Delicospermum, a phanezomonon. They tend to be smaller. And they're pretty interesting. This is very commonly a winter under ice bloomer, and they are already having blooms in Minnesota. And this is what it looks like. This is Cedar Lake. This was actually May, late May, or late April into mid-May. So pretty chunky here, kind of looks like a Microcystis in a way. Planktothrix and Microcystis are the most consistent toxin producers. This is from Canada. Has an odd iridescent look to it, looking down through the ice, and this is what it looks like, which actually this coloring is very similar to this coloring. Probably my guess would be the same species. Again, really predictable. 85% of the time it's going to be producing microcystin. That's a bad thing. So if you see this in the water, you shouldn't ice fish through it. Fish do keep microcystin in their flesh for a period of time. And I was just watching a talk by Ellen Priest the other day that looks like it could be as long as 6 to 8 weeks after the bloom is gone. So just kind of keep that in mind if you're fishing. We do have benthic producers, and this really disturbs me because not what you would think of with our previous pictures, right? So this water is actually, this is Zion National Park. This water looks pretty clear. This is what this stuff is and what this stuff is. This is nostoc. Nostoc is really hard to ID, but it has these really turgid, robust colonies, and it is a pretty predictable microcystin producer, and it also produces antitoxin A. It will produce it upstream and have it flow downstream. So you don't necessarily detect the toxin where the algae is. That is warning signs of nature, something to keep in mind. And it's equally as prevalent in standing water as flowing water. So this happens to be a creek. That's because most of Utah, the impoundments are reservoirs. There's not a lot of natural lakes in Utah. Excuse me. And the flowing water is where they're having a lot of these issues with benthic blooms. But they've had dog deaths in Michigan from benthic lake populations of some of these blue-greens. So this was something that really was disturbing to me, because this is, again, we have this out for DNA analysis. It's back, but it's being analyzed. Barry Rosen and I think it's Tychinema, but there's a possibility it could either include or partially be Microcoleus. The old Formidium autumnale is now Microcoleus autumnale. And this does not look like a blue-green. This does not look like a xanthophyte. This looks like a diatom scum. And it is all this stuff. These are Barry's pictures, and my pictures look the same of this material taken from this bloom. So the hard part is that it does not look, when you're looking at the water from above, this does not look like water you should be concerned about if you are a plankton ecologist used to lakes. And what I would have thought would have surely been something like diatoma or a whole bunch of tube dwelling, cymbellas or cyanoemas. This was all this Tychinema microcoleus, something to be pretty concerned about. So actually, I'm done a little bit early. So do you guys have any questions?
Okay, yes, we do have. So yeah, we are about 12 minutes ahead of schedule, and then we've got our 15-minute break. So we might actually have a little bit of a extended break here. So if anybody has any questions, this would be a great time to toss them in the question chat. So we do have a question from Andrew. I'm going to butcher this last name, Edgecumbe, I think. Is what is the type of algae that looks like horsehair?
That's probably Microsyra.
That's what I've heard it called. Yeah, again, there really aren't common names for these, but you'll hear it called horsehair algae in some places, and that has been what Microsyra, which we've also called Plectonema, and has also been called Lingbia. But yeah, it's a fairly large filamentous blue-green.
Okay, and then this is the question from earlier that I teased. This is from Jason Adolf. How do you feel about the many automated methods for cell counting, FCB, FlowCam, and others?
Yeah, so I actually use one of those in my lab right now. It's IFCB, which is imaging flow cytobots. That's where I got the FCB from. It was developed for, actually both of them were developed in marine systems. So you have two options, right? You have the basic Coulter counter, which is just going to do particles. You're really size limited, and then you have the imaging stuff, which is actually going to help you ID. So when I go down, when I do a count, I did a count before I came in this morning. Totally buggered it up. I have to go back and redo it. But other than that, because I was distracted, it took me about an hour. Previous to that, it had to be logged. For me, it had to be mounted. But there's a fair bit of, if Ken were doing it, Ken would have to settle it. It takes a fair bit of prep time and a fair bit of manual time in the scope. What that means is, for a production lab like mine, is that the average time to process a sample is anywhere from 14 weeks up to maybe a year, depending on how many samples we get. So I hope Todd Teachin's not watching, because I'm actually further behind in this stuff. If you use an automated counting system like the FlowCam, like the IFCB, you can do a higher level of analysis faster. Now, the caveat with that is that I don't know if anybody remembers me saying, but it bears repeating, there is no perfect way to do this. So no matter whether you count manually on a Cedric rafter, you're on a compound scope, an inverted scope, you're doing permanent slides, whatever method you choose, there are going to be bounds on what it can tell you, and there's going to be issues with it. And so I like my method because I started it a very long time ago, and based on the grazing experiments I was doing and the number of samples that I was generating, it was a good solution for me. But Ken does a settling method, and it works just fine for him. Andy Chapman does a settling method. It works perfect for him. He works on an inverted scope. Ken and I both work on compound scopes. So the IFCB and the FlowCam, without getting specific about which one, and I use an IFCB in my lab, as long as you have an expert to train it and an expert to moderate it, can tell you to species group about what's there. They are not, neither system is super quantitative. They're within, the IFCB is within, depending on what else is in the system, 10 to 20% of what I would count manually. It's usually going to underestimate rather than overestimate, which, if you're trying to do risk assessment, is a good thing. I would rather underestimate than overestimate. That lets you be conservative on what you're seeing. You know that what you're seeing is less than what's actually out there. It has a size limit. Both the IFCB and the FlowCam have size limits. So you're looking at 250 to 300 microns, maybe up to 600 in a FlowCam. That's not very big when you're talking big fascicles for a Phanosomnon, which luckily for us hates bottles and falls apart. That's a good thing. Microcystis large Dolichospermum colonies. They can get caught up in the pre-screen. So it's always a good idea to look at that pre-screen. I can tell you almost nobody does. And to look at the images, aside from whatever classifier you use, and getting a decent classifier is a lot of work. So even though the IFCB is less of a turnkey instrument than the FlowCam is, it still takes probably 6 months to a year of setup and experience before you can actually start using the results. And then you really need somebody looking at every single sample and perusing the images, scanning the images to make sure the classifier did what you think it was supposed to do. Every single sample that goes out of my lab, and we're now up to a couple thousand a year, in addition to the 2,000 to 4,000 I count, because I'm the only taxonomist. I look at every single sample before it goes out. So, but the thing is that if I didn't have Phoebe, I think you should name your instruments. Mine's named Phoebe. Phoebe can do 1,000 to 2,000 samples. I'm not going to get to. I'm not going to look at those samples. There's no way. I'm counting 6 to 12 hours a day as it is, 5 to 7 days a week. So it'll in the samples we can get out data with an IFCB in 24 hours. Now, it's not that there's a limit to that. There's only so many of me, and there's only so many samples she can run a day. But compared to if I had to stop everything in the lab and stop my counting, which I don't have to do because I can look at those images off hours, I think it's a really powerful tool. As Ken would say, a tool in the toolbox. It's not the only tool in the toolbox. And if you need fine level data, if you need to know the other taxa that are there, it's a great monitoring tool, but it's not detailed like a count would be. So it's not like, oh, and the same thing with genetics. Genetics are awesome, but notice I have been waiting now 8 weeks, and it took us 4 months to find a lab that could do the DNA on 2 species that we already know produce toxin, what habitats they're in, and we know what toxins they're producing. So it is a curiosity thing. It's not really helping me to know the genetics. It's just a curiosity thing so that I get better at what I'm doing. But I think that the automated solutions have a lot of power to them if they're used in concert with an expert counter. Problem is a lot of these instruments don't have an expert phycologist that's associated with them. So either somebody trains it and walks away and things shift over time, or somebody trains it who doesn't know algae to begin with, and some of their IDs are okay and some are not, even with like the libraries that that FlowCam produces to go with their instrument. It still takes an expert. You still have to understand your data. There's no substitute for understanding your data.
Yeah.
All right. I warned you that was going to be, and that's like the 5-minute answer. That's not even the 30-minute full detail one. So
give you a little bit of a practical take on this. I look at samples. I do not have an automated instrument. I look at 200 or 300 samples a year. What are you doing, Ann? 2,000 to 4,000? Yeah, I can't do more than that because I have other things I have to do too. I don't just look at samples. If you have a bloom, and it is a real bloom, I don't have to settle it. I can put a drop on the slide. I can make a fast count and I go, yeah, you got a serious problem with species X. But if you're trying to track it, if you're a water supplier, if you're a big time recreational lake complex with lots of users, and you need to know what's going on before it happens, somebody's got to look at every one of those samples, and they're too dilute early on. There really is no way that there will ever be enough decent taxonomists out there to do this for you. It has to become automated. And it's only in recent years that technology has caught up to the point where we can sort of trust it. You can overcome with quantity of analyses some of the deficiencies of our time limits, and that's what really helps. Somebody still has to understand it, they have to look at it, they have to make sure they know what they're looking at. But for the purposes of management, I think it's got to go the way of the automated system, because there's no water supplier that can wait even 2 weeks, let alone 2 months, to get the results back.
And like I said, we can get results out to them. So by myself, I can do 2,000 to 4,000 samples a year. With Phoebe, I can do an additional 1,000 to 2,000 samples a year, and I buy more instruments, which I'm saving up for. Then we do more samples, right? So you know, the other thing too is that like we were saying, there is no perfect thing. So you really have to define your goals. You should know your goals before you take your samples. Don't come up with goals after you take your samples. That's
Yeah,
bad.
good point. Yep.
Bad. So
I have one now. I'm waiting for delivery actually today or tomorrow. Someone was moving water between different reservoirs, and then they noticed when they brought it in and treated it, I think with alum, a coagulation thing, they got all these stringy things that were clogging everything up. They said, "We don't know what it is." They said, "Well, you brought the water in from somewhere else. Has it got algae in it?" "We think so." "What is it?" "We don't know." But it's doing this. It wasn't doing this before. Well, something bloomed in your lake and we need to know what it is. "Well, can we get a sample?" "Yeah, send one over and we'll look at it." But you have to look at them still, and that kind of quick thing you can do. But tracking and monitoring pretty much has to happen on a regular basis to be able to, and you'll get some of this in the management lecture, to stay ahead of it and to prevent blooms, not just get rid of them. And that monitoring is going to require a lot more looking than anybody has time to do without an automated system.
And just a caveat, just because it's green doesn't mean it's algae. So Pseudomonas, which is a filamentous bacteria, is green. And I had the folks at Cook Nuclear Plant contact me a few years ago because they were sure that one of their internal tanks had algae in it. And their comment was, "Well, it's in the secondary water supply. It never sees the lake." And I'm like, "It's not algae." So if it comes out of a septic system, it's not algae. So unless you have much bigger issues. So,
Okay, so I guess
I
we're up to a break at this point, so we can hang around and talk about stuff, but we promise not to say anything important until 2:30.
Yes. So once again, thank you to our sponsor, Earth Science Labs, and we will jump back into ecology and cyanobacteria toxins starting at 2:30.
If people have other questions or whatever, we're happy to talk about stuff.
Well, hang so they can unmute themselves, right, Chris?
I'm not sure they can. I think they can't. So, but if anybody just puts something in the question queue, I will pass it along to Ann and Ken. We'll get them there eventually.
Awesome. Well, I'll take a moment to try to warm up.
The heat's just out there?
It's just out. So this has a separate, the only reason I'm allowed to be here is because our upper floor, this suite, has a totally separate HVAC system from downstairs.
Okay.
So because I went to the funeral on Friday and was with, out and about in the world, our procedures here at the lab are is that I have to quarantine for 2 weeks before I can come back in the lab proper where there's people.
Yeah.
But upstairs is my teaching scope. And so they got it all ready for me to be able to teach this workshop and got my Ethernet cable in here. So far, I've not been kicked out of my own workshop. Yay. And, but I was in yesterday to hook up my laptop, do a dry run with you guys. It was fine. It was warm because yesterday was actually colder. Yesterday started at 16 and the high was 24 and I was okay in here. I walked in this morning and I'm like, oh, it's nippy. And it's 56. So, I mean, I've been colder for sure. But yeah, it's to sit here and do 56. It's a little nippy. And it was so funny. I got to show you guys this. So because I'm stuck up here, I can't go down to the fridge. I can't go down to the water. I have a bathroom, thank God. So Katie and my grandson Harry made me lunch for today, and they made me little olives on toothpicks. So I have all the cool stuff.
Very nice.
Awesome.
Yeah, yeah.
We do have another question. This one's from Lorraine. What is a good basic microscope to identify algae for quick field ID?
I really. Go ahead.
Do you mean out in the field as opposed to in a lab?
It says for quick field ID.
I mean, there are digital microscopes. If you have a power source, you can get an image right onto a camera or a little screen, and that's not bad. Those are okay. I think $600 to $1,000, you can get a decent one.
Yeah, I have a couple of things. So I have a Muscam, which goes up to 50x. It's like a stereoscope. You can see Pandorina colonies pretty predictably with it. And then there's some attachments you can get for microscopes. If you want a microscope, you can get some amazingly Amscope, and a couple of they're like wholesale microscope producers. They're almost all Chinese microscopes. But you know what? I've gotten some teaching scopes that have held up pretty well that are LEDs. They're chargeable LEDs. And they have a pretty decent image on them. And they've lasted 6th graders for about 6-7 years. So, you know, that's pretty impressive. You can last through a 6th grader. If you take care of them, they're bright field, but you know, they'll do the job. If you want a cheap scope for in the lab, I really like Olympus's teaching scope. It's a CX43. It'll cost you a couple $1,000s versus, you know, $200 to $500. But if you can get them with phase, they're decent scopes. So
Okay.
I prefer Olympus. But.
That was and yes, Lorraine did specify it's IDing grab samples collected from the field. So I think you covered that with the latter part here. I actually have a question for the two of you. Have you ever seen and or ID'd watermelon algae, the stuff that occurs on the snow, not like in the water, but like apparently like when snow melts or something?
Watermelon color, probably. Chlamydomonas. Yeah.
Chlamydomonas.
Okay.
Yeah, there's a flagellated green that will grow on the top of the snow, and it's pinkish to reddish in color. It has a lot of hematochrome in it. Yeah. That's funny because when I was in college, myself and another very nerdy algae student wrote a phycological key to the common detritus of algae samples. And we gave everything Latin names. There was Canis berii. And one of them was Cedium watermelonii because we were always spitting watermelon seeds when we were doing the sampling and such. But that I've never heard it called that. But yeah, that's the red algae. Well, it's green algae turning red, growing on the surface of the snow, and it's a type of Chlamydomonas.
Is it common in the Northeast or
not common?
Rare?
I've seen it once or twice.
Okay. If you've only seen it once or twice, then that's not common.
But I mean, really, if I'm out skiing, I'm not really looking for algae usually.
I think that it actually happens at elevation, pretty commonly.
Higher
like
elevations, yeah.
Yeah, and then like in the Northwest.
Yeah. Okay.
In the West.
Great.
I tend to worry less about red snow than yellow snow.
That is
you
an
shouldn't
important
eat either, though, right?
Yeah.
Okay, that's all we have question-wise for right now. So we will we had about 10 minutes or so to kind of hang out. And I will mark
ecology and cyanobacteria toxins next, and then the control methods. Okay.
Yeah. Yep. So, Chris, how many people are actually in on the workshop?
There were 123 right now.
Yeah, we capped out at, I think it was 134. I think was the highest. I think.
Big group. Yeah.
Is it okay when I resend the link? Because Amy had mentioned I can resend the link to the materials to everybody. Because there's still a few people who like registered last night. Can I also send a link to I want to do a Google Doc for an evaluation for the workshop. We've never had a way to really have people evaluate the virtual workshops.
Yeah, that will be fine.
Okay?
Yeah, we can do that.
Cool.
So yeah, we will. If Amy has not done it already, we will resend that link out to folks that has all of the workshop materials in it, the book, and I assume your PowerPoint slides are all in there and everything. I would like to remind everybody that at 5 o'clock tonight, if you guys are not all GoToWebinar'd out, we are doing a social, virtual social, throw back a virtual adult beverage, and we do actually have Solitude Lake Management actually put together a little plant ID quiz that's web browser. It's not the same as it normally is with our live samples in the room. Yeah, I miss that too, but it's the best that we can do.
Is there a code you put in to get the beer?
You got to provide your own beer. Although I think I
own. I'll get what I want.
Yeah, I think I owe both of you at least one beer when we get back together again, hopefully next year.
As it turns out, In Michigan, we are Group 1B, so I signed my lab up for vaccines this week. So we'll see.
Excellent.
Yeah.
Okay, we do have another question. There's a question. This is from Bria. Hey, Bria, good to hear from you. Going back to the microscope question, Ken mentioned that there are scopes that you can get pictures with that aren't all that pricey. Can you discuss any specific brands that you would recommend?
I don't know the names off the top of my head because I don't have one, but I've got several clients who went out and bought them because they wanted to look at stuff themselves. And they hook directly into a computer. I should say I bought an off-brand Chinese one just to have as a backup and let somebody play around with if they wanted to. And that hooks into the computer screen so you can actually look at it on the screen as well as in the scope, and you can easily take pictures. But there are ones that are meant, if you look up the EPA Cyano Lab, they have some recommendations on there, I believe. But either you're looking at it and a cord goes into a computer, or it actually has a little screen on it that you will see the image on, and you can tap and get it, take a picture for you. If you have pictures, and it's so easy now to send them to people. I mean, folks like Ed and I are happy to say, oh yeah, that looks like such and such, or could you take that again, more of a close-up or whatever. But it's quick and easy. We don't want you to abuse the privilege, but we do not mind helping it all so people learn what they've got. And it's really easy to to match your image up with a zillion things online too.
Okay. We have another question. I think you guys will both like this one. How long does it take to become an expert phycologist? And this is from Benjamin.
I don't know. I've never reached it.
Uh-oh. Come on, you gotta give some of our attendees some hope out there.
The line we've used in the past is if you spend 200 or 300 hours on it, you will have enough proficiency to believe that, yeah, okay, I'm in the right ballpark here. The old statement about becoming an expert in anything, 10,000 hours, probably about right.
Yeah, and the caveat I would put is that it's not just the time you spend doing it. It's verifying your results in QAing and being QA'd by other taxonomists. Even I'm occasionally wrong, and or I'm occasionally uncertain when sometimes you just have a brain fart and you're like, that just doesn't look right. So we always, Ken and Barry and I and Andy are always sending stuff to each other. So find your groupie, your algae groupie group, and don't be afraid to send send images back and forth.
It takes a lot of time, and of course you can't just spend a couple hours here, a couple hours there. It pays to spend a bunch of time doing it and then move on to other things. But I'll sit down and do 20 samples in a day is a lot, but 10 samples is not unusual. And I feel pretty comfortable. If I haven't been doing it for a while, I have to go back to that first one and go, okay, did you miss something here?
Yeah, I've had to fix things. Yeah,
it's really nice to do the same lakes year after year. You can go back and look and say, okay, what did I find before? And then say, okay, what did I just find out? Oh, geez, yeah, that's a such and such. It's not what you called it. It's tricky.
Because if I'm doing like monitoring, I really like the client to allow me to have the whole season at once if they don't have to manage.
Yes.
Because then I can see how everything plays out through the entire season.
Right. Fall is algae season for me. For a few clients, I look at stuff weekly when it comes in so they can react to it, mostly during the summer.
Right.
But really, most places send me their dozen or 2 dozen samples at the end of summer, September. And my typical Sunday afternoon, I put a football game on in the background and do algae. I never actually sit and watch it, but if it's something good, I could take a glance. And I just get algae done in a burst, a few days a week.
Yeah, I think that's similar to us that specialize more in the aquatic plants. You learn to take good records, to keep good records of what were found at specific places before, so that you can reference that. But then also to have your group of people that you can kind of bounce things off to, and like Ken said, we do the same thing with pictures, especially with cell phones now. I mean, it's easy to snap a picture, pull the plant right up out of the lake, take a picture, and send it off to some people, and probably get a pretty good ID quickly. I mean, again, or at least to get you in the ballpark of, is this something that's a concern, or is this something that's, probably going to be native? But it's the same thing. Don't be afraid to make mistakes. Don't be afraid to change. your mind or change your ID down the road, because you might get that better plant sample. Like, and definitely in plants, you might not have flowers or seeds.
Right.
So
You
you'll
might
be
get that
like,
structure that's like, yay.
exactly. You get that distinguishing characteristic, and you're like, boom, now I know. It's like, I know it's not X, Y, or Z. It's probably A, B, or C. And then, maybe 2-3 years later, if you keep good records, you might actually get that ID when it actually goes into flower, and you just happen to be on site when you get it. So similar thing. It's probably a taxonomy challenge overall.
Yeah, the worst thing are little green balls, little round things that don't seem to be attached to anything else, and you're never sure, did they come this way naturally? Is this because of the preservative? Did they fall apart over time? Is it actually green algae, or is it a blue green? And we just refer to the LRGTs, little round green things, or Confusococcus obscurum, and if it's abundant, you need to figure it out. If there's a couple here and there, I always use my algae mentors line, Hannah Crosdale. She would say, you need a good microscope, because if you can't see it, you can't identify it. But if after all that you still can't identify it, you didn't see it. And just nobody ever parked below her window because she would throw slides out the window, just get frustrated with it, just throw them out.
Well, I think too, Herbert responded to somebody on a listserv a few years ago that they were asking, well, how much can you really identify? And he said if the average plankton ecologist were honest, they could only identify about half of what they see. I feel like I could do better than half, but I have categories for sizes and shapes and divisions all over the place.
I have an other for almost every one of about 20 categories on my sheets. Sheet's 4 pages long with very small lines. So there's probably a couple hundred things on there. And I see mostly the same. And I am sure that I have conveniently mentally called something X when it was really X sub 1, because I had it on the sheet and I didn't have to go look it up. But yeah, and you're always finding something new. And it's usually the very first sample that you need to rush.
It's the first sample on the day with a hangover.
I get samples from one of the companies that make sonic devices just to look at. Sometimes it's before they put one in, sometimes it's after. I don't know. I'm just looking at it and saying what's there. They're not usually going to send me a clear sample. It's going to be full of something. It doesn't need to be settled or concentrated. I can drop, put it on the slide, and see what's in there. And 1 out of every 20, it's like, oh man, I have no idea what the heck is this? And usually it's little round green balls, or it's some weird shape. I had one that time we went around in circles. We thought it was some little obscure flagellate, and we found it in some really old manual somewhere. I forget what it was called, but it was like nobody ever saw it before. Well, somebody saw it before. We'd never seen it.
Right. I add new codes all the time. I see new stuff all the time, which is fun. But it's always on the day I can least afford it.
and the other thing you have to watch is what I alluded to in the taxonomic lecture, when there are things that change their morphology in response to water chemistry. Very low nitrogen to high nitrogen is a stimulus for certain greens to change their morphology. And you can call something X when it was really Y just because the nitrogen changed. That's where it's really nice if you have a set of samples and you can go back and watch it change. Or it's consistent, but you keep seeing it over and over. So something's nagging me here. I have a feeling there's something more to this, and you can figure it out. When we do the scope time at the end, we did that the first time ever with the NEAPMS conference, and it was fun. A lot of the feedback was they liked watching us argue about what we were seeing, because you were never sure, and it's okay.
All right.
Okay. It's better to be approximately right than exactly wrong.
Yeah, exactly.
All right, so it is a couple minutes after 2:30, so we're going to jump into our next lecture. This is the ecology and cyanobacteria toxins, and Anne will be leading this one up. So take it away, Anne.
All right. So, this stuff is complicated, right? But the whole point of the whole thing is to understand how these things are interacting in the environment and how to manage them, right? How to effectively manage them or not manage them, depending on what your decision is. And so this primary part of my lecture is essentially what are those things that contribute to what you actually see in the lake, and then a quick review. And this is a real this is definitely a satellite view of cyanobacteria toxins, what the basic categories are, what their effects are. So The really important thing to understand is a lot of times with population dynamics, it depends on where you are, okay? So we all have to use the same wording. So phytoplankton are algae that are suspended up in the water column, and periphyton are attached algae. And then there's this in-between group called tychoplankton. And tychoplankton is essentially bottom algae that gets moved up into the water column when there's a lot of turbulence or there's been an event like a flood or a geologic event. So phytoplankton or suspended algae, you know, you can see blue-green blooms, cyanophyte blooms in large rivers. It happens a lot in Minnesota, actually. You can see them in slow-moving streams. Periphyton are important not only just in streams, but also in the littoral area of lakes. And we don't do a good job with periphyton. We don't do a good job with littoral algae in general. So also important are stratification schemes. So if, and that's affected by a lot of stuff. So first thing is, okay, if you're sitting where I am, I'm sitting in southwestern Michigan, we generally have our lakes set up in the spring. They're stratified over summer period and they fall out again in fall. So it's isothermic from the winter into the spring, and then the lake starts to set up. That's based on how deep you are, how much wind action there is, what your sunlight regime is like, where you are in the country or the world, what your water chemistry is like, and what else is going on in the watershed. Because if there's drawdowns or there's other things happening, then that's going to affect how strongly you can stratify. If we think about lakes that are in, you know, typical what we think of a diamantic lake, you have an epilimnion set up in the fall or set up in the spring, stays there all summer. This is where the bulk of the action's happening. You might get a deep water metalimnitic pulse, and then down here, if it's deep enough and there's enough going on, your hope is that it doesn't go anoxic, but often in deep lakes anyway, down towards the bottom it's anoxic, and very productive lakes at the bottom it's anoxic. So the chemistry at this bottom part is very different than the chemistry up here, and therefore the pressures on the algae are going to be very different as well. So the processes that affect population dynamics, kind of like yin and yang, right? There's always, what you see is always the result of a growth and a death, always a growth and a loss at the same time. So I think one of the most interesting things when I do outreach in 6th grade and I talk to kids, well, when do plants photosynthesize? They had no problem going during the daylight when there's light out. And I said, so when do they metabolize? And they always raise their hands and say, when it's dark out. And well, not so, right? You're always metabolizing, and then you have growth processes going on during a certain part of the day. So those two things are always happening, growth and loss at the same time. So when we're talking, and I think I need to do that. Sorry. So for growth processes, primary production, right? You're controlled by light, temperature, nutrients, algal physiology, how healthy you are, and competition, right, between different taxa. Heterotrophy, so that means you're augmenting primary production, and that's dependent also on physiology and environmental conditions. It's often cued by environmental conditions. Release from the sediment, so that's going to be not so much of a growth thing as a importing and augmenting the population. From resting stages, could be turbulence, different life stages, and colonization, which is coming from upstream or an external source like ship ballast water or, you know, proverbial duck feet. So let's talk about each of these just a little bit. So primary production is adding biomass. It often, you're going to have the highest productivity at intermediate light levels. That's, you know, basic pattern throughout biology, right? Often you have the most of something at an intermediate level, not necessarily super high and not super low. It is impacted by nutrient availability, by physiology. So if you are not very healthy, you are not going to be accumulating carbon at the same rate as if you were super healthy, right? Or if you're in the beginning part of your growth phase versus the end of your growth phase where you're close to senescing, your primary productivity rate is going to be different. Heterotrophy, I find this the most fascinating part about algal ecology, and that you think of heterotrophy as, okay, phagotrophy, right? I am a cell and I'm going to eat another cell. So in the case of denobrium, it's a chrysophyte. You cannot grow denobrium azenically. It has to be bacteria in the water column with it. And they phagocytize bacterial cells. Serratium, believe it or not, as well as other thecate dinoflagellates also are phagotrophic. Same with Cryptomonas, it's phagotrophic, has a gullet, very protozoan-like, and it can engulf picoplankton. It can also absorb nutrients directly from the water column. Neetsia is a diatom, both paraphytic and planktonic diatom. It can absorb organic compounds from the water column. So it doesn't necessarily just have to be eating something else. They can actually absorb organic compounds, whereas most plants need inorganic compounds, right? They need inorganic NO3, they need inorganic P. These algae can either take advantage of phagocytizing other cells or absorbing organic compounds. This just is super cool. So and I don't have any good photographs or images of this. I've been looking for a video for forever of thecate dinoflagellates engulfing other cells. Serratium can do this with Fragillaria crotonensis. So they essentially exude this organelle called the pallium. It engulfs often chain diatoms. They suck out the contents and jettison the pallium. Pretty cool. And this is a common strategy that's used by some of the thecate dinoflagellates. Releasing from the sediments can actually be pretty important. So we think of it most often with cyanophytes, cyanobacteria. So we have aconites, which are the resting cells of the Nostoclean cyanobacteria. But there's also cysts for things like Serratium, Hormigonia for the Ossoteralis cyanobacteria. So Hormigonia are very short sections, 6 to 8, sometimes up to 10 cells, very dense, and they'll go down to the bottom. The rest of the sheath is gone, or there's a short bit of sheath, and they'll hang there until conditions are good, and then they'll start to grow, or in the case of aconites, they'll germinate. Also, chlorophytes and chrysophytes also produce cysts and oocysts that persist in the sediments. I would say I've looked at core samples. I do aconite analysis in core samples. Aconites survive the best. I do see occasional dinoflagellate cysts, not very often. The other thing I see that are not live are chrysophyte cysts, like Malamonas cysts and cyanophyte cysts. I rarely see them live beyond a couple 3 decades, whereas I have seen what appear to be fully fluorescing viable aconites from Dolichospermum and Phanerozomenon, hundreds of years old. It's very cool. And then or they get entrained from this flocculent sediment when there's turbulence or turnover. So they're either growing and then progressing up through the water column because they're getting buoyant, or they're getting washed up into the water column. Or your ability to maintain position in the water column. So this is Batriococcus up here. It produces its storage product as oils, and it uses those oils to be more buoyant in the water column. At one point, they tried to use them for biofuels. They're not very amenable for biofuels. Takes a few too many steps, but there's a lot of oil there, and that helps them stay buoyant in the water column. Something like Gliotrichia, see this refractive part, those are all the gas vesicles that are in the terminal part of the filaments. Not only that, but by having this kind of round conformation, it makes it harder surface area wise for it to sink. So you can produce oil, you can get an advantageous length to width ratio, you can produce gas vesicles and aerotopes. So there's, or you can be modal, right? Colonization can either be natural, water spray, aerosols, floods, stream flow from upstream, waterfowl, duck feet, right? They actually have a lot of water that's entrained up in those little feathers, and they can carry things from system to system. Amazingly, there's a lot of stuff in spray and water aerosols up in the air. I'm always amazed when you do long-term collecting, almost like a spat sampler in the water. There's quite a bit of algal cells that are hanging up in the water column that get moved around during large storms. Or it's anthropogenic, right? Ship ballast water, that's the one we all think about. Canals, Welland Canal, should I say more? Or something like live wells and engine manifolds. So jet skis, they encase water in the manifold that doesn't empty when they take the jet ski out of the water. They move that from system to system. It's easier to clean the hull of a boat than it is to clean the manifold of a jet ski. So those are big issues. And then of course, folks who will dump their live wells when they're done fishing for the day. When we talk about loss processes, so things like, sometimes you just die. Physiological mortality, it's inevitable, and it's highly variable. So often Either it's cued by change in environmental circumstances, it gets cold, it gets cloudy, there's a lot of rain, the stratification starts to break down and you get moved out of the light. Or it's something like part of the population starts to senesce for whatever reason, and there's actually chemical cues they share, and then everybody senesces at the same time. Almost like a lemming going over the cliff, right? One goes, they all go. Grazing, really complex interactions with grazing. Sedimentation or burial, so hydraulic washout and scouring. This is especially important in man-made systems and reservoirs. And desiccation. Turns out you can actually use desiccation to preserve things for long-term archival purposes in herbariums. So you can tell when when a population is beginning to senesce. In the case of dinoflagellates, you'll start seeing cysts, empty theca hanging around. Denobrian, you'll see that some of the loricas have monads and many don't. So if you start to see this happening, you know the population's getting ready to senesce for the season. It's not like there wasn't something here and then all of a sudden it will get populated by a monad or by a single cell. The cell actually develops with the monad or with the lorica around it. So it's not like there's a new cell that's going to come and populate that empty lorica. Once it's empty, it's empty. So these are clearly senescing colonies here. Pediastrum and all of the new genera that go with that. Monactinus and Pseudopediastrum, you can start to tell when you start to see empty cells here that the colony is starting to senesce, because it's not like a new cell comes and occupies that empty cell wall structure. It all develops together. Grazing, one of my favorite far sites. Broke my heart when he retired. So it's interesting because grazing can be it affects both quality and quantity of algae, because of the effect of grazers have preferences and they avoid or seek out certain foods. So grazing would be considered top down, whereas you have primary productivity with the nutrients and light and temperature, that's bottom up. Consumption and nutrient regeneration can both be a factor, and it's pretty interesting how this all plays out. So food webs are complicated, right? Whenever you're talking about who is eating who, in order to make this so that we can actually understand what this says, all of our nice little arrows go one way, but in fact, these arrows go multiple ways all over the place, right? Even fish will eat young of the year to some extent. So top down versus bottom up are like growth and death happening all the time. Depends on how big you are. Size does matter. How selective you are. Calanoid copepods are going to be less selective than a cyclopoid copepod. How dense you are. So very dense grazers are going to have more impact than non-dense grazers. What your specific excretion rates are. So think about, filter feeding is a specialization of grazing, right? And so zebra mussels will, and I'll talk about this in just a second, but they excrete at a very specific ratio, and essentially how you graze. So are you generalists like a Daphnia, or in terms of size, rotifers, their corona will just bring whatever can get into their gullet, whereas a cyclopoid copepod that is a little bit more selective about what he goes after. I always, because I have 4 kids, so I always think of cyclopoids as being teenage boys, and the calanoids, or the cyclopoids as being teenage girls, and the calanoids as being teenage boys. Calanoids will go for whatever they can get. Key algae features, so what your growth rate is, do you have resistance to grazing? Do you have resistance either because you're big or because you have structures that make it hard? Is there allopathy going on? So the big question about algal toxins, right? Is there a reason for that? And is that supposed to deter grazing? And your ability to migrate. You know what? If just like snowbirds, if you don't like the current circumstances, leave. So migration is a way of leaving, right? So if you're growing very fast, that's going to help overcome grazing. If you are big, it's going to keep most of you from being grazed, like large Aphanosomnium fascicles. These are grazed Goliatrichia colonies, that haircut that Ken refers to. This is daphnid, actually, grazing activity. For daphnids, if they're too complicated, like Aphanosomnium, like big Dolichospermum colonies, it clogs their gullet and they have to try to use their post-abdominal claw to clear that. That causes problems for them. They are not effective grazers in the presence of Microcystis, Vanosomn, and things like that. But they'll eat other things, and they'll eat as much of the other things as they can get into their gullets. If you have big spines, something like Rhizolenia or Acanthoceras, Urosylenia have big long spines, makes it pretty hard to get you in their gullet. Something like Chrysosphaerella is a chrysophyte, colonial chrysophyte. Each cell has 2 waving spines. You know, you get to a certain size, those just aren't edible. You're not going to be able to get them in your gullet. But generally, it's considered that anything under 30 microns is edible. But I've seen rotifers spin up Closteriopsis or Monoraphidium, almost like spaghetti, and get it into their gullet that way. That far exceeded that 30 micron limit. It turns out, just like our food, algae have different nutritional values. So the highest nutritional value is actually considered to be a cryptomonad. Cryptomonads are there all the time, but they're often kind of low level, except for very certain pulses in the spring, sometimes in the fall, occasionally over winter, but they're kind of always there. Make really good Daphnia food. Diatoms, because of the oils, are good food as long as they can get them in their gullet. Green algae are the next best, although often they can pass through the gullet completely. And blue greens or cyanophytes are the least nutritional quality. So, and then in response to that, algae can have no response, which means they are not a target. They aren't grazed and they don't benefit from other things being grazed. They increase. Something like Sphaerocystis that actually passes through the gut undeterred, yuck, and makes it out better on the back end, so to say. Decreased, you are totally susceptible to grazing. And then a unimodal response, which means down here, there's other food the zooplankton prefer. And so they leave you alone until they use all that food, think deer and woody browse. So they use all that food and then they switch over to you and then you have a negative response to grazing. So it's complicated. Microcystis has been associated with blooms in Lake Erie with the onset of the invasion by zebra mussels and then quagga mussels about 15, 20 years later. So it turns out zebra and quagga mussels don't like Microcystis, so they actively reject it, and they eat everything else. They eat all the other algae that are present so that they take all the competitors for Microcystis away, and they excrete at a nutrient ratio that's perfect for Microcystis. So pretty commonly when you see big populations of zebra or quagga mussels, you might have a short-term few year clearing phase, and then all of a sudden you'll start to see horrific Microcystis blooms. Is not just happening in Lake Erie, it's happening in other lakes as well. Sedimentation burial has a lot to do with stratification, turbulence, sediment load. If you have a reservoir, you got a much higher sediment load than an oligotrophic system. Less healthy cells sink faster. So if you start to see a lot of epiphytes like choanoflagellates and chytrids on diatoms, that tells you, okay, they're infected, they're going to go down pretty fast. How modal they are, they're less likely to be buried if they have either a rafe, which allows them to climb back up to the light, or they have flagella like chrysophytes, cryptophytes, dinoflagellates, things like that. They're going to be able to migrate up and keep up in the water column to some extent. Sedimentation will not be as important for those taxa. Hydraulic washout and scour, so that's very much an effect in large rivers and reservoirs, and that has totally to do with the flow, rainfall morphology, and the velocity of the water. And again, whether these mats the flow is high enough and they're at such a growth place in their growth phase that they will detach from the bottom and get washed up in the top. It's not unusual actually to see things like Didymosphenia whole, like all the sheath come downstream in a big flooding event, and just how old the mat will be. Desiccation, so you know, it acts as a loss process technically, but as Ken was saying, the cyanobacteria figured out, well, you know, just dry me out, I'll hang maybe for a few decades or a few hundred years, and then, you know, things get better again, and add some water and voila, I'm going to regenerate and grow just as well as I did before. It's pretty amazing, actually. So it has a lot. It's a great way to, if you can survive being taken out of the water column by one of these activities like an earthquake or a volcano, subsidence of floodwaters, and then, wait for it to get wet again and come back again, ephemeral streams and rivers especially. So let's talk a few minutes about cyanobacteria and the toxins that they produce. Let's check my time here. Okay. So we have several categories of toxins. There's way more than this. I know that probably doesn't make you happy, but we're going to talk about the main ones and about the most common toxins. So we have hepatotoxins, that's microcystins, nodularian cylindrospermopsin. These are cyclic peptides, and that has some pretty nasty implications for water treatment and for how you deal with the water once you realize that stuff is there. And neurotoxins, they cause a paralysis. Usually it causes respiratory failure. This usually ends up that you lyse the cells in the intestines and you bleed to death. It's a nasty way to go. Dermatoxins, which look like they might have a little bit more effect than just being rashy on your skin, but that's what a dermatoxin means. Usually happens pretty fast, hours to days. Then BMAA, and this is still, we're still not sure about BMAA and DABA. The interesting thing is that the methods that we use to detect these toxins are not near as well developed for these as they are for these guys, and there's a lot of chatter in the literature about how much of a problem or not a problem BMA is. And it has been anecdotally linked to cells for ALS and Parkinson's and Alzheimer's, as has microcystis actually. Cyanopeptides beyond microcystins and abinopeptins. This is interesting because you can actually get stick tests for this now. They're starting to develop methods for these, but they don't appear, they're not as produced reliably as some of these other toxins are. And so you don't see a lot of reference to them in the literature as being ones that are a problem, but we also don't look for them as much. When you think about it, any toxin can act like a neurotoxin if it's a high enough concentration. So if you have microcystin that is up in the, 60,000 to 100,000 micrograms per liter, which can happen, it's going to act like a neurotoxin. It can be really concentrated. Give you an idea of how toxic these things are. When you look at LD 50/50, a lethal dose for 50% of the population, saxitoxin, not too bad, right? Ricin, cobratoxin, most of the algal toxins are actually more toxic than cobratoxin. Strychnine, curare, actually it takes quite a bit of those. It takes a lot less of algal toxin. So nodularin, something you wouldn't necessarily have on your radar unless you're in a brackish water area. Most of the data is based on ingestion or direct dosing. We don't have a lot of good information about inhalation other than to know, yes, these toxins along with algal cells do get brought into the air by turbulence, and inhalation is a problem, especially if you have a respiratory issue. So we know that they cause problems. We just don't have a lot of good data about how important that information is. So when you think about measuring toxins, you have a couple choices. You can directly measure the toxins, or you can measure the presence of toxin-producing genes, and that's assuming that you already know that you have a potential toxin producer there, which you really need to know for both of these methods because you'll spin your wheels a lot if you don't. So your choices on analytical methods are mostly ELISA, LCMS, and LCMSMS, and there's also GCMSMS. This is wet chem essentially, and probably the easiest one to do is ELISA, and LCMS and LCMSMS are the most analytical methods, and they're going to give you, if you get a screen for ELISA and you need to have a really good idea, okay, how high really is this, and you're going to move to one of these other methods. The most expensive one is LCMSMS. These pieces of equipment can cost anywhere $50,000 to $300,000. ELISA, you can get started for, by the time you get everything you need, probably $2,000 to $3,000, so much cheaper overall. In my lab, we only do screening tests. We just use strips for anatoxin A and microcystin. So, and we also have cylindrospermopsin strips as well. We made the decision a long time ago to not measure algal toxins. My expertise is biology and taxonomy, not chemistry. I could do it, but it's expensive. and I am busy 100% of the time doing taxonomy. So I'll let somebody who loves to do chemistry do the chemistry, and I'll stick with biology. ELISA kits are available for microcystins, nodularians, saxitoxin, antoxin A, cylindrospermopsin. So the thing you need to know though is that some of these are cogener specific and non-cogener specific. And right now, I believe that there's only standards for 10 of the microcystin cogeners. I believe it's something like that. But there's like 250 cogeners for microcystin, and that number goes up every single time I do this lecture. So every single time I hear Barry talk about it, that number goes up. So it's complicated, and it's based on, for microcystin and nodularian, on the presence of an attostructure within the molecule, but only about 85% have that. So it's complicated. And anytime you go to analyze any of those by ELISA, you know that you're essentially estimating. You know, LCM SMS, that's using, you know, pretty complicated equipment that, you know, is pretty pricey, you know, on the order of $150,000 to $200,000. You have to have an analytical chemist, a biochemist, run this kind of instrumentation, and it takes a lot to maintain this kind of instrumentation. So I send it out to a lab, and you're essentially looking at something, you know, being held on a column, and then being released based on how hydrophobic or hydrophilic it is, and how much affinity it has for the column, if you're using a column like GCMS, or how heavy it is. So, you know, and you have to make sure that this peak is actually what you think it is, and that's why you always need to have standards. EPA has guidelines. I kind of feel like Pirates of the Caribbean. You know, it's really more of a guideline, which is a little frustrating. I wish they had actual regulatory language when they released this, but they didn't. They're advisories. But what they say is essentially for drinking water, they have microcystin and cylindrospermopsin. They have stratified it by school age, which I understand the biology behind this, but no family, unless you have one child, are you going to be able to stratify by school age? So I pretty much ignore these higher ones, and I always go with the lower ones because I have grandkids now. So I have little guys in my household, and we're not making formula, but my 5-year-old grandson is drinking water out of the tap. And yes, we're filtering that through the fridge, but there's still a possibility that there could be microcystin carrying into the distribution system. So. And then, so you always want to go with, I prefer to go with the more conservative 0.3 micrograms per liter and 0.7 micrograms per liter for cylindrospermopsin. Don't often see high cylindrospermopsin hits. Microcystin is definitely a hit or miss, but about 85% of the time it's producing a toxin. It just depends on whether you can detect or not. For recreational guidelines, this was interesting. They originally had this set at 6 or 4, which was more restricted than Ohio. Ohio is 6 micrograms per liter for microcystin. Ultimately, by the time this came out, by the time they actually released the guideline, it was 8. I don't disagree with that, but I'm not sure I would be swimming in 8 micrograms per liter of microcystin. For cylindrospermopsin, it's higher, it's less toxic, and that's 15 micrograms. They did not stratify that for age group. Realize, though, that little guys are behaviorally at risk. So they, just like dogs, they're in the water, their hands and mouth, they're smaller, so their small body size makes them more susceptible to toxin issues. You need to be careful. If you see a scum, you shouldn't be anywhere near it. You shouldn't be in it, your kids shouldn't be in it, your animals shouldn't be in it. So, you know, the axiom is when in doubt, stay out. And my when in doubt gets wider every year. So the other option for us is to actually look for the genes, not necessarily for the algae themselves. If you're trying to figure out risk and you're trying to figure out, okay, do we or don't we have potential toxin producers there, and you don't have access to a phycologist, then you can look for the toxin producing genes. The problem with that is that you don't always know if those genes are turned on. So, and that's, the epigenetics of what's triggering that particular thing to either turn the gene on or not. Now, they know that there's an association with nitrogen. They know that there is some allelopathic stuff going on, but in terms of being able to truly predict, it's not really possible. And the problem is it's not fast. So they do have really good PCR tests now for Primesium, which is the golden blossom, golden bloom that Ken was talking about in the Southwest. And that's because it tends to bloom with Eunice specifically. It's the only thing there, up to several hundred cells per ml. The problem is with algae is with the cyanobacteria is that they don't always conserve their number of genes in the cell. So it's not one-to-one. And it takes time and it takes pretty good equipment. And I don't necessarily, I think it's another tool in the toolbox, and I think it can be helpful. It's just that I would rather, if I'm going to assess risk, I would rather have a good count and have a good ID and be able to go from that. To me, genetics informs what you're doing, but we're not at a place yet where it can be applied quickly and evenly throughout the environment. I don't think it's a great thing to dissociate understanding natural history of the organism you're dealing with from the genetics of what's there. So PCR is another genetic technique, and it involves quite a few steps, and you have to have the right amount of material, and you've got to have sequences to be able to actually do the analysis. It takes time. Now, like I said, they've done better with single taxa than they have with consortiums. So when you have something that you only have to look for one thing, and it's a relatively simple, relatively simple community, detecting like Primesium actually works pretty well. And they've done the same with Microcystis. They're having pretty good luck with Microcystis in some cases, but that's usually in places where they know Microcystis already is. So we don't know exactly what the environmental triggers are for toxin production. It is staggering the number of new toxins that we keep finding. I can no longer fit my nice little, if I have my full ecology lecture where I talk about all the toxins, they're not all the toxins anymore. They're not even close. I'm now into multiple slides actually. It's probably pretty likely that all cyanobacteria can produce some kind of toxin. Remember, it's just a biochemical, it's just a chemical that we react adversely to, right? So chances are they probably are produce toxins to some extent, or can to some extent, or used to be able to some extent. There's a lot of variability among fish and invertebrates, their impact on fish and invertebrates, and vertebrates as well. There's differences in bioaccumulation, there's differences in deprivation rates. So it might clear a mussel in 10 weeks and clear a snail in 2 weeks, or a fish in 3 weeks, a bass in 5 weeks. We don't have a good handle on that yet. We know that they bioaccumulate, and sometimes they biomagnify depending on if there's other things eating those things, sometimes not. We have a really poor understanding of this, acute versus chronic. So, acute means you drop dead. That's pretty clear reaction, right? Toxins are super high and the organism was super sensitive. We don't have a great handle on long-term effects from low-level exposures. So those anecdotal cells of ALS and Parkinson's and MS, we don't have a good idea of tumor production. We know it takes decades sometimes, but not sure exactly how long. And of course, your physiological, just like with growth and death and growth and loss, your physiological state has a lot to do with how you react to that. So we just don't have a really good handle on chronic exposure and what those effects are going to be like, and the actual degree of impact on humans. If you were in other countries, if you were in China, Australia, South Africa, Brazil, you would be talking about human death. We rarely talk about human death in the US. And unfortunately, it's not because our genetics are any different, right? As organisms, we are the same, Homo sapiens sapien, and we react to toxins the same way. The problem is associating exposure with the result, and the epidemiology of it is complicated. Are more algae producing toxins, or are we just now detecting it better? It's probably both, in my opinion. And the ability to produce toxins doesn't necessarily equate to having lots and lots of toxin out there at hazardous levels. So when they did the National Lake Assessment, the NLA, in both 2007 and 2012, they found that there were lots and lots of producers out there. They often were not very high. So lots and lots of systems at or below 1 microgram per liter, right above detection, right at detection, with a few that were super high. So the detector, you know, they're out there and they're producing. They're just not necessarily producing a lot. And what causes them to go over that threshold to hit that tipping point, we don't have a really good handle on. So, and that's Ken's picture, by the way.
All right, thank you, Anne.
Yeah.
We are a couple minutes over, but we can
Sorry.
spill into the live session if we want to. So we do have one, while we're switching everything over to Ken's presentation, we do have one quick question from Lou Feeney. Do toxin test strips give enough info for lake users, and can you recommend test strips?
I can tell you what I use. I use Abraxas strips. They are nice screening tests. They're not an end all be all. So, you know, I'm going to use multiple indicators of high biomass. So I'm gonna use a test strip when there's a fair bit of algae there. They're not great at low levels, even though the tests go 1 to 10, recreational tests go 1 to 10 microgram, drinking water goes 1 to 5. It's still a rough indicator of worry, not worry, contact somebody, don't contact somebody. It's a nice tool for being on a lake association or a quick decision whether to send off a sample, but it is a very rough indicator.
Great, thank you. All right, one more lecture to go before we get into bonus time. So Ken is going to, and I bet a lot of people are waiting for this one. Ken's going to talk about control. So probably
the heck
what
with
everybody's
figuring
here
out.
for.
Yeah, heck with figuring out what they are, how pretty they are, what toxins. I just want to know how to kill them. It's, just a comment on the toxicity issue, particularly for those of you working in New England, because I run across this a lot recently and it bothers me. You should be toxin aware, not toxin terrified, and you should treat your clients and colleagues the same way. I've run into too many groups that will tell someone else, whether it be within an association or a consultant working for them or a state agency, whatever, we have found toxic cyanobacteria in your lake. And they say, whoa, 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 back up. You did an analysis of the toxins. Well, no, that was too expensive. Well, how do you know they're toxic? Well, they were cyanobacteria. Well, it's probably true that they could produce toxin, but how do you know it was a toxin of some level of concern? Well, we don't, but this is okay. We're going to post the lake anyway. Better safe than sorry. And it's hard to argue with that logic. However, there is a huge difference between a pea soup green lake in its entirety and that tiny little fringe of blue greens that blows up when the lake turns over in the fall. Very, very different. Now, Ann was right. You stay out of the scum. If you can, when in doubt, stay out. Don't let your pets lap it up because that could kill a dog. But you don't know that it's toxic, and scaring everybody that's toxic is just not good science. I hate to sound like the mayor from Jaws. Oh, it's okay. You can go in the water, but be aware that you could really be hurting property values, people's psyche in terms of what's safe in their lakes. If you're out on Cape Cod, people are terrified to go in the ocean anymore because of sharks. So they're all going to the lakes. And now we've got blue-green algae showing up and they're being told they can't go there either. It's a little bit scary how many people use scare tactics. Again, it seems a societal thing. You can see it in our political arena as well. Be cautious and be accurate in your statements about what's there and whether or not you know it's toxic. Because the reality is, when it's all said and done, even pea soup green with cyanobacteria only has about a 20% chance of having toxins at a level of concern. Now, 1 in 5 is still good enough odds that I don't think I'd want to bet against it, but it's not guaranteed that there's a problem. And we need to be doing the toxin testing to know. So whether you're using strips or a reader or getting the full analysis done, ELISAs, whatever, make the effort to find out whether there's really toxin present. I have been consistently surprised at how little toxin has been in some of the bad situations I've seen in the last few years. Okay. We're now going to talk about.
Ken, it's Chris. Real quick, can you make
Yep.
sure we can't see your slides right now? So
You can't see them?
no. Do you have them on a different screen?
All right. How's that?
That's it. You got it.
Okay, that's the answer.
Thank you.
That's right. Only I need to see them. Okay, the ecological basis for algal control. I'm going to scroll through very quickly because Ann has really covered that. There was some. We were both trying to figure out how to make sure we work this in. It really is the same routine. All right, if I can get it to move. All right, the same problem I had when I started last time. I couldn't, there we go. Okay, growth processes, the various ways that algae grow, loss processes, the various way we lose them. Algal blooms or problem algal situations arise when the growth processes overwhelm the loss processes. And everything we do to control them is either trying to reduce growth or increase loss. It really is that simple. Of course, there's a lot of detail to how we go about it. The key factors, you need to know what you've got for phosphorus and nitrogen. You need to know the abundance and size structure of zooplankton because they're the second most important thing in how much algae will be present. Temperature and oxygen depth profiles are extremely useful. pH matters to the types of algae. Dissolved organic matter makes a big difference. Color may be a sufficient measure. And the algae in the upper waters and the metalimnion, just sampling at the surface won't tell us everything we need to know. Okay, there are three major modes, really four modes of bloom formation. One is it washed in from upstream. Okay, we're not controlling that, but that's an option. Organic growth in the upper water column, a little bit is present. There's enough nutrients, it grows into a bloom. Turns out not to be the most common thing. More often than not, it's either forming at a mid-depth if you have stratification and then moving up, or it's forming near the bottom in shallow lakes or at a mid-depth in deep lakes and popping up. So these blooms tend to show up kind of quickly. All right, so now we're into the methods of control. Simple process. Don't lose your head, get your ducks in a row, don't bite off more than you can chew. First and foremost is understanding the watershed and managing it. I get dumped on a lot for making it sound like you shouldn't bother with watershed management. That's not what I'm saying at all. As will become apparent, I'm saying that you're unlikely to solve most of these problems with watershed management alone, but it is job number one. We have natural background levels for phosphorus that are higher than some areas, in some other areas than others. We have land uses that make a huge difference. Anybody know where this is? Of course, you can't respond to me directly. That's actually Mexico City. There's their drinking water reservoir. There's part of the city and a golf course all draining into their drinking water reservoir. Yeah, great idea. But sometimes you don't have a choice. Managing the watershed is not an easy task. It got made to sound easy, or we can do it with just enough effort by a lot of agencies, including the EPA throughout the 80s and the 90s. And I'm here to tell you, it's just not true. Extremely difficult to take a damaged watershed, something that's more than, say, 30% developed for agriculture or urbanization, and make the water quality that comes off of it acceptable. We have source controls to work with, and that's a great way to go if you can, but you're not going to do that in Mexico City or next door to me in Springfield, Massachusetts. Those things already exist. Pollutant trapping, detention, infiltration, uptake and treatment, maintenance of facilities, all these things are important. They're all good to do. We even have regulations that call for them in many areas. The bottom line is once your watershed is developed to a certain point, the odds of you being able to make the water quality acceptable for the lake into which it goes is really reduced. Just the quick rundown, developed land increases phosphorus loading by an order of magnitude, usually greater than tenfold. Common best management practices reduce phosphorus by maybe 50%. There's a few things that'll get better, but not routinely, and you have to apply them all over. Agricultural impacts are at least as great as urban impacts. I'm not here to beat up on farmers. I like to eat fresh produce too. I enjoy meat, sorry. But, the bottom line though is we run a risk when we have the situation we do with our agricultural industry and our urbanization of impacting our water quality. And it is not a simple matter of, we just have to put some VMPs in place. Again, with more than about 20%, yeah, you could stretch it to 30% of that watershed in urban or agricultural uses, the receiving lake is going to be impacted. There's just nothing that works well enough. The bottom line is we cannot, in this lower picture, we cannot make land that looks like the part on the right behave as though it's the land on the left. There is nothing that will do that. I love this. This is actually a study done by Kathy DeBusk, who's got married and changed her name, so I've lost track of her. She's in North Carolina. We had her come to the NEAPMS conference back in, I think, 05. She's like 15 years old. This is what it took to reach, or to attempt to reach, water quality standards for phosphorus by reducing the loads through watershed management. This is the cost per square mile, and this is the percent load reduction. They didn't even reach 30%, and they were leveling off after about $3 million per square mile being spent. How many watersheds do you know where they can spend even $3 million per square mile to minimize impacts? Not many. And yet, you know, that's only getting you a 30% reduction. That may not be good enough. So there are very real limitations to what we can do with watershed management. Yes, it's still job one. Yes, you need to understand what's going on in the watershed and how much of the problem in the lake is caused by it. But in the end, you're going to need in-lake management to be able to do anything in the vast majority of cases. I have 2 or 3 projects in my 43 years of consulting where I can say we fixed the lake by fixing the watershed, and I have been involved in thousands. It almost always involves an in-lake solution, which is why organizations like NEAPMS exist. We know our businesses, it's not like we've created this. This is very real. They need our services. There's a lot of data you need to have something on it to understand the system, to properly manage it through in-lake controls. It is not a matter of going out and tossing a chemical in or dredging or whatever else. There's all these things matter. You may not have to know every last detail about it, but you've got to know something about it to have some idea of the predictability of your results. Okay, so the run through of techniques. This goes pretty quick. It'll probably take me at least 2 breaths. Yeah, in-lake detention. It's basically relocating a lake or a watershed function into the lake. You can do it. This happens to be a small lake in Arlington, Massachusetts, where we took a piece of the lake and created a detention area. And the stormwater went in there and it cleaned it up. We were getting 200 micrograms per liter of phosphorus there and 20 out in the lake. This looks really big compared to the lake. It's not. It's just the perspective of it. Dredging. Dredging is pretty drastic, you know, when you do this in a lake, but I've seen it do wonders for algal mats. And of course, you're removing the nutrient reserves, you're removing the seed bank, which includes algal resting stages. You can get very good control of algal mats by this in most cases. It's very expensive, very difficult to permit in many places. So it's not like this is something that you're going to do routinely. Where it's been done, it's almost never done just for algae control. If you've got enough depth that you don't need to reclaim the depth, it's way too expensive for what you get for it. Harvesting, another issue. The use of a harvester, a typical, you know, back up one, weed macrophyte harvester is not going to work very well for algae. The mats are going to stick to it and roll over and go right back in. If they're really big and bulky, you might, but they do make harvesters with fine mats or even like a tight conveyor belt where you can pick them up. I know the Chicago Botanical Garden uses one. If you're in a situation where you can't control the nutrients, but you want to pick up the mats, you could do that. They run around early in the morning before they open up and make sure the water, you know, places are clean. It's a little hokey. It's not really getting at the real problem. It's not something you see done a lot, but yes, it can be done. Dilution. Again, dilution and flushing are interesting things. Dilution is just putting any amount of water in to keep the thing flushing so that the algae don't build up. Excuse me, that's flushing. Dilution is putting in water of a lower nutrient concentration so that you get an average that's lower than you had before. The catch is you're still putting in more nutrients than you would have had because it's a flow times a concentration. So you're loading it up, but you're loading it up in a way that reduces the average concentration. That's pretty tricky, and it doesn't happen very often. Flushing is more often used where you've got a fairly small water body and a big source of water. The example I'm showing you here is actually a reservoir on the Columbia River, and they're diverting the massive flows of the Columbia River through it. Moses Lake, and it did work. I've seen it work on a smaller scale as well. But most of our lakes, we don't have that kind of quantity of water to mess with. To make flushing work well, you have to turn the water level over. You've got to completely replace the water in the lake at least every 3 weeks and preferably every 2 weeks. That is not something you're going to see in New England very often. Selective withdrawal. This is an interesting technique, and we've had some pretty good successes with it in certain areas. You're basically pulling the water out of the deep zone of the lake and up and over and out the dam, either by a siphon, a pump, whatever you want to set it up as, so that the water down here is constantly being replaced. If you do it well, this never becomes poor quality water. But again, it means you have to pull the water out at a rate that may cause the lake to be drawn down or may cause it to destratify. And there's a lot of places where that isn't going to work out very well. So again, a run of the river impoundment, this could work. A basic kettle hole sitting somewhere, nah, not going to happen. Dyes, again, dyes get permitted like herbicides, which is just not right. They're not killing anything. They're just decreasing light penetration in the water. They create an interesting color. It's fairly artificial. Looks okay in a golf course pond or out by Rockefeller Center in New York. It's not the kind of thing you do in a recreational lake. I once was involved putting a small pond in for a wealthy family in Western Connecticut, and the woman said, well, the dye in is just spectacular in terms of the appearance. What will happen if I swim in? I said, oh, you'll turn blue. And she was all packed. No, not really. It's again, it's kind of hokey. It's kind of artificial, and it's still going to allow some things to grow. It's going to cause the lake to stratify as shallow as 5 feet. You may get anoxia in relatively shallow water. So it's not perfect by any means, but it's an option. Sonication, this is one that's gotten a lot of bad and good press and mixed reactions. It's pretty clever. You're basically using sound waves to disrupt the cells. We use this sort of stuff in the lab. It's used medically. I mean, it works. Whether or not you can make it work out in the lake at this expansive range where you need it is up for grabs. Most of the product literature shows you something like the bottom here. It looked like that. We made it look like this. Well, that's not what you're trying to do. You're trying to keep it looking like this and never let it get to that. Some of them, the simpler ones, are just, unidirectional thing that sits there and it shoots the sound waves out. The fanciest ones actually have them pointing in all 4 directions and will have a sensor with it that uses fluorescence to figure out what kinds of algae are there and then sets the wavelength of the sound to be most damaging to that type of algae. That's pretty darn clever and sophisticated, but what happens when it changes a little later? How fast does this react? If you've got a real mixed bag of algae, are you going to favor some over others? Not all algae are susceptible to this either, so it gets a little bit complicated. I will say I've seen it work pretty well in small controlled circumstances. I've never seen it really do much on a big lake application. Here's an example though from one of my own projects. This is where we didn't have the sonication pointing. This is where we did. It sure did keep that surface clean and did not let the algae build up. Fundamental problem here is, though, if you are successful with one of these things and it keeps the algae down, rooted plants may grow and they're going to block the line of sight and block the sound waves, and now you may go back to where you are. So it may not be a very complete solution, but certainly if you're in the intake area of a water supply or something where you're getting algae accumulating along the rocks and it's unaesthetic, this does have potential. Algicides. Okay, we could go on a long time here. There's many people on this call that know more about algicides than me, so I'm not going to go into this in great detail. Pretty much the 2 basic things we use are copper-based or peroxide-based algicides. There's some use of endothol and flumioxazin and other things, some of which have been pretty darn effective, particularly flumioxazin on filamentous greens, which are hard to kill by any other means. Herbicides being, algicides being herbicides can get a bad name in certain circles. You're putting in a chemical, you're killing things. Yeah, we are. But would you rather have a little copper in your system or a whole bunch of algal toxins? I mean, it's not it's an either or situation. Copper gives you rapid results. Dave Hammond was on at the beginning showing some examples of what they've done with Eartec, which is copper based. You can get pretty darn fast results. And if you are a water supplier who's got to provide this water to people, whether you've got a bloom or not, you need this kind of thing in your back pocket. It'll lyse cells, not always, but often releasing the contents into the water. You may kill them and get them to settle if it doesn't just right. The formulation affects the time in the solution and the effectiveness. So it's not like all copper is created equal. There's a lot of different formulations. It's really important to think through what you're trying to do, what the goals are and what the best approach is going to be. It's definitely something where getting professional involvement is worthwhile. It's not a matter of just throwing some copper in the water. Yeah, copper is possibly toxic to other aquatic organisms. At the levels we tend to use it in the Northeast, there's not a whole lot else that's going to be grossly damaged. I've not seen fish kills result. Massachusetts Division of Fisheries and Wildlife did a survey for 20 years and couldn't find a single case where algicides had caused fish kills. In fact, they found very few where any kind of herbicide use caused it. But it is possible, and the worst effects I've seen are actually on zooplankton, because if you let the thing get a little too high, the zooplankton can be impacted, but they'll come back. Long-term buildup in sediment's a concern, but there's really been no paper out there that says it's really bad. It could be a problem if you then later go to dredge, because now you've got a high metals concentration. It may make disposal more expensive, but it's has not been proven to be a big problem. Folks in New York have looked at that pretty extensively and reported on it at NEAPMS in the past. We do have some species that are resistant, and blue-greens and green mats are among them, certain ones, so it may not be effective all around. You can put it at the surface. You can also inject it deeper. I find that where we have problems with the blue-greens that grow at the bottom and then pop up, if you treat a donut in the lake with a chilled solution that sits on the bottom, you can often prevent that from happening. We've had it work very well in several reservoirs we've managed. Again, it's less effective at colder temperatures, but it does still work. Peroxide, the other major one, can also lay cells, tends to be more effective on thin-walled species, less so on diatoms and greens, so you tend to get the blue-greens taken out better. It degrades to non-toxic components, so that part's fairly favorable. Also applied to the surface, but with granular things, you can drop it in and get to a greater depth with slower release. You don't get any unwanted contaminants, but it tends to be more expensive than copper. You got to work on the factors of dose and frequency to see if it works out. I know there's folks in Connecticut in a water supply system that are using peroxide pretty extensively, and they feel they've gotten the cost down to being on par with copper because they're able to do it less often. So there's some playing around to be done. Both copper-based and peroxide-based algicides have merit. There's a catch. They should be used to prevent a bloom, not remove one. If you get called and it's already pea soup, you don't get a lot of choice. But the object is to track when the algae are growing, figure out when the bad stuff is in its exponential increase phase, understand the water chemistry, pick the right form of algicide, and apply it at the right dose, in the right places, at the right time to keep the bloom from ever forming. I've got a number of places where I've done that, and well, I haven't done it. I've recommended it. Actually, it's folks at Solitude have done most of these, and it's been very successful. Phosphorus inactivation. This is one that's near and dear to my heart because I've done a lot of it in the last 20 years and been pretty darn happy with the results. Basically, you're putting in a compound that will bind up phosphorus and make it less available. As we've discussed multiple times, nitrogen is important, but nitrogen is more important to the type of algae, not the quantity of algae. It's the phosphorus that tends to control the amount of algae you're going to have. If you can depress the phosphorus, you can depress the amount of algae in total. Inactivation will put in any one of a number of binders. Iron's the most common natural one, followed maybe by calcium in certain higher pH systems. But what we find is aluminum, which is also a common natural binder, works really well because it's not subject to pH very much once it's reacted. It's not subject to oxygen changes at all. And you can inject it and you can use it very flexibly. You can treat inlets, you can treat the water column, you can treat the sediment. Also lanthanum, which is the main ingredient in Phoslock, and that's a really interesting one. Lanthanum is more specific to phosphorus than aluminum, so it is a more efficient phosphorus binder. You may need more of it. It is not a coagulant, so it won't take particulates out of the watershed, out of the water column, and it tends to be more expensive, but there are reasons why this could be a superior product under certain circumstances. The factors in planning the treatments, okay, got to understand the existing load, particularly the internal versus the external. If your lake is a run-of-the-river impoundment where the water is replaced every couple of weeks, it settles down in the summer and sort of festers, but it's based on what got loaded into it a few weeks or a month before. Treating with a phosphorus binder in the lake is only going to buy you a certain amount of time. It is not going to really fix the problem. It becomes a maintenance technique. And I'm not saying there's anything wrong with that. I think in a lot of urban lakes, that's exactly what we should be doing, acknowledging the maintenance need and covering it. But if you understand that you've got an internal load that's really driving the system, kettle hole lakes, things with relatively small watersheds where the phosphorus is accumulated over many years and is now internally recycling, in a one-shot deal, you can buy 20 years of good conditions. So there's a lot of potential here. You have to understand the sources and the activation needs. You need to know how much available sediment phosphorus there is, particularly the iron-bound and the biogenic portion. The biogenic part is just the organic portion that's readily available, and that's a specific extraction test you do. The bathymetry and hydrology of the system works. Is important to understand. Potential water chemistry alteration. You can't let the pH get out of whack. You have to acknowledge metals levels. You have to understand the oxygen concentration. Any potentially sensitive receptors will matter. Fish, zooplankton, macroinvertebrates can all be directly impacted. There can be toxic results, particularly from aluminum, if you don't balance the pH well. We've had plenty of tests where we've not had an issue. There hasn't been a fish kill from an aluminum treatment in like 20 years. We had a few rainbow trout die, a few more than I think should have, in a treatment last spring. And I still can't quite figure it out because nothing else was dying, but these recently stocked rainbow trout did, and we're not sure that they just weren't very well suited to the lake to begin with, but maybe there was an impact. We're not sure. But again, it was 100 or so, or 90 some out of several thousand that were stocked literally the day before treatment. So, you know, there are sensitive receptors though, and you have to be aware of it. Reptiles, amphibians, waterfowl have really not been much impacted, but if they're laying their eggs or something's going on right at the time, people are going to be concerned. So you have to at least take that into consideration. And then the accumulated residues. If you're doing this on a maintenance basis, you will accumulate a certain amount of inactivation sludge. I've never seen it amount to anything I was concerned about. I've heard it sometimes has been an issue at Universal Studios in Florida. I don't know what it is about their water because Disney doesn't seem to have a problem, but you do have to be aware of it. Okay, lake water column treatment. This is one in Connecticut. As you can see, we had outrageous phosphorus levels building up over the summer, clearly coming from the sediment and mostly from what seemed to be organic decomposition, but it also had a really nasty watershed with a lot of stuff in it. So we started doing these low dose treatments. These are milligrams per liter over this lake. They're not big doses. And boy, it knocked it down, knocked it down, knocked it down. And we're down to almost where we want to be with 2 small doses a year. It's maintenance. If we stop, it'll probably go back to what it was within a year or two. It's not as satisfying as managing it in the watershed, but it works. And we got no prayer of making this watershed do what we want. We can't get cooperation from the state or the town. So this is a case where managing it in the lake has real potential. Tributary injections is one I've been doing for over a decade now in Wellesley. It actually doesn't look like this. This is one of our early tests, but you can't even see it going in now. It's so minor. But basically, we have 2 lines that go out to 2 parts of the inlet, well, 2 different major inlets to the lake, and we dose them just when it rains, just from about Memorial Day to 4th of July. So we clean up all the water getting into the bottom part of the lake by then, and we've had fantastic clarity most of the time. Here you see this is before treatment. You know, clarity was hardly ever 10 feet or 3 meters. Phosphorus was usually above 20 micrograms per liter, which is a problem here. When we first set up the system, it worked really well for 3 years, but we were using alum and aluminate, and it was eating up the equipment. We had a whole bunch of other issues, some of which were human error, and we didn't really do so well for the next 3 years. So we revamped the system. We used polyaluminum chloride, which is really easy to work with. We automated the system so nobody has to go out and turn it on. It comes on its own. I can track it on a cell phone, and you can see the results since 2014. They're just phenomenal. The place is good. The last couple of years haven't been as good because we've been experimenting with how little can we use. And it's still, we're getting through the swim season. Everybody's very happy with the results there, just treating the inlets. They're working in the watershed, but the town only controls 22% of it. So there's limits on what they can do. And then finally, lake sediment treatment, which probably more people here know about than the other things, where you're basically just taking a long-term buildup in the sediment, dosing it in the water column, letting it settle and inactivate that sediment. This is pretty much a tried and proven technology. We do not know everything we need to know. Picking the dose is still a little sketchy. You'd like to go higher than lower, but it costs more. And if you go too high, it interferes with itself and you may get less of a result. The current thinking is if you're more than about a 50 gram per square meter dose, think about splitting it into 2 doses and spread them out over a couple years. Seem to get better results for that. And we're experimenting some with that now. So there's still more to learn. It is not a perfect solution, but where you've had phosphorus buildup from the watershed over many, many years, and the current inputs are really not enough to make a difference in any given year, this sort of thing gives you back excellent conditions quickly and for a fairly long duration. This is Hamlin Pond, kind of the poster child for it. Really poor Secchi disc in the summer for a number of years before the treatment. It had a duck farm on it for 30 years. That was long gone, but the lake was still a mess. Hydrogen sulfide in the bottom, horrible cyanobacteria blooms. Town put a $7 million beach on it and couldn't even use it. They did this treatment in 1995 and the Secchi disc just shot up. You can see it kind of declining over time, and it like all went bad all of a sudden. 2014, well, the very end of 13, there was a bloom in like late September, early October, and then 2014 was bad again. So they treated again in 2015. You can see we're doing great again. So this has worked well, and we got 18 years out of it there. 20 years is about the average you get for a deep lake. Tend to get more like 10 years for a shallow lake. But again, when you divide the cost by what you're getting for it and the resulting conditions, it tends to be worth it. The aluminum longevity is a function of external loading still coming in, release from phosphorus and organic matter that may not have been inactivated when you did the treatment, and upward migration of sediment, iron-bound phosphorus through the zone that you did inactivate. So there's a bunch of things going on at once here. The upward migration is the one that kind of gets you in the end. That's why you get maybe 20 years out of it, because you can't do much better before that gets up there. The other two can make it so that the treatment results aren't as good as you'd hoped or don't last as long as you'd like. So understanding these is pretty important. Okay, lanthanum modified bentonite clay, Phoslock. I don't have a lot of personal experience with this, but I know a lot of people who do, and I like the product overall. It's developed in Australia. It's used more in Europe, some in Canada, a little bit in the US. I know there have been treatments done within the NEAPMS zone. There's no direct pH change. You don't have to buffer it. It's pretty specific to binding free phosphorus. It's pretty stable once it's formed. It doesn't seem to have any toxicity issues. CPRO is the one that markets it on behalf of Phoslock in the US. I like the idea of the clay coating. It's put in with bentonite clay. If you can get enough of that on top of the organic sediment, you can reduce the oxygen demand. Now, I don't know if that really happens or not. It hasn't been well studied, but the potential is there. So it's another way to go about it. Keep in mind, if you have a problem in the water column with actual algae, the aluminum will bind them up and help settle them out. Phoslock isn't a coagulant, though will take out free phosphorus in the water column. It won't take out phosphorus already bound up by algae or other particles. And then it's going to attack free phosphorus down in the sediment as it's released. So the Phoslock might give you a longer term result if you have biogenic phosphorus being released year after year after year. There's still more work needed. It does not have as long a track record as aluminum, but overall it seems to be pretty positive. Lake Lorraine in Washington was an example that was given to me by the CPRO folks. You know, they went from the top picture to the bottom picture. I have heard that it has since gone back. I don't know if it's been retreated. I don't know why that was. Again, none of these are permanent solutions, but look at the watershed. You're not going to make those people go away. You're not going to get the nutrients to disappear. So you have to do something about it in the lake. Okay, selective nutrient addition is one that admittedly scares the hell out of me. There's great literature out there showing that if you manipulate nutrients, going back all the way to 1973, messing with silica, you can prolong diatoms. Increasing the nitrogen, you can shift things to green algae and away from blue greens, but you're still going to have a lot of algae. They use this in fertilization for fish production, and they're not trying to cause blooms. They're just trying to bump up overall productivity so there's more algae, more zooplankton, more fish. Again, there's nothing wrong with high productivity as long as something's eating it at the next level up in each stage. The problem is when you get something that's inedible, like a lot of our big blue greens, and they just take over the system, now you have a very energetically inefficient system and a whole lot of other problems to go with it that may be harmful to the fish. So the biological structure is really important when you're playing around with nutrient ratios. I personally do not have the guts to go tell an association, oh, just add some more of this, it'll get rid of your problem. Pretty much we try to reduce nutrients, not increase them. But it does tell us that there is potential for this, maybe in places where you can't possibly control the amount of phosphorus, a golf course pond, something sitting in the middle of a whole bunch of septic systems. Maybe there are places where playing around with this makes the most sense. Okay, oxidation and circulation could be the subject of a very long lecture by itself. There are many different means of getting oxygen into the water, circulation being one of them. There's a whole lot going on here, and just to make a claim of oxygenation or circulation is not adequate by itself. You have to understand the details of the process. So, oxygenation and circulation, again, all circulation tends to be oxygenation. Not all oxygenation is circulation, but it can work by adding oxygen and facilitating phosphorus binding. It can alter the pH and the related water chemistry in a way that favors preferable algal forms. It can create a suitable zooplankton refuge that will enhance grazing. It can also destroy them, as some recent papers have shown. And then circulation by itself will create turbulence that may neutralize the advantages conveyed by certain buoyancy mechanisms, particularly in the cyanobacteria, which may shift you away from cyanobacteria and into something else. Just be aware that not all cyanobacteria depend upon buoyancy, and you may get other cyanobacteria in place of the ones that you knocked out. And finally, homogenization yields consistent water quality, which may not be what you're looking for in a recreational lake, but water suppliers will take consistency over a slightly better water quality that's more variable every time because they can treat it and deal with it. Okay, non-destratifying oxygenation comes in 4 major ways. The simplest is basically dissolved oxygen injection. If you have a deep enough lake that those oxygen bubbles can be absorbed before they get to the thermocline, so they will not cause destratification, this can work. It's the simplest, easiest way to do it. It's one of the oldest ways, but it was not until we developed better delivery mechanisms and had more oxygen produced and had on-site oxygen production that this became economical and beneficial on a longer-term basis. But it doesn't work if you've got a lake that only has 5 or 10 foot of hypolimnion at the bottom, because you don't have enough run time to get all the oxygen dissolved. So then you need to either bring the water into a chamber and oxygenate it or aerate it, or pull it out of the lake and oxygenate it or aerate it out on shore and put it back. Basically, these hypolimnic aeration chambers were used for many years. You're seeing them less and less frequently now because they're energy inefficient, but they work. You're putting air, you could do it with oxygen, into a system. You're pulling the water in, it's oxygenating over a vertical rise, and then the water goes back to where it came down the outside of the chamber. It's effective. It isn't efficient. If this breaks down, things go anoxic, then you got problems. And of course, your equipment's in the water, which makes it a little harder to maintain. Same thing for downdraft bubble contactors or species cones. They're under pressure because they're deep already, so you get tremendous transfer. You're putting oxygen bubbles in that want to go up. You're pumping water in that wants to go down, and you want to balance it so that all the oxygen gets absorbed. Otherwise, it's got to go up and be wasted to the surface. But these have had some super success stories. The problem is you need this major structure on the bottom. It's got to have a platform that it can sit on so it doesn't settle into the sediment. If you maintain it, you've got to go down there with divers. It's expensive, it's efficient, and it may be effective, but it hasn't been favored in many cases. You can, however, pull the whole thing out of the lake, put all your equipment on shore, pressurize the container like you'd get naturally if it was sitting in deep water, add oxygen to it, and then put the water back in. That is really effective and can work in very shallow hypolimnetic situations, but it's more expensive. Also, if you don't pressurize and oxygenate quickly and get it back, you're going to get an increase in temperature, and now that water is going to want to rise, and there have been some disasters created by that. But this is the side stream supersaturation is a technique that's coming into its own. You're starting to see commercial versions marketed. If you don't have a deep enough lake to let dissolved oxygen work by itself, this may be the next most effective way to do it, and it's easy to control. Side stream supersaturation is one term. It wasn't sexy enough for one group, so we came up with nanobubbles, which you're hearing a lot about lately. It's sort of a version of this. The difference is here it's pressurized, and you're pretty much guaranteed that those oxygen is actually dissolved in the water. Nanobubbles are bubbles in the water that are so tiny that, technically they're so tiny they qualify as dissolved by the definition of dissolved. That gets to be a real sticky issue. But the claim is they're really bubbles. They're not dissolved. And they're so small that the surface charges on them hold them in solution. They do not rise and disappear. They're as likely to go laterally as they are to go vertically. That's all believable. And then you've got to get enough of them into solution to put out the oxygen capacity you need to put them back in the lake. In the cases to date, and there aren't that many out there yet, we're still learning, where they've not had to worry about it being stratified, they're just trying to get more oxygen into the system overall, it's worked fairly well. Where we've had stratification and the object is just to oxygenate the bottom water, we've had less desirable results. The primary claim has been that they're not actually getting the transfer of oxygen into this chamber that they want. And what we're putting out here really isn't as much as they think they're putting out, but what gets out there seems to help. So I've got one on the Cape that we've done now for 2 years. It has not worked out particularly well, but a lot of it's failure of implementation, not failure of the theory behind this. It's just a technique that's not quite ready for prime time yet, and we're going to need to mess with it a whole bunch more to get it there. Okay, the biggest challenge to successful oxygenation is induced oxygen demand created by water movement. If you push water across the sediment with oxygen in it, whether it's being moved by the air or being moved by a water current, you get the oxygen where it's needed, but you also cause more oxygen to be demanded. The best way to think about this is if I put a whole bunch of ping pong balls in a pool, kiddie pool, and I put you in there with the net and say, go collect as many as you can in a minute. You'll get a bunch. But if I was able to stir the pool and have it all moving so they all came to you, you could get a lot more in that minute. So that's what's happening here. So whatever you think you need, when you put the oxygen in, you tend to need more. You basically create an induced oxygen demand. So for oxygen, pure oxygen systems, we usually think you need about twice as much as what you might calculate. For air systems, you might need as much as 5 times as much because it's only 1/5 oxygen. Key factors in oxygenation, you got to get enough oxygen to counter the demand and that induced oxygen demand along with it. You've got to maintain the levels suitable for the target aquatic fauna or high enough that it doesn't let the chemical reactions occur that you're trying to avoid. And that number is about 2 milligrams per liter. For fish, you want something more like 5 milligrams per liter. You've got to have enough phosphorus binder present naturally, iron, calcium, or aluminum, whether you're putting it in or it's naturally there for it to bind up the phosphorus. And you don't want to break stratification if that's part of the goal. Destratifying oxygenation or artificial circulation is most often just done with diffused air. And there's all kinds of ways you can do it. There's a zillion things out there. There's lots of companies, including a number of them that sponsor an exhibit with NEAPMS that know what they're doing with this. It's tried and proven methodology. There's still a certain amount of trial and error that goes with it. There's still some playing around to be done. There's more to be known, but this is stuff we know how to do. If you don't mind having the whole system mixed, we can make this work fairly well. If there's not enough phosphorus binder, there's still an issue. If we're actually causing poor quality water to be mixed into the upper water column, well, there may be another issue. It often doesn't result in lower oxygen, or excuse me, in lower algae than we would like, but we usually do get a shift away from the blue-greens. And if your biological structure is good, then they can eat the other algae. But then you've got to worry about the biological structure part. It doesn't have to be done with diffused air. There are downdraft systems. This is one by Resmix, largely an Australian company, but there's a few in the US and a bunch in other countries where they pull the water down. You're taking oxygenated water from the top and pushing it to the bottom. That's philosophically pretty desirable. The alternative is an updraft system, Solar B being an example, where you're pulling the water from the bottom to the top. That's okay as long as you pull enough water that you keep the water circulated well. If you don't circulate it well enough and the bottom water becomes of poor quality, now you're pulling it up to the top where it can do some real damage. Solar bees claim this long distance circulation where they pull the water up, it goes laterally across the top and then eventually goes back down. Well, that eventually is pretty darn quick. It doesn't go that far. And you tend to get a donut around it. And it's usually a lot smaller than what was being claimed in the literature. So there's an issue there where, yes, you get pumping, but these are usually not put in at a density high enough to actually get the job done. And that's a constant source of aggravation going back and forth between the company and practitioners and so on. But the theory's sound. It's just that it doesn't do everything they say at the level they claim. I want to mention their fountains also are the same idea. You're throwing the water in the air, you're oxygenating, putting it back down, aesthetically pleasing, not very efficient, but it could be effective. Okay, the biggest challenge in successful circulation is maintaining the mixing. Water warms through the summer. You're not getting rid of heat from this, you're just mixing the water. So the water's getting warmer and warmer. It's a simple physical fact that more energy is needed to mix warmer water than colder water. For every temperature degree difference, when the water's warmer, you need more energy to mix it. So this gets harder and harder and harder to do as the summer goes along. Almost no one installs a circulation system with enough power to mix come August or early September. It just doesn't happen. So you just have to hope you get through the summer or it's not too bad too fast, because otherwise you've got to build an oversized system that you don't need most of the time. And that's generally not what happens, but it can work. And the key factors, you got to move enough water to prevent the thermal gradients from dead setting up. That means no more than about a 3 centigrade degree difference from top to bottom. And that's the easiest way to measure if you're successful. General guide, if you're using air, 1.3 cubic feet per minute per acre served. You might need more than that. 2 cubic feet per minute per acre is a good target. If you can, that's a little more oversized. If you're doing it by pumping, you need to move 20% of the target volume per day. So about every 5 days, you've moved it all. Could need as much as 100% per day. It's a question of how warm it is and what the conditions are. Almost none of this is going to overcome high nutrients if you've got no control of the nutrients, though. You may get different types of algae, you'll still get a bunch of algae. You have to balance the delivery of the oxygen near the bottom of the lake with avoiding sediment resuspension, and that's a difficult challenge with these systems. And then you've got to move the surface water to a depth that's 3 times the Secchi depth if you want to actually lower biomass so that they're spending more time in true darkness. In a shallow lake system, you really don't have much of a way to do that. So you really have to expect you're going to shift the types of algae, and then it's a matter of biological structure, whether or not they can get eaten and whether or not you have a bloom or everything works out. Okay, just a few other ones. Barley straw, I mean, it's basically an unlicensed or unregistered herbicide, but it can work. The barley straw decays into material that or compounds that seem to have allelopathic capability and particularly are damaging to blue-green algae, so there's some benefit to that. The guy in the pictures here is Steve McComas, who said it best. He said it works sometimes, but I'm not really sure why. It's not as easily controlled as a lot of other algicides, and frankly, a licensed herbicide applicator can't use it because it isn't a licensed project or product. Viral controls are really popular twice, once in the 70s, once in the early 2000s, at least on the research front. We've never really developed any commercial products that work. And I think the real reason comes down to this. You have your untreated, it's just horribly green or dark. Your treated looks like that. Are you really going to be any happier with your lake looking like that? It's hard to get complete control with biological means. Okay, bacterial additives, again, could be the subject of a whole course, which I'm not really qualified to teach. There's so much to be known here, and a lot of it isn't known because there's virtually no peer-reviewed papers on how these things work. Most of it's proprietary. You get a simplistic claim that they allow bacteria to outcompete algae. That's not really that likely. When you get shown an image of here's what it was before and here's what it is 5 or 7 days later and it's gone clear, there are no bacteria that can outcompete algae in 5 to 7 days and then disappear. It doesn't happen that way. You can get a potential organic sediment reduction. They often pair it with circulation or oxygenation because it does work better when the oxygen's high. We get variable results. We have inadequate scientific documentation, but I will tell you there's successes out there. I've seen it work very well in many places. I myself used muck away. I didn't get less muck, but I got way less dense muck. It definitely ate up the organic component. It's a potentially effective but not necessarily reliable approach. And by reliable, I mean that in the engineering sense, that can you guarantee that you're going to get what you expect? And the answer is no, because usually you're putting in 1 or 2 products. You're just buying something that's supposed to work, getting it applied, and there's not a whole lot of monitoring that goes with it. There's not a whole lot of testing. Most of this stuff was developed in the wastewater and the sludge handling industry. And they're doing a lot of testing and a lot of manipulating, a lot of tweaking to get the best results. We just throw the product in and expect it to help us. That's really not that effective a way to go. The best explanation I've heard of this is from Pat Sims-Geiger's Diversified Waterscapes. He's been to NEAPMS before. He's from California. 4-step process. Use an algicide to kill the algae. Secondly, use enzymes to break down the long-chain hydrocarbons, their cell walls and such. Allow bacteria to metabolize the shorter-chain hydrocarbons. Helps if you have oxygen. And then use a settling agent to drop out the particulates. That's what clears the water and keeps it clear. If you're using one product, you can only be doing part of this. I believe that a lot of the enzymes that are put in actually work like algicides. Now, it makes them unlicensed or unregistered algicides, and that creates a lot of heartburn in the industry because nobody wants to have to go through that process, and I get that. But the bottom line is you've got to look at it as this 4-step process. And if you're only using one product, you're doing part of the process. Maybe you'll get results, maybe you won't. It's a little harder to figure. Okay, biomanipulation, near and dear to my heart. I made money doing my PhD work. My PhD research wasn't on biomanipulation, but I did it, and that paid for my PhD research. Basically, if you've got lots of big zooplankton, they're the cows of open water. They chow down on the algae, and they can filter it pretty clear, as long as the algae aren't things they can't eat. Smaller zooplankton, like this Bosmina or these Chidurus, eat a lot more. The power of filtration is related to the cube of body length. So being bigger makes a difference. Size matters in this. However, if the phosphorus level is too high, 80 parts per billion or micrograms per liter is usually thrown out, that's going to cause problems. You just can't overcome that much fertility. So you have to keep the phosphorus down to a reasonable level, but then the biological structure makes quite a bit of difference. Balancing the game fish with the panfish, with the zooplankton, with the algae, so that you're getting good productivity and not a bottleneck at any step is what you're going for. So that means now you need fishery scientists. You got to really look at your lake in terms of its fish community. This is the brand newest stuff I've got in here, and I'm really happy with this. These are lakes that have been treated with aluminum. They do not have phosphorus problems. These are like within the first 2 years after treatment. But if there's alewife present, they are eating all the zooplankton. I mean all of it. There's just like nothing there. The biomasses are really tiny. And as a result, even though the Secchidica is an awful, at least 2 meters, it's not nearly as high as when we have lots of bigger Daphnia out there. And does happen in the lakes where the alewife are absent. Same statement would go for lakes with gizzard shad in them and a few other things. Golden shiners can be particularly damaging to the zooplankton. If your planktivorous fish community is really dominant, you're going to have less zooplankton and you're going to have more algae per unit of fertility as a result. So if you can keep the fertility down and you can manage your fish community to get more big Daphnia, you will be doing way better. Rough fish, same deal. They stir up the bottom. Some of that becomes usable nutrients. They make turbidity, if nothing else. Controlling them if they get out of hand can be a benefit. And then finally, rooted plant assembly. You know, if it's really dense with rooted plants, you usually don't have algae blooms. Well, then you got another problem. You're going to get called in anyway. So getting the balances right, again, it's really more about nutrients. The biology is the icing on the cake. It's not the cake itself. And there's a few techniques that really didn't quite make the list. You know, you can't do circulation with the minivan. Stale bread is not the same as barley straw. And for gosh sakes, never mix Viagra with algicides. Okay. So the roll call for algae control, you know, watershed management's always job one, but it's not always going to get the job done by itself. In fact, it's rarely going to get the job done if you already have a problem. Phosphorus inactivation for internal load control or managing inputs can be really helpful. Circulation to 3 times the Secchi depth, if that's possible, is useful. Circulation, possibly with inactivators or dyes or bacterial additives, can do well in a shallower lake. Oxygenation, mainly for deeper lakes where internal loads are dominant. Dredging where it's feasible, especially for algal mats, but the expense and the permitting are just so difficult. Algicides with proper timing and limited uses. I would say that if you're treating more than once a year, it's too much. You really got to look for a way to control the nutrients. But if every so often you have a problem and you deal with it with algicides, you are not a bad person. This works. This is effective, and done right, it can be effective for most of the year. I've got lakes where we hit them in June and we're good for the year. We've stopped the problem. We didn't wait for it to get green. We hit it when it was starting. Sonication for susceptible algae, particularly where nutrient control is limited. Golf course ponds, manure pond, or, you know, farm ponds, things like that can be pretty effective. Biomanipulation, as long as the phosphorus is not too high and you don't mind some wide variability, you can get a lot for it. And of course, if your fishing's an interest, it all works together. And then any other technique as the scale and circumstance dictate. I don't like to see techniques bad-mouthed or thrown out. Everything has its place in the toolbox. Just don't put in screws with a hammer and don't try to put in nails with a wrench. Pick the right tool for the right job. And that is it. Only a few minutes over, not too bad.
Not bad at all. All right, so we're officially in bonus time now. So hopefully Anne is kind of getting set up with her microscope and sharing her screen. But we do have a couple of questions here, so we can handle those while we're switching things over. So the first question is, Ken, similar to your statement on Phoslock being maintenance, but could buy you 20 years. How many years do you think dredging could buy you?
It depends on what the external loads are like, but generally speaking, when I do dredging, I'm thinking in terms of a 50-year timeline. I don't think I would dredge a lake that was going to need to be done again in 20 years, although I've actually got one in Wellesley that's exactly what happened. They dredged it in 20 years, again, it's full, but it's basically a stormwater pond. But in terms of a typical recreational lake, I mean, if you take out all those nutrient reserves, it's going to take a long time for it to build back up again. You take out all those resting stages. In 1993, we dredged a lake in Newton for algal mats. They've not had an algal mat problem since. That's what, 27 years? You know, and again, I wouldn't expect them to have an algae problem for about 50 years. I wouldn't, you know, again, it's hard to judge, but it's a big timeline. Dredging is not something you think about doing every couple decades.
And asked by the same person, these questions, I do not believe they will be available in text form. However, we are recording the entire conference, so those recordings will be made available to you after the conference. So you will be able to go back.
Be used against us. Yeah. Okay.
Yes, exactly. Let's see. Next question. Yeah, this is going to be a great one. Of the algae control methods Ken described, which would you consider to be the most effective? That's not a loaded question at all.
Well, I gave them in order there. All of them have caveats. If you're giving me no other information, you're telling me I have a lake with an algae problem. What should I do? I can guarantee you results with phosphorus inactivation. I know I can make that work. It may not really be the best choice, and it may not last as long as we'd like, but I know I could make it work. But of course, you want to do the study, you want to evaluate. You know, well, it turns out it's a pond that has, you know, this ridiculous amount of development in the watershed, but it says this as one inlet. Well, we could treat it the inlet. Could we build a detention system? Could we infiltrate stormwater? Oh no, it's actually a fairly nice lake with very little around it, but it's got 2 or 3 little streams that come in, one of which comes from a dairy farm. Well, boy, I'd sure want to look at that real closely, that one tributary, before I made a decision. But in the lake itself, ignoring the watershed management, which is always job one, at least look at it, I would say that you start with, okay, how would I lower phosphorus? Do I need a one-shot deal to get rid of internal recycling? Do we need some kind of intermediate or maintenance program to take care of the inlet inputs coming in? Can I do it by oxygenating, which would also give me better bottom conditions for fishery purposes? Can I do this with some bacterial additives that allow me to get some control over other things as well? You know, there's a whole lot of things you look at, but that listing I gave is kind of the order I would look to. I would start out with the watershed, then I'd go to inactivation, then I'd go and see if circulation could help me, then I'd think about oxygenation. I would work my way down that list. I'd look at them all, but they're kind of in the order that I would look to what's most likely to work in the broadest number of circumstances.
All right. So we do have a couple of other questions, but I think maybe we want to start hearing about what's on the screen there, and then we can get to some of the questions while we're going through some of the live specimens. So let's have at it there, Ann.
All right.
Looks like a phenomenon to me.
Yep. And what I did was, and what I'm doing is taking pictures. So anybody who is interested, I'll put all the pictures once I get them labeled for taxa. I'll at least do them to the genus level. I'll put those and also up on WeTransfer for anybody who would like to download those who's watching. So this is a Phanosomnon, and I kind of have my slides set up from blue-green, green, and then I've got some diatoms and some other cool things to look at, just to kind of show you what I would look for. So this is kind of classic of Phanosomnon. Notice that there's no fascicles, there's no grass flakes here. Two reasons for that. Phanosomnon doesn't like to be contained. As soon as you contain it, those grass flakes are going to fall apart. And as soon as you preserve it, those grass flakes are going to fall apart. And so you collect it with multiple filaments together, but chances are 90 to 100% of what you see in your actual sample that you analyze is going to be single filament. And you know, as we look through the sample, this is really classic. So that refractive, when you're looking at phase, that refractive essence there, that's the aerotopes. And then you come out to the terminal cells, and they are way expanded, and they're all just gas vesicle. If I go to bright field, essentially what you're going to see is just sort of a grainy cell contents here and here. Those are the aerotopes along with cell contents, and then you have the expanded terminal cells on the cell. Does anybody have any questions about a Phanosomnon?
I will let you know.
Okay, so then what I'm going to do is show you this sample. This is much more typical what I get. And
Crappus, or is it Crappus lumpus? I always get the species wrong.
Crappus and Crappus lumpus. Yes, it is. So, okay, clearly this is a live sample. See all these bacteria? Not uncommon for a bloom sample to see a whole bunch of Aeromonas and other lovelies swimming around your sample. So what are some of the things that we can use to help us figure this out? Well, we know this is late in a bloom. All these cells have aerotopes, all right? And I've got pictures of that for you. So we're going to kind of look, and, well, we get some longer filaments. Oop, here's a longer one right here. So, and there's a longer one right there. There's a Cryptomonas swimming through here. So what this is it is a degraded Phanosomnon sample from a bloom that was taken at the height of a bloom. And again, it was already degrading, and they don't like to be contained. And so this was sent to me live, actually, from Illinois, and it was sent to me Friday. So this is an ongoing bloom. So it is not a great place to be in this particular sample, right? This lake is not fun right now. So that if you focus up and down, you can see enough hints to help you get where you need to be. There's some, you know, even though most of these are short filaments, 2 to 4, 3, 4 cells, you get some longer ones in there that tell you, that's what I got. I've got a Phanosomnon. Now, both of these populations are from lakes that have pretty high nitrogen, because we don't see any aerotopes, so, or any heterocytes. So the next thing that is most easily confused with a phanosomnon, although I have to stop here because it's just a super cool nucleus. You know, that's just fun, right?
They're cute.
So I know. So here is a interesting thing, not what I'm looking for, but interesting to look at. A couple of things tell you that this is degrading. This is Stereonella. It has choanoflagellates parasitically on the cells. And see how these are wavy and they're bent? That is very indicative of a senescing diatom population. So the diatoms, either it's been preserved in a sample that has too much iodine, but I know that the sample is not Algal's iodine, so the silica is not dissoluting back into the sample. This thing is actually dying, and so it's losing silica and therefore rigidity on its cells when that happens.
And when you're showing these, if you know the lake where they're from, I got some of the New Jersey folk are curious. If you can let us know what the site is, that would be great if you can.
Sure. This is from Cedar Lake in Minnesota. The previous one was actually from Utah Lake. So the long, nice phanosomnon was from Utah Lake in Utah. The degraded one, I'm not sure where it's from, but it is from a mid-Illinois lake. And then this one is from Cedar Lake in Minnesota. And then, okay, did I find what I was looking for? Yes, okay. This is the thing you're most commonly going to confuse with a phanosomnon. This is Planktothrix. Now the question is it Prolifica or Garrity? This is from that pink bloom, that rust-colored bloom that I showed you earlier. If I take it off a phase and go to bright field, now we could see got a slight pink cast to it. And the aerotopes are so numerous and so close together, it's hard to see them. The thing is that the cells on Planktothrix are about as long as they are wide, maybe a little longer, maybe a little shorter. And a phanosomnon,
if you were looking at this, you'd want to blow it up to a higher power so you could see it better. Just we know what the stuff looks like. It was you need calibrated eyeballs to tell when it's tiny like that on the screen.
Yeah, this is 400x, guys. So
right.
Yeah.
No, but I mean, on my own screen, I just blew it up and can see it way better. But if you're like working on partial screen or something, the size matters to be able to see it. Yeah.
Right. And unfortunately, I can't make it any bigger for you.
That is a classic look for it. Very straight.
Yeah.
Cells about as long as they are wide, with all those little bubbles in it that are making it float.
And the ends are slightly conical truncate. Yep. So, but you may have to look at quite a few filaments. Actually, the sun just came out. Let's make it a little hard for me. Here and here, slightly truncate conical. And what I want to try to do is find some more in here. Here's Cryptomonas, and a lot of them. So here's something, note to self. All these are Cryptomonases. And the problem is when you preserve Cryptomonas, it fires off these things called nematocysts. And I'm going to turn this a little bit more towards me so I can see it. And it makes the particulates around it stick together, and you get these huge clumps of Cryptomonas and crap. And that is very much a preservation artifact. And it's something that you just have to deal with when you're counting. So understand Cryptomonas is not colonial. And so it's not that these things are appearing in clumps naturally. This is a preservation artifact.
That appearance where you've got a clear area down the middle and sort of a cup-shaped chromatophore, it's really two of them around the outside, is very typical.
Yep, and I've got some other ones that are only or primarily Cryptomonas. I want to go to 100.
Ken, I'm going to ask you a question from your presentation while she's moving things around. There's a question. This is from Lou Feeney. Did you imply that if alewives are in the lake, that sediment inactivation or other control approaches are unlikely to be effective? Can you follow up on that a little bit?
No, I'm saying that they will not be as effective as they would be otherwise. No, in fact, all those lakes that got aluminum treatments have shown major improvement over before treatment. The point is that not all of them got as clear as we would have liked. So for example, Hamblin Pond, which I showed as my poster child for an activation, the Secchi disc there averages 8 meters. Averages 8 meters in the summer. I mean, it's just crystal clear down through there. Hinckley's Pond, which we just did 2 falls ago, that's one of the ones that's on that graph. It has major alewife run. It had huge amounts of zooplankton in May and a clarity of 6 meters. But by June, all the zooplankton were gone. It dropped to 4, it dropped to 3, it eventually dropped to about 2.7 meters by late August. So is 2.7 meters bad? Well, it wasn't. There were no blue-greens. It was all diatoms and chrysophytes. It wasn't bad, but there was nothing to eat them, and there's still some phosphorus around. So we built up some algae. So the point is, biological structure matters. If you remove phosphorus, you will get less algae, but you will still get accumulation of algae and lower clarity if you have nothing to eat the algae that are there. And alewife, along with certain other planktivores, really make a difference. The problem is most other planktivores were happy to get them eaten by game fish. The alewife is a big push in a lot of states, Maine included, where they're trying to restore alewife runs and bring more alewife back because it's great for the marine and the estuarine environments. But this is a real problem for the lakes they're going in if they haven't had alewife for 200 years. They're not adapted to this. So it's more like an invasive species in that case.
Great, thank you. Okay, back to Amy.
All right. Yeah, so here's another larger filament of Planktothrix, and again, it's either Prolifica or rubescens, and it's got that truncate conical cell at the end, and the cells are just a little bit shorter than wide here, but if you go down to the center of the filament, they're about as long as wide. So the thing is that in a Phanosomnium, the cells have to be longer than wide. And you never get empty end cells in Planktothrix. So I have another filament over here, if I can find it. So there's this one. Again, there's less aerotope, but there's cell contents all the way to the end. And this one over here. So here's one where this end is actually has almost what's called a end cap or calyptra to it. It's that conical truncate, but if you go to the other end, it clearly is wider and it does not have that. And that's pretty common for Planktothrix. So if you're chasing an ID and you're having problems, you really need a good 50 to 100 examples to be able to ID it well and to feel confident. If after 100 you haven't seen what you need to see, you could probably give up. It's probably not worth it to keep looking. Just because it becomes diminishing returns, right?
What I would point out is really worthwhile to learn to recognize Planktothrix and not just call it Oscillatoria, because the toxin profile in this thing is among the worst.
Yeah.
Most of the dog deaths I am aware of in New England have been a result of Planktothrix.
And it is, like I said, it's like microcystis. About 85% of the time, it's producing toxin. Now, it might not be always measurable, but it's a nasty one. I always get concerned when I see Planktothrix.
And because it does tend to grow metalimnetically, you often miss it if you just take surface samples.
Well, and they were in Cedar Lake, in Nokomis Lake as well. They were having these blooms of Planktothrix and didn't know what it was because I quit counting at the end of December before the lakes ice over. And they were ice fishing and collecting collecting fish. So
yeah, it can go any time of year.
Yeah, so I would be very concerned if somebody were ice fishing on a lake with a Planktothrix bloom in winter without a toxin test. So this is one of those circumstances where, you know, having an underphase is a problem because this thing is so dense. So if I go to Brightfield, it's pretty obvious pretty fast, right?
Yeah.
So and part of the reason it was so hard is because this is a Dolichospermum lemmermanii, and it's got aconites all through the center of the colony, which make it very dense. So, and it's so dense, I have another set of this because I can't always find it. I can't see the heterocyte aconite pattern, but I know it well enough to know that this is.
Yeah, when you get that batch in the middle, and this one is very common in the NEAPMS zone. In our region, this is a very common one that will sit on the bottom. It will pop up anytime from late May to October, and just the bloom comes out of nowhere. And if it's been there for a while, it's going to come up with stalked ciliates and everything growing on it.
Yeah, I love the
One of the
vorticella
ones
on it.
treat with copper in a weighted hose or a chilled solution, you can kill it before it ever comes up.
Okay. So,
Question for you, Anne, while you're on the topic of Aphanizomenon. This one's from Anne. Let's see, does Aphan ever have spherical or roughly spherical heterocytes, or are they essentially always more elongated in shape?
oh, I know why you're asking that question. Generally, they are longer than they are wide. If you start to see rounder heterocytes, chances are you have a mix of Sphaerospomopsis or Chrysosporum and Aphanizomenon. So there's Aphanizomenon was split into a bunch of different genera. Aphanizomenon, Cuspiditothrix, which has the kind of telescoped terminal cells. Sphaerospomopsis, which actually has a aconite heterocyte aconite pattern. But sometimes, especially, and I have a slide, I don't know how good it is, but I have a slide of Sphaerospomopsis Aphanizomenoides that has an ovoid-ish heterocyte, and it was originally in Aphanizomenon, spent some time in Dolichospermum, and now they've realized, oh, it's its own genus, as well as, and then Chrysosporum was also split off from Aphanizomenon.
And there were pictures of all those in the taxonomic lecture.
Yeah, yeah. Yeah, so it can, if it starts to appear roundish, I would get suspicious. Now, sometimes they can be a little round, but they are always longer than wide. If you see a round heterocyte, it is not Aphanizomenon. So this, one of my very favorites, is Gliotrichia. And it's a big one. It's a big colony.
Yep.
So, and it turns out that he's not alone. So there's this huge colony of Gliotrichia. It's not been grazed. It comes out in really nice thin filaments or trichomes all the way out. And if we look at it under 400, it's hard to look at because it's so dense, but these are all heterocytes in the center of the colony. And then they go to these long filaments, but there's also Dolichospermum mixed in with it.
Oh yeah, tucked in there.
So pretty interesting. And honestly, I haven't found any heterocytes or aconites on this thing. Oh, there's some heterocytes. I can't ID it without the aconites. So it's got heterocytes, but it doesn't have aconites. So there's a lot of things it could be. Subcylindrica kind of comes to mind, maybe. I don't know. I'd have to have an aconite of some sort, but you could tell
it's a Dolichospermum, and that's probably good enough for starters.
Yeah, I wouldn't try to take it past that, honestly, because it's not worth the effort. Unless I have to. So now you can really see how that's intertwined through these filaments. Yeah. Pretty cool, actually. And if I go into the center of the colony and I go on bright field, now I can see all these heterocytes and a little bit of aconite action there in the background. So definitely an older colony. It's easier to see. Now here's one of those Dolichospermum colonies all by itself. Might be Flassaque, but without an aconite, I got no idea. So I have to actually, these things are huge, so in order to place them, I have to take it back there we go. So the interesting thing is, if I take this and put it on phase, you can actually see that there's those all the way intertwined, all the way circularly around this Gliotrichia colony. And this one has had some grazing. Pretty cool stuff. And Gliotrichia, like I said, is a boom or bust. It can produce toxin, not a very reliable producer. So if I see Gliotrichia, I'm mostly just going to go, oh, cool, I love that, and not be super concerned.
But there have been issues with it in New England, particularly Maine and New Hampshire, where they get blooms of it that are dense. Well, we didn't talk about it much. When you get the stuff rising to the top, then the wind can concentrate it in coves and such. And that's often a major effect. And even though most of the lake is okay, that area where it's been concentrated is a mess. If it happens to be a toxin producing form, now you have a health risk.
Right. It takes a lot of gliotrichia though to
Yeah,
produce merciful
gliotrichia is less
toxin.
of a risk than a lot of the other ones. But
Right.
then, of course, you saw you had a Dolichospermum mixed in with it.
Mixed in with it, exactly, just like it was with that Magotia. Yeah. This is Sphaerospomopsis. I know that because I did the sample quite a while ago, but most of the filaments do not have heterocytes and don't have aconites. So when you have something like this, it tends to be more constricted. That's how you kind of separate it from a phanezomonin. And it tends to have a longer than wide heterocyte. That's most of the Dolichospermums have a round heterocyte, but you know, see how it's kind of this end cells, terminal cells tend to be a little smaller than the main cells, vegetative cells. That's also a Sphaerospomopsis thing to do. When you see it, it's really hard. And Sphaerospomopsis has been in a phanezomonin, Anabaena, and Sphaerospomopsis. Now here's one that is clearly Dolicho. And again, I don't have heterocytes or aconites, so I can't tell you which one it is. There's a handful that are in this size range that that you could figure it out. And those are generally
Point out, you've
round.
been going by a couple of denobrians. There's another one there.
Yep.
Let them make sure they notice those. The wine glass one.
Yeah, I've got a really nice sample with a ton of live denobrium
Okay.
in it. Yeah, so that's probably social. And there's also Planktolimby in here, Limnetica,
Oh yeah.
and
Yeah,
it
that's
is
important because
tiny.
it's really, it's the kind of thing you would blow by and not pay any attention to it. If
Yeah.
it's one or two, fine. But if there's a lot of them, you need to know
If
it.
it's a lot of it. And if I start seeing a lot of Planktolimby limnetica, then I'll start looking along with Dactylococopsis, I'll start looking for Rhiphidiopsis. They co-occur pretty predictably. And here's another Sphaerospomopsis. So the hard part about Sphaerospomopsis is that, especially Phanizominoides, a very predictable toxin producer as well. And probably when she was asking about, okay, what about, here's a whole bunch of, what about the round heterocyte, or, you know, are they always, you know, elongate or kind of cylindrical? When you start seeing rounder heterocytes, chances are it's going to be Sphaerospomopsis. And this isn't giving you really any help at all because we don't have heterocytes or aconites.
Yeah.
So I can guarantee that even though I'm confident because I did this sample before, I would call it Sphaerospomopsis. I know for sure that Andy Chapman would call it Nastocales. I know for sure he would. He's very conservative.
What would Andy call it?
He would take it all back to order. He would take it completely back to
Okay.
order.
Okay.
Yeah, I don't feel the need to do that. But so here is somebody before had asked about Limnorhapis. Limnorhapis is a bugger because it facultatively makes a sheath depending on how pissed off it is. And this one has a sheath and this one is just developing the sheath. This one doesn't have a sheath at all. So it's the most planktonic of the Limbia group. And the Limnorhapis, this is interesting too. So if you looked in the keys from a year ago, 2 years ago, they would have Limnorhapis as a genus and they would have it as non-toxin producing. They no longer have it as non-toxin producing.
Yeah.
So it produces microcystin and it produces some other toxins. It actually produces quite a few. So depending on how many erotopes it has, it's either darker or a bit lighter. Cells are always shorter than long. Sheath is facultative, it's not obligatory. So in Lingbia, the sheath is obligatory, which means it must have the sheath to be Lingbia. And then here, interspersed with this, which is very common, at least in my area of Southwest Michigan, I often see Microcystis viridis or Microcystis with Limnorhapis. It's a very common association. And 2 years ago, here's a Hormogonia. 2 years ago, I went, gosh.
Wait, a hormogomium is just a bunch of cells that have created a resting stage and drop down.
Yep. They just kind of collapse in on themselves and hang out. And if I kind of increase this, you can see these, they're dark brown because they're full of erotopes. Now, whether this is actually aeruginosa or not, who knows? You know, if we go with that thing, oh, well, all of these are the same thing. But generally, viridis is packeted like this is, and the cells are slightly larger than what we would call nominate Microcystis aeruginosa. And then this is a nice medium-aged aphanosomnid right here with a nice cylindrical heterocyte, and these cells are starting to elongate and fill with erytope. And then,
if I can jump in real quick, question for Ken: is Lingbia controllable?
Well, okay, when you say Lingbia, again, true Lingbia is mostly a benthic species that makes mats. It is controllable if you get to it early. Once it has formed a dense mat, it's extremely difficult to kill. But if they mean all these extended sheath versions like plankton Lingbia, Limnorhapis, all those, most of those, if they're loose and in the water column, will react to copper or peroxide.
Great.
now, especially
Lingbia, in the classic sense of the benthic Lingbia mats, pretty hard to control, just like the greens with Cladophora, Rhizoclonium, Pithophora.
and
Microsyra.
yeah, Microsyra, yeah. So there's some aphanosomnida mixed in with this. The first time I found this in 2014, it was producing a ton of toxin, around 11,000 micrograms per liter. But when I sampled it last fall, last summer to go to a workshop, so August of 2019, same lake, same assemblage, no toxin. So, and this kind of shows you what happens when these cells start to go wonkers inside the sheath.
Yeah. The Limnorhapis it's actually pretty common in the Northeast, but it's not it's rarely a bloom former. Last year or the year before, one of the lakes in the Berkshires, I think it was Pontusic, had a bloom of it. It's the first time I'd ever seen an actual bloom of it where it was pretty dense. You know, physically see it in the sample. Usually it's like if you a net tow for zooplankton, you'll frequently see one or two. But your whole water plankton sample that you've sedimented or concentrated, I don't see it all that often.
Yeah, it's I have seen it quite a bit out west actually. It's a big filter clogging problem. But it doesn't I haven't had a whole lot of issues with it unless it's mixed in with like that microcystis and the other. So this is an interesting one, and I'm going to take it off a phase. So okay, I've got this big colony. So here's how I would go about identifying this. So the first thing is that yes, this is a pretty big colony. I'm only at 400x, and it's taking up the entire field pretty much. So we have a couple of choices. The first choice is a green. The second choice is a chrysophyte. The most obvious chrysophyte would be Uroglena. So the most obvious green was.
Explain why it wouldn't be a blue-green though.
It's not going to be a blue-green because of several things. First thing is these cells have flagella. See these? So these cells have flagella. It cannot be a blue-green. They have structure. They have, you know, there's pyrenoids in these cells. They're connected to each other. Yeah, so there's structure there. And so that's how I would know it's not like something like a Phainocapsa or Microcystis, right? There's some regularity to it. Another thing that tells me it's probably not going to be something like a Microcystis. Cells are really not round. They're kind of they're round-ish, but they're clearly not super round. That's because they have connection from here to here to here to here to here to here. The cells have two flagella that are about equal. Can't be Uroglena. Uroglena has subequal flagella and can't see the pyrenoid because it's chrysophyte. And the fact there's little tendrils, this is Volvox. Pretty unusual to see Volvox in an open water sample in a big lake. This lake is 900 acres. So kind of fun because normally you wouldn't see this stuff in midsummer. This sample was taken in August. So yeah, pretty cool actually. And other than that, that was pretty much what I wanted to. Now it's not uncommon too. So this will make ID frustrating because there's a little dinoflagellate in the middle of this microcystis colony.
But it's important to compare that microcystis to the Volvox, just in the overall visual image. They're very different. The spacing, the organization of that colony is very different than in the microcystis.
And it has
as you focus
flagella.
up and down. Yeah.
Yeah. It's got flagella. That's actually a daughter colony starting in there. The cells have structure. You can see cell contents.
The
They
cells
have
are all on the
flagella.
outside of this globe. You can see them in one plane. The microcystis is scattered throughout.
Yeah, the cells are peripheral, which they can be in, and this is where phase really comes out. Wash this stuff out and you can see these peripheral flagella, and there's 2 equal flagella.
Yep.
So it can't be a blue-green because this thing is motile. Whereas you come over here, and here's our embedded dinoflagellate who probably just got caught up in the sheath, and microcystis cells that are roundish, kind of no cell contents per se, other than to look at some of the erytopes. Has a sheath that's sort of roundish and amorphous, doesn't have any real structure. Okay. And this is also meant to confuse the crap out of you. Just because I'm that kind of lady. And this consistently happens to me all the time.
Confusococcus obscurum.
So I'm going to take it off phase, put it on bright field. Come on, behave. All right, there we go. All right, doublets. These are single Veronica cells.
Yeah, popped out.
Common in a Lugol's preserved sample. So that sample I just showed you was in glutaraldehyde. So the microcystis stayed intact, but often in glutaraldehyde, microcystis, or in Lugol's, microcystis will do the same thing. It will fall apart. And every once in a while, you'll see a few cells that are still hanging together. Not very many. So the original colonies probably had hundreds of cells. But most of these are single, and then every once in a while, you'll get a pile. In the original colony, the cells are peripheral, and they are going to be kind of like a gumdrop, kind of like sugar on a gumdrop. And the sheath is so diffluent that they'll shoot out of the colonial sheath, and you can actually watch them. So if you get a sample like this, it's either degrading or it was preserved in Lugol's iodine probably a fair bit ago. So let's look at some other groups then. The samples that got sent to me from New Jersey are fun, and I'm going to get to those in just a second.
While you're doing that, quick question for you, Ken. This is from Angela. If you're only going to measure the internal P load of a reservoir once, what is the best time of year to conduct your sampling?
Well, there's more than one way to do it. If you're going to do it based on what's in the sediment and some estimation about what will be released, the timing doesn't really matter. The mass that's in the sediment will still be much more than whatever got released into the water column, so you could do it almost any time. I like winter because most of it is all stuck in the sediment and it's easier to work from the ice up here. But if you want to do it by the accumulated amount of phosphorus in the bottom of the lake, if it's deep enough to stratify, doing it in late August or early September will have the highest accumulation. And then you can compare the top to the bottom or the bottom to whatever it was back at the start in May.
Great, thank you.
This is degrading Spirogyra. So the chloroplasts are not real tightly wound. I will include a couple of nice Spirogyra pictures. We didn't collect any nice, pretty live stuff. And I might have a really nice Spirogyra sample, but it's in my office and I'm banned for another 10 days. So anyway, when it gets older and starts to senesce, the spirals unspiral. And so they don't look as pretty. They look further apart. Sometimes they completely unspiral and you just have to kind of look for a few. Here's a pyrenoid, here's a pyrenoid, here's a pyrenoid. He's just not happy. It's the end of the season. This was a late summer collection. So I just kind of want to show that to you. And, oh, you know what? I forgot my microsyra. So this is microsyra. And what I want to do is also have my limnorhapis again. I'm going to go back to 400 so you can see what the big size difference is. So microsyra, which also the species name is wolii, also Plectonema and Lingbia, has a laminate, really thick sheath. So, come on, behave. So this whole thing is the filament, and this is the trichome. And it's easier to see if you go on to Brightfield because it is so thick. So this sheath is just gargantuan, and then individual cells that are only a few microns deep. And the end of it looks pretty nondescript. Actually, tends to get a lot of epiphytes, leptolingbia and a lot of blue greens and edegonium, especially like growing off this stuff.
And again, this grows as a benthic mat, but will pop up and get into the water column late in the growing season.
Yeah, and it can be hard once it gets to this this growth stage. It's hard to control.
Yeah, well, almost all the major mat formers are really tough. This one, though, because of the thick sheath, you can't even get at it.
And then sometimes it'll have some greens that are locked up in it.
You got some edegonium on there, it looks like.
Yeah, edegonium is kind of the thing that it's nothing else.
Yeah.
Because there's actually growth rings there that are hard to see, and there's growth rings here.
Yep.
It tends to be asymmetrical. It's a green algae. So, and here you can see one where it's definitely starting to come apart here. So see how wide this is. So this is a little over 50 microns wide. If I take that off and put on limnorhapis, of course, see how much thinner limnorhapis is? It's around 18 to 20, much narrower. So that was microcyrus. And then I'm going to show you, I'm going to go to the samples that you sent me. I'm going to start with B, and B was, I'm going to just butcher this, Chris. Manasquan?
Manasquan, yeah, you got it.
Yeah, Manasquan. I did a paper once with Ken on Lake Pocopatog. I swear I practiced that name in the bathroom for like 10 minutes. Lake Pocopatog. Okay, so this is beautiful actually. What this is Tabellaria and probably flocculosa, because I don't see too much fenestrata. So here's something to look for. So I'm looking through this sample and I'm seeing all this beautiful Tabellaria. It's a diatom. You can imagine they just collected this, so lakes are broken down. There's a fair bit of turbulence. Lakes are technically isothermic, so lots of stuff to keep this up in the water column. So you know, it's big and it's beautiful. And then I run across this. That right there. And that right there is an aconite, Dicospermum aconite. And then I ran across this. And that is Phanezomenon. So when you're looking through this stuff, what you can find is not only what is dense, and this Phanezomenon actually is sticking, it has a buddy, and they are moving a little bit along each other. There's no heterocytes, but you can tell that the end cells are, you know, there's erytopes there, but it hasn't really inflated out here. Winter blooms of a Phanezomenon are very, very common. And this, although I have better ones from the other sample you sent, that is Algacyra. That's Algacyra, which is another chain diatom. Most of this sample was this beautiful Tabellaria. And so I know that the population is super healthy because it doesn't have a lot of epiphytes, a little bit of stuff going on right here. But for the most part, cells are chock full of pigment. They're very healthy. The colonies are large. So it's really nice. And one of our favorites, yeah, Desmid.
That's one cell, but it has two symmetrical parts.
Yeah, and they also split star astrum into several genera, and one of the defining features is whether there's teeth on these legs, which there are. So this one along Paradoxum, there's like 1 billion star astrum species, but this one stays in star astrum. It doesn't get moved. And the nice thing about Tabellaria flocculosa is that I'm pretty sure it's not been moved in my lifetime, which I greatly appreciate. At least one thing in my life has been consistent, and that is Tabellaria.
These are pretty easy to filter out in a water supply situation because there's it's a chain of cells. However, when they get really abundant, they can often clog the initial filter screens.
Yeah, because these things will interlock.
Yep.
Yeah, they create quite a problem. They'll interlock with each other. They're beautiful to look at. But just like Fragilaria cruentensis, which there is some in here.
You got a couple rotifers there.
Oh yeah, I've got some nice Polyarthra vulgaris and some
at the bottom and Caratella.
Caratella. So also remember that that aconite I saw.
Yeah. So
what's not the same, right? And if I go to Brightfield, those are going to look nice and brown for me. Look at that, almost black. And they're looking that way because they have aerotopes. So they're chock full of aerotopes. Again, there's a number of species that this could be. My best guess is based on its size, that it's Planktonicum. But it could be Smithii as well. There's 2 or 3 that are in this size range. So really nice though, very nice. And it's got a lot of aerotopes in it. But notice this is a winter sample, so it does not have heterocytes, and the aconite we did see was a free aconite. Again, here's a shorter.
I have a question. Can Tabellaria blooms be visible in the water column with the naked eye?
You'd see a discoloration of water. You wouldn't see clumps. It'll make the water brownish, but those are not so big that you can actually see them in the water. I think if you got a glass and held it up, you might see the filaments, the colonies in it to some degree, but just standing there looking down on the water, it's just going to make it look brownish.
And here's one that is a little confusing. So if you only looked at part of this, right, it would look like a Stereonella. But what it is diatoma. It's Diatoma tenuis, which has a growth habit kind of like a Stereonella, but it branches off and does this zigzag thing. And you have to get a pretty good look at it to know that. And then here's another Phanosomnon with some nice aerotope action going on here. And again, really common in the winter to not find it with heterocytes. And of course, he's pretty happy, so there'd be no reason to find it with aconites. So that was the main one from that one. So, and then this is from Glendula. And I believe Glendola. This was, so the other 3 were you can see my video, right?
Yes.
Yes. This is the sample. So this is pretty brown. That's about what that diatoma or that Tabellaria would probably look like. The water's pretty colored.
Focus it a little, Ann. It's out of focus.
Oh, sorry.
There you go.
Yeah. So this sample that we're looking at now, and I'm going to put it on phase real quick here. But this sample is this sample. And see how brown that is? So the other bottles, unfortunately, are amber bottles, but they probably would look about the same. So your question about would you be able to see the Tabellaria, you'll see the color in the water. This is all Alacosira. And probably, I think the species, at least there's a fair bit of it in here, is ambigua. I don't see any, you don't always get the nice, beautiful spikes you get with granulata, but there's indentations that you capture on some of the cells, and there's a fair bit of width difference among these filaments, which is also kind of typical. But when you see them and they're kind of not very happy, you could tell that you could see there's little U's here. That has to do with a structure that's hollow in between the cells, And that tells me that these are mostly ambigua, if not all ambigua. And there's a lot of, when they're healthy, you can't see the individual chloroplasts, chromoplasts, because they're discoid and there's a ton of them. Here you can see them. Here's one,
Yeah,
here's
the
one.
lower
Here's
left there's got two really nice
Yeah.
cells in focus.
one. Yeah. So, but if it's super healthy, like this guy is over here, there's not much you can do to actually see those individual chloroplasts.
Yeah,
So
just
he's
be careful
actually,
when you're looking that you don't think this is some sort of filamentous green. The color is totally off, but it does look, you know, it's very much little barrels stacked end to end. But without seeing that structure, you might think, oh, it's some sort of little filamentous green, but it's not.
right.
They all
And
have
this
sort of bow
sample,
tie look to the chromatophore too, in many cases.
right. And this sample is not, you know, this is a dense sample. This is a really dense sample. So it's beautiful. And it's pretty typical for what I'd expect. So it doesn't surprise me. Now you can see quite a few of those individual discoid chromoplasts. So the thing is that it doesn't surprise me to see either the Alacosira or the Aphanizomenon in these lakes, because those are two common winter samples. Now there's also a fair bit, so here's more Aphanizomenon, and what's hard to show, I'm gonna go back to phase. See this right here? This is a long pinnate diatom, it's ulnaria. Sometimes
Which
they
for
can be
anybody
super.
that's had any background before is what we've called Synedra for many, many
Synedra.
years.
Yep. And they keep moving more and more taxa out of Synedra. So now there's only like four left, I think.
What is it called now? Or
It's
what is the
ulnaria.
most common one called?
Was Synedra ulna, now it's ulnaria what?
Ulnaria.
Lovely.
Here's another diatoma. Oh yeah, it's a pain in the ass. And there's definitely one I want to show you. And I'm hoping I can find now. Yeah, okay. So here's the other thing you're going to see. This is not from your samples. This is from another sample. It has a fair number, actually, that is Chlamydomonas that dropped its flagella. Super common in preserved samples. This is preserved in iodine. And look at our, this would have originally been in Synedra as well. These are now in Fragillaria. See how these are bent? You got two options. Look at that. So the first option is that, see here, see, look at that. It's something called aberrant. So if there were a chemical in the water, it can do that. It can cause these things to grow wonky, and it's actually an indicator. More likely though, this sample was preserved in Lugol's iodine. And Lugol's iodine scavenges silica because they tend to be acidic, and it scavenges silica out of the frustral, and voila, you get all these bent fragillarias all over, and Synedras all over the place. This is another nice little Chlamydomonas without flagella. You got no flagella. So I just want to show you what that looks like and how I know it's Chlamydomonas and not something else.
Real quick question, is Alacosira and Melicera synonymous?
No,
Yeah,
but
no.
Alacosira got split out of Melicera.
So
Okay.
Melicera is benthic for the most part and does not have markings on the valve wall. And Alacosira has both benthic and planktonic members, but it has puncti all over the cell walls.
Yep.
And here's three of these.
Melicera is mostly Melicera varians, which is very easy to recognize.
Yeah.
Alacosira's got a bunch of different, maybe 6 or 7 that are pretty common.
Yep, really big. And this is Pseudanabena limnetica. Teeny tiny. But those fragillarias are really what I wanted to show you. So if you're new to the system and you don't know what was wrong with the sample, your choices are it was degrading, it was the end of the season. This is Pandorina, by the way, or it's a preservation effect. I don't see that effect very often in glutaraldehyde samples. I see it
I've
all the
not seen
time.
that affect much at all. Again, most of my samples are glutaraldehyde though.
Right. It depends on the time of year.
Was that Pandorina you had on there before?
Yeah, that was Pandorina.
Okay. It's a colonial green. It's a bunch of chlamydomonases that cohabitated.
Yep. Yeah, it is. The cells are pyriform, so they're kind of cool to look at. This is a dinoflagellate, a thecate dinoflagellate, and I have a couple choices, and I have to look at it a little closer. So it's either Paradyniopsis umbinatum or it's Paradyniopsis quadridens, and I think it's umbinatum. I don't think that's enough of a peak on the cone to be quadridens. But I'm out of practice.
Somebody wants clarification. What was that name of the new name for the Synedra? Again, with a U.
Oh, it's
Well,
the big one
was
is
Ulnaria.
Yeah.
A lot of them have got, there was an initial move to say Synedra and Fragillaria are the same thing, just the ones we see as Fragillaria become colonial or filamentous. And then they decided, well, there's more to it genetically. So what we've called Synedra appears to be getting torn apart and thrown into other things. And that Synedra ulna, which is one of the really big ones, can be a couple hundred microns long, became Ulnaria. I don't actually know why. And so they just took that one out on its own. Again,
Yeah, and
my taxonomic form has Synedra and related taxa. I don't bother to split it.
So here is a nice one that doesn't have cell contents. So you can see it's pretty beefy. The ulnarias are pretty beefy, and it has to do too with the striae that go across the center of the cell, and there's a big central area in the center of the cell.
Yeah.
There's a couple, so there's that one in here. Then there's a bunch of smaller ones. This is Nietzsche. It's symmetric this way, so you can fold it over on itself. And it's common to see a certain number of the Nietzsche's up in the water column. And all of these little guys are little chlamydomonases that have dropped their flagella. Yes. And there was something else I saw in here I wanted to show you. Oh, here's a nice Nietzsche.
Yeah. The way to tell the Nietzsche for the most part is it has two chromatophores and they are separated longitudinally. They're along the long axis that they're separate. Most other diatoms have them running side by side. You can separate the Nietzsche ones by cutting across the short axis, by cutting that.
Right.
The other ones, they tend to be along it.
Yeah, here's another one.
Physical features, if you could see the ultrastructure and all the punctae and the way the raphe set up, you would be able to tell as well. But just at this level with the image you've got, you can tell it's a Nietzsche because of the alignment of the chromatophores.
Yep. Pretty cool stuff, actually. And if I come here now, you can see that.
That one shows up a lot in the water column because it has a raphe, which we've always sort of assumed helps them move. Well, it doesn't help you much if you're in the water column. There's nothing to move against.
It doesn't, but they move all over the place.
But they're there, yeah.
Yeah, they're there.
Pretty commonly there. Again, not a typical bloom former, but they can be pretty abundant.
And then one of the last ones I want to show you is this. And if I take it back to actually, it's hard to capture these well because they're kind of 3D.
Yeah, look at the edge. It's kind of fuzzy.
So all the way around this edge. This is Cynura. And it's almost impossible to speciate Cynura unless you're at 1000x and can look at the scales.
Yeah, Pete Cyber, who's at Connecticut College in Connecticut, is like one of the real experts on these. And I don't think he'd recognize one at full size. He only looks at the scales on electron microscopy.
Yep. So you can see, you can tell that it's different from the Volvox that we saw for a few reasons. One, it has scales and spines. It's flagella, which you can only see a few of them like poking up here and there. The flagella are unequal to the point where you almost can't see the second one. And in Volvox, they're equal. And then here's a Phanozomenon hanging around with them.
If you break them out, you might have to see it on phase. They're really not round. They're sort of teardrop shaped with a long, which is what it's an attachment that binds them together the
Yeah.
posterior end.
Like this one, and like all chrysophytes, they have a large central vacuole. So they tend to kind of look like they're bifurcate. So you can actually, it's hard to capture it. You can see it comes down to a point here, and then there's that large central vacuole. And if there's another colony over here that has some as well. Big
And
taste
again,
and
they
odor
can
issues.
fall apart, so there's nothing left but those sort of teardrop shaped
Yeah.
cells. This
It's
one's
actually
pretty nice.
impressive. Yeah, you sent this one to me, Ken.
Is that the one I sent? Yeah, it's one of the best ones
Yeah.
I've ever had for
Yeah.
them staying together.
Why it stayed together, who knows? But and there's
I
some
don't
of the
know
fruit
either.
up there.
Glutaraldehyde. Yeah.
Yeah. So yeah, so it's pretty cool. We don't often get to see that. And then here's some more typical Aphanosomnin. Elongated, empty terminal cells. Let's see. Oh, that's
It is 5 o'clock. I just want to remind folks that if folks are going to head over to the social and the aquatic plant quiz, that is a different link. That link has been emailed to everybody around 4 o'clock. It's also in the conference handouts. If you click, there's a little tab on the side here that actually has handouts. It's in there. It's also in the agenda. So the social will not be happening in this room. So if you're looking for that, you need to jump over to another link.
And far be it from me to stand in the way of drinking time.
Even virtual drinking.
And this is dual lake, just for those of you who can't see it. I actually do have to go because I have to go bail out my daughter and take my grandsons home.
Well, thank you.
They started school, but they didn't open up her daycare. So we have been doing double duty, watching small children this week.
I have to go because somebody has to open up the bar.
Well, I would like to extend another thank you to both of you for your expertise, for talking for 5 hours straight, basically. You know, it's overwhelming at times, especially this is condensed down from basically an 8-hour kind of workshop. So, but thank you both again. You know, I'm sure we're going to get a lot of positive comments from this. And you know, you just completed something off my bucket list, so I'm good.
You should do it in person. It's so much more fun in person.
Yeah, I know. I need to redo it in person. It's been too many years since I've actually done it. So once we start doing that, so thank you again, and thank you for everybody hanging with us. We still have over 60 people here. We will see hopefully lots of you people at the other link in a few minutes, and then hopefully we will see everybody tomorrow morning at 8:45, 8:30. I don't even know, 9 o'clock.
I'll try to join you guys later. I'll get my grandkids home and go grab some proper 12.
8:45 tomorrow morning. So okay, thank you everybody.
Very good.
You're welcome. Thanks,
you all
guys.
for hanging in there. We love talking about algae. Take care.
Thanks, Ken.
Yep. Bye.
Thanks, Chris. Bye.
Bye.